Hi everyone, am I audible? Hi everyone, am I audible? Okay. Hi everyone, how are you doing? Everyone, how are you doing? I believe I'm perfectly audible and visible. Yes, yes. I believe I'm perfectly audible and visible. Okay, that's great.
Okay, that's great. So, welcome to
I'm your biology educator, Ambika Sharma, and the chapter that we are going to discuss today is molecular basis of inheritance. So, in the last marathon, we have already completed principles of inheritance and variation. Today, we will discuss about the molecular basis of inheritance. We are going to finish it today. After that, the next chapter that we are going to start is sexual reproduction in flowering plants. So, sexual reproduction in flowering plants, human health and disease, we are done with the disease part. Studio is working fine. So students, I will put the video for the immunity and the drugs. So basically, that is going to be for around 3 hours. So you guys can watch that. So with that, the reproduction part is done. Only reproductive health is pending. That we will complete later because that is not a lengthy chapter. After this, we'll complete sexual reproduction in flowering plants, and next weekend both the chapters of biotechnology in one session. Voice is not fine. Okay, just a minute. Let me check. Now I believe it's fine. Right? Now I believe it's fine. So all I'm saying is that next week on Wednesday, we'll complete sexual reproduction in flowering plants. I'll put the video for the rest of the part of the human health and disease because now my studio is working fine. And next Sunday, biotech, both the chapters in one go. Done? Both the chapters in one go. So that is what we are going to do. Okay. So I think now the voice is fine. Yes. Now it's clear. Okay. Done. So guys, so this is one of the most important chapters, right? So from this chapter, you can easily expect 5 to 6 questions minimum. It can even go for 7 to 8, it depends. So 4 to 5 questions so we can get from this chapter. So 4 to 5 questions, if I take 5 questions means 20 marks. So this chapter is going to be very important. So let's discuss that. So how are we going to approach here? We'll start from the basic part, right? We'll start from the basics, of course. You know that is the speciality of this Game of NEET series. So we will start from the basics, right? And we'll talk about about the nucleic acids, right? About the nucleotides. For that, we will definitely discuss what are the nitrogenous bases, the sugars, right? The phosphoric acid, the bonding part. We'll complete everything. And then after completing all that part, right? After completing all that part, we'll talk about the structure of DNA and all the experiments. Okay? Done? So this is what we are going to discuss. Right, this is what we are going to discuss. So, as I said, I'm just telling you the minimum number of questions from this chapter. So, let's get started. So, tell me, from this chapter, which topic you guys hate the most? I'll keep it in my mind and I'll make it more easy. Which topic you people hate the most? That, ma'am, this is the topic we hate the most from this particular chapter. Quick. The one that you hate the most. Yes. Quick, everyone. Which chapter you hate the most? Topic rather. The whole chapter. No. This is a very good chapter. Trust me on that. It is amazing. It is amazing. And what is amazing about today's lecture, that you will also get my handwritten notes, right? You'll also get my handwritten notes, so you better focus on understanding the topic. So we'll start with the term known as nucleic acid, right? This is what we are going to discuss first, that what exactly are the nucleic acid. So students, when we discuss the biomolecules, okay, when we talk about the biomolecules, so basically in simple words, if I have to define, so what will I say? The things which are present in a living body, the things which are making the living body, this is what I'm going to say. If you read the NCERT says that all carbon containing compounds are basically making the biomolecules. But we know that it is not just the carbon containing compound. There are other inorganic things as well. So that is what we discussed when we talk about the biomolecules. And if you remember, we conduct an experiment there, right, where we discuss about the chemical analysis. And when we discuss about that chemical analysis, you remember, we have to take any living tissue. Let's suppose you are taking the rat liver, right? In the mortar and the pestle, you'll put it. Then you will put the acid, trichloroacetic acid. Do you remember this part? Then we have to put what? We have to put trichloroacetic acid. So what is the acetic acid? What is the formula for that? That is CH3COH. Am I right? So if it is trichloroacetic acid, it is going to be CCl3 and COH. Right, so we have to put this acid there. Then obviously you'll use the mortar and pestle. Then we are going to make a slurry. Right, slurry is like you are just putting the pressure there. You have the acid there. You're going to get a kind of paste there, the slurry. So when we filter that out, right? When we filter that out, the filtrate part, right? When you say this is the filtrate, and whatever will remain in this cloth, we'll call it as retentate. So when you talk about, after this experiment, when you talk about this retentate, here in the retentate, we discuss that bio-macromolecules are present, isn't it? Bio-macromolecules are present except lipid. So lipid is present there, which is not the biomacromolecule, which is the biomicromolecule, right? But this is what we see. And in the filtrate, what do we see? We see that there is the presence of biomicromolecule. So what are these biomacromolecules and what are these biomicromolecules? So when the weight is less than 1000 Dalton, then this is going to be biomicromolecule. And when the weight is above 10,000 Daltons, then we'll say that's the biomacromolecule. And here we'll see polysaccharide. Remember, we will see nucleic acids, of course, which we will discuss today in detail. We'll also talk about the proteins, right? And here we also see lipids, which are biomicromolecules, which are what? Which are biomicromolecules having weight less than 800, molecular weight less than 800 Daltons. Remember? Do you remember that? So this is something that we have already discussed, right, students? So now why are we starting from this part? Because this part is also important. You should know that we are going to talk about the nucleic acids in this chapter. And when we discuss the nucleic acids, we talk about the DNA, we talk about the RNA. And when we perform the chemical analysis, so we are going to find it where? In the retentate. In the retentate. Okay, that's what we discussed there. Moreover, why do we put the trichloroacetic acid there? Because it can just precipitate out that proteins, right? It is good for the precipitation. That is why. Okay, so that's what we have to discuss in detail. So now when we talk about the nucleic acid, as I said, their molecular weight, is going to be what? Their molecular weight is going to be above 10,000 Dalton. So what exactly we have when we say nucleic acids? We have RNA and we have DNA. This is what we have. So what exactly is the RNA? RNA is ribonucleic acid, students. And what exactly is the DNA? DNA is deoxyribonucleic acid, isn't it? RNA is ribonucleic acid, DNA is deoxyribonucleic acid. So the RNA. See, we know that RNA perform many other jobs, and when it comes to the DNA, it is the genetic material. What exactly it is? It is the genetic material in majority of organisms. It is the genetic material in majority of organism, and when you are talking about the RNA, it is the genetic material in some of viruses, and help in transmission of information. So this is a very important line that I am discussing here right now, that these two are the nucleic acid, and when it comes to the DNA, right, when it comes to the DNA is basically the genetic material. In majority of organism, it is the genetic material. We have the DNA as the genetic material. So DNA is basically for the storage of information. Right, DNA is basically for the storage of information. That is the role of genetic material. And RNA helps in what? RNA helps in transmission of this genetic information, or I would say it helps in the transmission of information. So these are the two main points. This is what we are going to discuss today. We'll be talking about the RNA and the DNA. So RNA perform other jobs, like it acts like messenger RNA, will copy the information from the DNA, will help in making the proteins, right? So we have the rRNAs that will form the ribosomes, right? And then we have the transfer RNA, again, that will help in the translation. So this is the RNA, this is the DNA, and this is what we have to discuss today. So when it comes to the nucleic acid, right? So here we need to know about the Friedrich Mischer, right? What is the name of the scientist? The name of the scientist is Friedrich Mischer. So Friedrich Mischer in 1869, who? Friedrich Mischer in 1869, he was the one who first identified DNA, but he didn't call it as a DNA because he found it in the pus cell, in a WBC basically, in that nucleus. So he called it as nuclein. What he named it? He named it as nuclein. Another important MCQ. So who discovered the nucleic acid, or I would say who identified DNA first? Friedrich Mischer in 1869. He called it as nuclein. Where he discovered it? In the nucleus of pus cell. All the points are important here. And later on, the term nucleic acid, it was coined by Altman in 1899. Okay, so Altman, after checking out its acidic property, called it as the nucleic acid. Is that clear? So yes, this is important. So now when we talk about the nucleic acid, so what we need to know, right? What we need to know here when we say nucleic acid, what do we need to know here? When you talk about the nucleic acid, you will be talking about nitrogenous base, sugar, and here we'll talk about ribose in RNA and deoxyribose in DNA. And what exactly are they? They are the monomers, monosaccharides, right? And to be more precise, it's a pentose having 5 carbons. Okay, then we are going to talk about the acid, and which acid is going to be there? It is the phosphoric acid. Remember, it is not phosphorus or something else. It is phosphoric acid. What is it? It is phosphoric acid. That is what we have to remember. So H3PO4. So basically we say phosphate. So this is what the nucleic acid is. So in simple words, if I have to define the nucleic acid, so we will say that it is the polymer of nucleotides, or it's a polynucleotide chain, or polynucleotide chains make the nucleic acid. That's what we have to remember, right? That is what we have to remember. Okay, so this point that it is having the phosphoric acid, it has been asked in chemistry part, and I believe in paper 2024, right? They have asked this part. So nitrogenous bases, 2024 or 2023, you can check it. So nitrogenous base, monomers, right? Sugar is a monomer here, the monosaccharide here. It's a pentose because carbon number is 5 here, right? So in RNA, you will see the ribose sugar, and in DNA, you will see the deoxyribose sugar. So these three things are going to make the nucleotide, and so many nucleotides together, they form the nucleic acid. That is why when it comes to the nucleic acid, always remember you are going to find it in the retentate, and it's a bio macromolecule having molecular weight above 10,000 Daltons. Is that clear? Above 10,000 Daltons. So let's talk about the nitrogenous bases first. What's the first thing that we have to discuss here? Nitrogenous bases. So from the name itself, one thing should be very clear to all of you, that if you're talking about the nitrogenous bases, means they contain what? They contain the nitrogen. Am I right? Obviously, it is a chemical compound that contains the nitrogen. So here we'll discuss it in two parts. We will talk about the purines and pyrimidines. Okay, purines and pyrimidines. So in purines, the trick is PUGA, P-U-G-A, right? Right? And here the trick is CUT cut. So what does PUGA means? PU means purines, and G here is guanine. And actually this G is denoted by, guanine is denoted by G, and A here is denoted by, A is adenine, and actually adenine is denoted by A. So just look at this, 9 and 9. Can you see that? Can you see that? 9 and 9 in the spelling. This is another way to remember that. So 9 and 9 in the spelling. So these two, right? 9 and 9 in spelling, they are the purines. So purines overall, what are they? It's a 9-membered ring. So here, what is the C? Cytosine. It's not cysteine. Cysteine is an amino acid. U stands for uracil. T stands for thymine here. Okay, so it is basically a 6-membered ring. What's that? It is actually a 6-membered ring. So nitrogenous bases, two types are there, purines and pyrimidines. When you talk about the purines, so purines, they are what? It is basically a 9-membered ring, right? And pyrimidines, 6-membered ring. 2 purines we have to discuss, guanine and adenine. 3 pyrimidines we have to discuss, that is cytosine, thymine and the uracil. So in DNA and RNA, this information is very important, students. We will not discuss it in the biomolecule chapter. We'll cover it here only. Okay. So in DNA and RNA, both guanine and adenine present, right? But here in RNA, cytosine and uracil, and in DNA, it is going to be cytosine and thymine. Okay, it is going to be cytosine and thymine. And remember, we'll discuss the differences as well. So thymine is also known as 5-methyl uracil. Okay, what do you call it? You also call it as 5-methyl uracil. So I believe this part is clear to everybody. So yes, you get the question from this part, right? This is a previous year question where they have asked that out of the following, which one is, common to both DNA and RNA. So you know that guanine and adenine present in both DNA as well as in RNA. Now when you talk about the pyrimidines, which is a 6-membered ring, here you have CUT, cytosine, uracil and thymine. So in RNA you have cytosine and uracil, and in DNA you have cytosine and thymine. Okay, so thymine is also known as 5-methyl uracil. That is at 5th position. You will see the methyl group. Rest everything is same like the uracil. So that is again a previous year question where they have asked which pyrimidine is common to both DNA and RNA. So you know the answer. This is cytosine. Any doubt? Any doubt here? Everybody? Any doubt here? Sure. Okay. So now when we talk about the purines, as I said, it's a 9-membered ring, right? And here you see there is one pyrimidine ring, that is 2N plus 4C. Means this ring is having 4 carbon, 2 nitrogen, and 1 imidazole ring, which is 5-membered, of course. Here again, you will see 2n plus 3 carbons. So it's a 5-membered ring. This is what we discuss in the case of purines, right, students? So purines are having one pyrimidine ring and you'll see one imidazole ring. So let's suppose if I have to give you the rough structure here. So this is what you guys can see. And of course, there will be alternate single and double bond here. So this part, can you see this part? This is the pyrimidine ring. So one pyrimidine ring, 2n plus 4c. And as I said, this one is the imidazole ring. Can you see that? This one is the imidazole ring. So this is what a purine is, right? One pyrimidine ring, 2n plus 4c, one imidazole ring, 2n plus 3c, alternate single and double bond will be there. And there are two examples that we have to study here. So here you guys can see that examples as well, right? So look at this structure, purines here. See, and here itself, let me mention that, then I'll come to the diagram part. So here at 1379, N is present in the ring. I'm not talking about outside the ring. In the ring, at what position you will see the nitrogen? At 1379. And in pyrimidine, you'll see it at 1 and 3. So this is about the purines. Is that clear? This is about the purines. Is that clear? And when it comes to the pyrimidines, so again, they'll have 2n plus 4c, you know it. So at 1, 3, nitrogen is present in the ring. That's what you have to remember, right? Nitrogen is present in the ring at position 1 and 3. So we have 3 examples here. We have cytosine. So the trick here is cut, CUT, uracil and thymine. So this YY is common. So it is in the DNA, and these two, uracil, R is there. It is in the RNA. So that's what we have to remember. So just look at this diagram here, students. So you people can see nitrogen at 1 position, 3rd position, 7th position and 9th position. So 1, 3, 7, 9. Alternate. See, double bond, single bond, double bond, single bond, double bond, single bond. This is what you have to remember. In the pyrimidine, again, this is position number 1, 3. So 1, 3, rest 1, 2, 3, 4. 4 carbons are there. So it is 4C plus 2N. And this one is basically, pyrimidine ring is 4C plus 2N. And it is actually 3C plus 2N, right? So this one is imidazole ring. This one is pyrimidine ring. Together they are making a purine. Okay. So now look at the example here, students. So you people can see it very clearly. This is the guanine. Right, guanine and adenine. Guanine, go, O sound is there. So you can see here, right, at 6th position, you'll see C double bond O, 6th carbon. This is what it is showing. But here in adenine, it is going to show what? NH2 group, right? Which group will be there? NH2 group will be there. Is that clear? Is that clear? So which group is there, guys? Which group is there? You people will see the NH2 group there. Okay, NH2 group is present there. So C double bond O, CNH2 is there. Rest here in the guanine, right? The 2nd carbon is also having the amino group, and their 6th carbon is having this NH2 group. Is that clear? Is that clear? So I believe the structure is clear. So if, let's suppose if you get the structure in the paper, at least by looking at the ring, you can say whether it is purine or pyrimidine, right? Alternate double and single bond, 1, 3, 7, 9, nitrogen is there. So how to differentiate guanine and the adenine? So guanine at 6th position, C is attached to the oxygen, and here in adenine, it will have NH2 group. Plus in guanine, 2nd carbon is also attached to NH2, right? So that's what you have to remember. Now, just look at the pyrimidine ring, cytosine, again, double bond, single bond is there. So see this. This is again this is 1st carbon, 2nd carbon, 3rd carbon, 4th carbon, 5th carbon and 6th. So cytosine, 4th carbon is having NH2, right? Here, right? Both are having C double bond O. So here at 2nd carbon, you'll see C double bond O. So that is common in all 3. And here in the uracil, this is the structure of uracil. This is thymine. So in thymine at 5th carbon, methyl group is attached. So that is why thymine is called as. So that's why thymine is also named as what? It is named as 5 5-methyl uracil, right? It is also named as 5-methyl uracil. Again, it is also a previous year question. Any doubt here? Yes, everybody. So do you need to remember this? Of course, you have to, right? You have to. So next part that we are going to cover here. So we are done with the nitrogenous bases, right? So the next thing that you have to discuss is sugars. Okay. So sugars, basically, what does it mean? What does it mean? We talk about the monosaccharides here. What do we discuss here? Monosaccharide. So mono means single. Saccharide means sugar. It is having single sugar. So for monosaccharide, the general formula is, student, CnH2nOn, where N is the, you know that, right, number of carbon, you can say CnH2nOn. So we have, right, when we talk about the monosaccharide, we know that here carbon number in monosaccharides, carbon number is from 3 to 7. Yes. When can you say it is a monosaccharide? First of all, monosaccharide, they are having single sugar, right? They are the compounds which on hydrolysis, right, they do not further yield sugar. These are the monosaccharide, right? So basically they are made up of, obviously we know that carbon, hydrogen, oxygen is there. Overall in carbohydrates, that's how we define them, that carbohydrates are the hydrates of carbon. So here the carbon number and the water molecule, CnH2O whole square N. So carbon number water is same. Okay, and in other ways, how can you define it? That in the case of carbohydrates, right, in the case of carbohydrates, you'll see, what will you see? The hydrogen and oxygen, right? It's just like the water. Like water is having 2 parts of hydrogen, 1 part of oxygen, same in the case of these carbohydrates. So chemically they are polyhydroxyaldehyde or polyhydroxy ketones, and the compounds which on hydrolysis yield such compound. Now when you're talking about the single sugar, monosaccharides, in their case what you have to remember, that the carbon number is from 3 to 7. So the one having carbon number 3 is triose, the one having carbon number 7 is heptose. So here you see it from triose to heptose, and what we have to see here, we will see the pentose, right? The sugar that we are going to discuss in the nucleic acid is pentose. So here carbon number is 5, students. So we have 2 examples here, ribose, and here the sugar is an aldose, means the functional group is aldehyde here. So again, I'm repeating this part. When you define the carbohydrates, chemically they are polyhydroxyaldehyde or polyhydroxy. Poly means many, hydroxy, OH. Polyhydroxyaldehyde or polyhydroxy ketones, or the compounds which on hydrolysis yield such compounds. Okay, so poly means many, hydroxy means OH, plus we know that either these sugars, they have aldehyde group or the ketone group. So ribose is an aldose, it is having the aldehyde group. So one more thing that you have to learn here. See, as I said, the general formula for monosaccharide is CnH2nOn. So here carbon number is 5, so it will be C5H10O5. And in deoxyribose, it's having 1 oxygen lesser than the ribose. So C5H10O5, C5H10O4, that is their formulas. So look at this. Look at this, your deoxyribose. It doesn't obey general formula for monosaccharides, right? It doesn't obey general formula for monosaccharides. That's what you have to remember. It doesn't obey the general formula for the monosaccharides, so it is an exception here. Okay, now look at their structure. So this is carbon number 1, carbon number 2, carbon number 3, carbon number 4, and here we'll have carbon number 5. Okay. So look at this, students. This one is your beta ribose sugar and this one is beta deoxyribose sugar. right? Why do we call it as beta? Because when you see the carbon number 1 here, so this is 1, 2, 3, 4 and 5. So when you see the carbon number 1 here, so the hydrogen is below, oxygen, oh sorry, hydroxy group OH is above. So hydrogen is below. That's how you can remember. So here, this is the position below, hydrogen below, beta, right? So it is going to be your beta sugar. This is going to be your beta deoxyribose. So how can we differentiate it? Look at it. Everything else is same except this part. So in deoxyribose, at second carbon, O is missing, right? That's why it is having one oxygen lesser than the ribose. So it is C5H10O5 and this one is C5H10O4. So this one is an exception. It doesn't obey the general formula. So I believe this part is clear. Please take a note. Everybody quick. Please take a note. Look at it. Compare the structures. Everybody quick. Look at it and compare the structures. Done? Sure? Okay, so this is about the sugars. You can again see this part, okay? You can just have a look. Now, the next thing that we have to discuss is nucleosides. What we have to discuss? Nucleosides. So guys, how can you remember that? It's very simple. N stands for nitrogenous base. S stands for sugar. That's how you can remember. Nitrogenous base plus sugar. Nitrogenous base plus sugar. Okay. So yeah, that's true. Nitrogenous base plus sugar is going to make nucleoside. And we know it very well. We have to connect it with a bond. So which bond is going to attach them? The name of the bond is N-glycosidic bond. Okay, N-glycosidic bond. So again, that's again a previous year question. So now, let's take the example. Let's suppose you are writing adenine. Then you are writing ribose. So what's that? That will make nucleoside. So that is adenosine. So there is another PYQ where it has been asked that how do you differentiate adenine, adenosine and adenosine monophosphate. So adenine is the nitrogenous base, adenosine, when it is written as adenosine, so that's what is it? It is the nucleoside. So that is a PYQ. Adenine is a nitrogenous base, adenosine is a nucleoside. So let's suppose if at the place of ribose, if it is deoxyribose, then what are you going to call it? You will call it as deoxyadenosine. That's the name. Okay, same way. Guanosine, uridine, thymidine, cytidine. Okay, so they all are nucleosides. Are you getting my point? They all are nucleosides. So thymidine, you know that here you will see thymine plus deoxyribose. So you can also call it as deoxythymidine. You can also call it as deoxythymidine. thymidine, and in uracil, what do we see? Ribose. So it is going to be uridine. Am I clear? It is going to be uridine. So that's what you have to remember. So nomenclature is important. Now I'll tell you about the bonding. See, how do I remember this? I remember it in a very simple way. Sugar is sweet, so everybody is interested in having bond with sugar. Okay? Sugar is sweet and everybody is interested in having a bond with sugar. So tell me which sugar it is, ribose or deoxyribose? Quick. Which sugar it is, ribose or deoxyribose? Quick. Ribose or deoxyribose? What's that? Yes, please. What's that? Ribose or deoxyribose? Which sugar it is? Of course, it is the ribose sugar. How can you check it? Second carbon. OH group is there. Now, let's see. This is your, I'm not making alternate double and single bond, or I'm not making any specific structure here. Just trying to show you. Is it a purine or a pyrimidine? This part. Right? I'm just ignoring this double bond and the single bond. What's that? Purine or pyrimidine? That's a purine, of course. Hai na? So 1, 2, 3, 4, 5. This one is 6, 7, 8, and 9. This is the 9th nitrogen. So this 9th nitrogen is having H. H group is also there. Here you have OH. So what will happen here? Water will come out. What is going to happen here, students? Water will come out, right? Dehydration will be there. Dehydration will be there, and what's going to happen? Water will come out. So now when water will come out, just look at it very carefully. So it's a deoxyribose sugar. First, let me draw the sugar here. So when water will come out from this part, so you can see, right, because this H2O will be out. So obviously this bond, this will directly connect to this nitrogen here. See this. That's what you people will see. Just have a look. This is what you guys are going to see. So this H here, OH here, water will go out, dehydration it is, and it is the formation of the glycosidic bond. But why do we call it N? Because here, this bond is in between carbon and nitrogen. So this one is your N glycosidic bond. So when you talk about this N glycosidic bond, what you have to remember here, you just have to remember one thing, that N glycosidic bonds connect nitrogenous base with sugar, forms the nucleoside. So in purines, 9th nitrogen at 9th position, nitrogen at 9th position, your nitrogen of 9th position form bond with the first carbon of sugar. Do we need to remember it? Of course, we have to. But in pyrimidines, the story will be little different. In pyrimidines, you'll see nitrogen at first position form bond with the carbon C1 of sugar. That's it. So pyrimidine 9th nitrogen, purine 9th nitrogen, pyrimidine nitrogen at first position. So of course this information is very important, and here also this is the bonding part Again, very important. So this chapter overall is very, very, very important. Nomenclature and everything. So here, if it is guanosine, you know that if deoxyribose will be the sugar, deoxyguanosine, right? Deoxythymidine or thymidine, same. So deoxycytidine or simple cytidine. That is how you can differentiate the sugar as well. Any doubts? Yes, please. Any doubts? So here you guys can see the bonding. So not just some MBOI, just open up biomolecules. In the chapter biomolecules, just open up NCRT, right? Just check out NCRT. There are diagrams now. There is uridine given there. That is also a PYQ. So whatever structures are given in the book, be it in, your 12th biomolecule NCRT, your 11th biomolecule NCRT, even in your molecular basis of inheritance chapters NCRT, you should know about it. Is that clear? So this is adenosine. So first carbon of sugar is always engaged, and when it is the purine, it is always the 9th nitrogen. And see, when it is the pyrimidine, it is the first nitrogen, right? Again, this is the purine 9th nitrogen. This is the pyrimidine. This is going to be the first nitrogen. Okay? Okay. So, next thing. So, we are done with the nucleosides here. Okay. We are done with what? Nucleoside here. Now, we'll see that in nucleic acids, what do you see? You will also see phosphoric acid. So, how can you remember? See, nucleic acid, ic, phosphoric. That's how I remember this. So it's H3PO4. So that's what we have to discuss. Now, you already know about nucleoside, isn't it? That it is having nitrogenous base plus sugar. Now, when a phosphate gets attached to it, right? So when it becomes nucleoside, monophosphate, a single phosphate, when it gets attached to it, then you will call it as nucleotide, right? Then what will you call it? You will call it as nucleotide. So what exactly is this nucleotide? From this T, you have to remember 3, right? That here 3 things are attached. Which 3 things are there? Nitrogenous base, sugar plus phosphate, right? In ionic form when you write this. So nitrogenous base, sugar plus phosphate. Nitrogenous base, sugar plus phosphate. That will make nucleotide. So obviously nitrogenous base plus sugar is nucleoside. So because one phosphate is attached, so this is going to be nucleoside monophosphate. Am I clear? It is going to be nucleoside monophosphate. So if I have to show you the nomenclature, then I'll come to the bonding part. Jaise adenosine. Adenosine is a nucleoside. So you add one phosphate there, adenosine monophosphate, that will become a nucleotide. You can also call it as adenylic acid. Then guanosine monophosphate. Then it is guanylic acid. Cytidine monophosphate. Then of course it is also known as cytidilic acid. Uridine monophosphate. So it's one and the same thing. You are talking about the nucleotides then. Then it will be uridylic acid. Okay, so I'll just write one reference example here. So if it is deoxyguanosine monophosphate, means the sugar is going to be deoxyribose. So you can also call it as deoxyguanylic acid. Deoxyguanylic acid. That's how you can just write the nomenclature. So nitrogenous base plus sugar plus phosphate it is, students. So this will be joined by N-glycosidic bond, and here sugar and phosphate, they'll attach with phosphoester bond. This is not diester, this is ester in single nucleotide. So again, the important information. Okay, so these are the bondings that you are going to see. Done? This is the bonding that you will see. So N-glycosidic bond, phosphoester bond. N-glycosidic bond and phosphoester bond. Understood? I hope it's done. Just give it a reading, then we'll see the bonding part as well. Is it done? So quickly. Okay, so next is the bonding part. So see, this is the sugar, and sugar is in which form? This is furanose. This ring is actually furanose, remember? So here H, here you will have nitrogenous base, here H, OH, H, OH, and here you have, let's say CH2, I'm writing it in this way. Chalo, I'll write it simply. CH2OH. Okay, this is CH2OH. Okay, so already in the case of the sugar, first carbon is all right. First carbon is actually engaged here in bonding with the nitrogenous base. So it is a nucleoside. Now you have this, right? You have this. So even if you take it in the ionic form, what you have to consider? So you know that here you'll see minus H2O, isn't it? What are you going to see here? Minus H2O means water will release. You know that this is the ester bond, right? That's how we make the ester bond, isn't it? That's how we make the ester bond. Okay. So now we call it as phosphoester bond. Which bond it is? It is phosphoester bond. So that's how you are going to make it. Are you getting my point? So P double bond O, OC, right? So now look at that structure. And which sugar it is? Ribose. So CH2, then O, then P. If I do not, so this is how it is. So here you'll see phosphoester bond. This is a single nucleotide. Okay, so in a nucleotide, you'll see, again I'm repeating, see, N-glycosidic bond, and of course, phosphoester bond. That's what you have to remember. Right, N-glycosidic bond and the phosphoester bond. It is not diester, it is phosphoester, like C. this is what one nucleotide is. Am I clear? This is what one nucleotide is. You want to write it in ionic form, you can also write it phosphate, then you can call it. Done. This is another nucleotide. See, this is next nucleotide. Kindly pay attention. This bonding part is actually very important. This is next nucleotide, isn't it? That's what we have, right? That is what we have. Done? Okay. So this is the next nucleotide. See this. So let's suppose if I have to, like it is in the same way, but just for your clarity, let's suppose I'm writing it in this way. है ना? It's all same. See, O negative, P double bond O, this O. Now what's going to happen, right? Just for your understanding, I'm writing it in this way. This is your first nucleotide. Yes, people, this is your second nucleotide, or I would say the next nucleotide. So this is 3 dash, this is 1 dash, carbon number 1, 2 or 2 dash, whatever you want to say, 4 dash or 5 dash. This one is also 1, 2, 3, 4 or 5 or 5 dash, right? In any way you can write it. Now here also, what will you see? Water will release, right? From this side also, H2O will come out, okay? Because now we have to make a chain. Can you see that? What? So this is the first nucleotide. This is the second nucleotide. So the 3 dash carbon, the 3 dash carbon, the OH at 3 dash carbon of first sugar will connect with the OH at the 5 dash carbon of next sugar. Okay, so what is going to happen here? Nitrogenous base, hydrogen, so that's how it will link. Just have a look. That is how it is going to link. Just have a look, please. Okay. That's how it is going to link. So when you look at this part, you can see one phosphoester bond, another phosphoester bond. So this is phosphodiester bond, and we also write it 3 dash 5 dash phosphodiester bond. Can you see that? And here, if somebody asks you about this bond, this is phosphoester bond, because this is the first nucleotide, right? This is the first nucleotide, this is the second. Okay, so this is phosphodiester bond. Phosphodiester bond. So how will it form? It forms in between three dash OH at, I would say, of first nucleotide sugar with OH at 5 dash carbon of next nucleotide sugar. So that's how this bond forms. So it is the 3 dash 5 dash phosphodiester bond, students. Okay, so that's a previous year question. In nucleotide, which bonds are present or which bonds form a nucleotide? So simple, N-glycosidic and you see phosphoester. But if it is a polynucleotide, or let's suppose if they ask dinucleotide, in dinucleotide which bond is going to link it, then you will say phosphodiester. Phosphodiester in dinucleotide or in polynucleotide. Individually you can see at first, if let's suppose this is the structure that they gave you in paper and then they name this, that what is A, then it is phosphodiester. What is this B? Phosphodiester. So it is the phosphodiester bond that is going to link the sugars. Am I clear? It is the phosphodiester bond that is going to link the sugars. Is that clear? Sorry, that is going to link the sugars of the nucleotides, or I would say the nucleotides. Okay, so that's why even in DNA replication, even in RNA formation, we say 5' end is having free phosphate, 3' end is having free OH, because actually they are involved in the bond formation. So when it is the first nucleotide, it's the phosphate at the 5' end is not involved in any bonding, right? This phosphate. So this is the, here the phosphate is free. So we call it as 5' phosphate end, right? 5' end, or it is having the free phosphate. Now when you look at, see, this is the second nucleotide. Obviously you'll keep on adding the nucleotides like this. So when you focus on that last nucleotide now, so you'll see this OH. In that case, it is not involved in bonding. This OH at 3' position. So 5' end is having free phosphate, 3' end is having free hydroxyl group. When in the case of, yes please, in the case of polynucleotide chains. Am I clear? In the case of polynucleotide chain. Am I clear? In the case of polynucleotide chain. Understood? Understood? So if it is done, do let me know. Please have a look. If there is any doubt, do let me know. So I hope it's done. So this is all about the bonding part. So in the same way, you can see it here as well, right? So this is the sugar. So first carbon, right? First carbon of sugar will form bond with the nitrogenous base. And here you will see N glycosidic bond, right? And you know that in case of sugar, it is going to be carbon at first position. And if it is a nitrogenous base, in the case of purine, this will be N at 9th. And in case of pyrimidine, N will be at 6. And here, this is the phosphoester bond. Sugar, C5, and this is the phosphate, right? So that is the nucleotide. Okay, that is the nucleotide. Everything is mentioned here. So this is one nucleotide. This is next. How are they attached? 3-OH of first sugar and the phosphate of next sugar. You guys can see ligase. What is this enzyme? This is the molecular glue, right? What is this enzyme? This is the molecular glue. It helps in forming sugar phosphate. It helps in forming this phosphodiester bond, right? This enzyme actually forms this phosphodiester bond. So we say this enzyme helps in forming sugar phosphate backbone, right? So all the diagram, the points, everything is mentioned here. You people can simply check this out, okay? So it is going to be sugar phosphate backbone. That's it, right? So that's how the polymer will be formed. So here you guys can see, right? You can just have a look. So 5' end, okay? This is the 5' end. Though here it will, you will see 5' end will have free phosphate. This is a 3', 5'. So this is 3', 5' phosphodiester bond. Okay, that is linking it. 3', 5' phosphodiester bond. This is again 3', 5' phosphodiester bond. So 3' end is having free OH, 5' end is having free phosphate. So that's how even the chain will form. Just simply keep it in your mind, okay? Done? So see this, this is your nucleoside. When a single phosphate is attached here, this much is your nucleotide, okay? So if 2 phosphates are attached, nucleoside diphosphate. If 3 are attached, nucleoside triphosphate. Like when you say ATP, what do you say? What is the full form here? Adenosine triphosphate. So you'll see the 3 phosphates there, not 1. And when we make the polynucleotide chain, the nucleotide is having 1 phosphate. We'll use this information when we'll study the DNA replication. And here you have the bases in front of you. Definitely you need to remember that. So just have a look. See, this is the diagram from the NCERT. Ultimately, we have to simplify this NCERT, okay? So look at this. 5' end is having free phosphate. Important. Let's suppose in the final paper, they give you this diagram and instead of phosphate here at 5' end, they put OH, then that will be incorrect, right? And here in between, let's suppose if in the paper they ask you to name this. So this is your phosphoester bond. And what's that? Sorry, phosphodiester bond. This one is here, phosphodiester bond. Am I clear? And this one is phosphoester bond. Am I clear? So diester, and see 3' end is having free hydroxyl. Okay, and these are the bases, right? These are the bases. Okay, so phosphate moiety means molecule is at 5' end, which at 5' end of sugar, which is referred as 5' end of polynucleotide chain. So at the other end of the polymer, sugar is having free 3' OH group at 3' carbon, so which is the 3' end. So backbone of polynucleotide chain is formed due to sugar and phosphate, and nitrogenous bases are linked to the sugar moiety. They project from this particular backbone. So that's the example also. Phosphate part, ester, phosphodiester, that's how it forms. So all the diagrams are also included in this particular PDF. So this is all about the basics that we need to understand. Okay, so now we'll be discussing the structure of the DNA. But before that, right, in detail obviously we'll discuss, but just I assume you know that much. that DNA is a double helical structure. DNA is a double helical structure, right? Basically a double helical, helical structure. So DNA, it is double stranded. Okay, it is double stranded and it is helical as well. I believe you remember that much. So we call that this DNA is actually what? It is anti-parallel in nature. If let's suppose instead of anti-parallel, if parallel is written, is it true? Yeah, it's true. Right, it's true. Definitely it's true. Why do we use the word anti? Anti is just telling us that they are in opposite direction. Here 5' end is having phosphate, 3' is hydroxyl. So here in front of this 3' you have 5' end, and here you have 3' end having OH and having OH, right? So sugar phosphate backbone is there and there are bases in between. Like let's suppose this is one nucleotide chain, this is another. Let's suppose here adenine, guanine, cytosine, thymine is there, right? So here also adenine, guanine, cytosine, thymine is there, right? It's not mandatory that they'll be in this order. I'm just telling you. So all I want to say is that this adenine and thymine, these nitrogenous bases, they project here in the center. There is a sugar phosphate backbone, right? And these bases, right, they are attached to each other with the help of hydrogen bonding. Like in the case of adenine and thymine, right? 2 hydrogen bonds are there that are connecting them, and cytosine and guanine, they are connected by 3 hydrogen bonds, right? How are they connected? They are connected by 2 hydrogen bonds. They are connected by 3 hydrogen bonds. Is that clear? Right? Okay. Right, so this is as per certain rules which we are about to discuss. So sugar phosphate backbone is there and that's how the structure is. So that's how you can relate. Okay. So there is a sugar phosphate backbone. Right, so sugar phosphate backbone and bases are there projecting in the center. That's how the structure is. So synthesis will always be in 5' to 3' direction, 5' to 3' direction. Both the strands are moving in anti-parallel direction. So these are certain bases. So let's quickly revise the important points from the NCERT and then let's start the structure of the DNA. So I believe up to this part everything is clear to all of you, okay? Okay. So if you read this first part, so DNA and RNA, these are the basic points that they have mentioned. 2 types of nucleic acids found in the living organism. In majority of organism, it is DNA, and RNA it is in some viruses. Okay, and these viruses are RNA viruses, of course, right? Right. So if it is a single-stranded RNA, then it will be the retrovirus. So in retrovirus, single-stranded RNA is the genetic material. Reoviruses are also there, which are having double-stranded RNA. So RNA mostly function as the messenger, and all I want to say is just keep it in your mind that RNA, right? It is good for the transmission of information, but when it comes to the storage of information, DNA is better, okay? So RNA is having different roles. So we'll see the structure of DNA, replication, process making of RNA, everything there. And these are the points that they have mentioned. So now let's discuss the structure of the DNA, which is a double-stranded. So for double-stranded, that's how we will write double-stranded DNA. This is double-stranded. If it is written as ssDNA, right? I'm just telling you the short form. So SS means single-stranded. Okay, SS means single-stranded. DS means double-stranded. So just remember this. Okay, just remember this. Okay, so now when you see the structure of DNA, we talk about the scientists Watson and Crick. Which scientists do we discuss? Watson and Crick. So they explained the structure of a right-handed DNA, the most commonly found one, and it is also known as bDNA. Okay, actually that was not their work, right? It was actually the work of Rosalind Franklin, the dark lady of DNA, right? She worked hard. She performed X-ray diffraction or X-ray crystallography techniques. She got the photograph 51, right? And that photograph helped the Watson and Crick to explain the structure of DNA. So without her knowledge, right? Without her knowledge, her assistants, or I am not sure assistants, or Morris Wilkin and Rosalind Franklin, they were working together. So without the knowledge of Rosalind Franklin, right? The Morris Wilkin, he passed this photograph 51 to Watson and Crick, right? They were also working on the DNA, right? I will not dive deep into the story here, otherwise you'll say, ma'am, you have become the feminist here. No, but actually that is the case. Her data was shared to the Watson and Crick without her knowledge, and by using that photograph 51 that she obtained from X-ray diffraction or X-ray crystallography, right? The Watson and Crick, they were able to explain the structure of DNA, the bonding part. Plus, they also used the Chargaff's rule, obviously given by scientist Irwin Chargaff. He gave that. So they applied this information and they were able to explain the structure of DNA. So, posthumously, like after the death, posthumously Nobel Prizes, they are not awarded. So, Morris Wilkin, Watson and Crick, they shared the Nobel Prize for the discovery of the DNA. But actually, Rosalind Franklin, she is the lady behind. She's also referred to as the dark lady, right? Dark lady of DNA. She was actually the person, right, who helped us to understand, or to whose discoveries basically are responsible for knowing the structure of DNA. So this is a diffraction pattern. This is a photograph 51, which helped them. Right, so these are Watson and Crick, right? So they got the Nobel Prize, they got the recognition, but they were thieves basically. Okay, so she is the one, Rosalind Franklin, and that's the photograph 51. So you guys can read about that, right? So it is obtained by X-ray diffraction. One day read about her. Okay, you'll be inspired. She worked really hard. She deserved the Nobel Prize for her work 2 times, but she didn't even get it once. Right? She didn't even get it once. Okay. So, they are the thieves. And she is the dark lady of DNA. So, let's discuss that. Then let's discuss about the Chargaff rule. And then, after the Chargaff's rule, right? After reading the Chargaff's rule, I'll tell you, the dimensions that in the case of DNA, bDNA, the right-handed DNA, the most common one, the diameter is 2 nanometer or 20 angstrom. All that things we will discuss then, okay? Chali. So here you can see the elucidation of structure of DNA. It was elusive for a very long period of time. So in 1953, information is important, Watson and Crick, James Watson and Francis Crick, based on X-ray diffraction. You can also write X-ray crystallography here. So once this question was asked in the paper, okay, that which technique was used? So it was X-ray diffraction. So data produced by Morris Wilkin and Rosalind Franklin. Wilkin was also jealous of her. So they proposed a very simple but famous double helical model for the structure of DNA. So it is the bDNA, the most common one, and the right-handed one. Okay. So Watson and Crick and Wilkin, they were awarded with Nobel Prize in 1962. So they gave the structure in 1953, got the Nobel Prize in 1962. Is that clear? So it is very important for the exam, Chargaff's rule, right? If you follow our channel, I believe you follow it, and you should, and do recommend our channel to your friends as well, because you know that here the content is, right, the quality of content is top notch. So in the short, in the shorts I put the, there is a biology 1-minute series, right, short video series also you guys can say. So here you can see I have explained the cardiac cycle. Okay, I have even explained the blood coagulation here in detail. Okay, and here I have explained the Chargaff's rule. So you guys can just simply, subscribe to our channel if you're new to it, and you can simply check out these shorts where I have posted, right, not just important update or information, but actually the content that you can, actually the videos that you can use to revise a topic, and that too in less than 2 minutes. Okay, so here, Chargaff in 1950. In which year? Year is important. Yes. He made some molecular estimates of different components of DNA. What he did, he made the molecular estimates of different component of DNA and what he observed, right? He observed and his observations were named as Chargaff's rule. So number one is molar amount of purines are what? Adenine, guanine. So it is equals to what? Cytosine and thymine. So obviously, so if I will take the ratio here, A plus G over C plus T, that is equals to 1, right? That is equals to 1, isn't it? Isn't it? So this is what he observed, and these observations, they were for the double-stranded DNA. If let's suppose, if somebody asks you, right, that in the case of single-stranded DNA, are we going to see the same thing always, that purines, their molar amount is equals to the pyrimidines? The answer will be no. In the double-stranded DNA, you are going to see this. Secondly, he observed, right, that adenine, molar amount of adenine, that is A is equals to T. This is not the hydrogen bonds, equals to. And C is always equals to G. That's what he observed. Okay. So first of all, purine to pyrimidine ratio is equals to 1. Then adenine is equals to thymine. Cytosine is equals to guanine. That is another observation. Then the ratio of adenine thymine, because their amount is equal to the guanine cytosine, it is variable. But how? In different species. Like in our case, this ratio will be different. In other species, this ratio will be different. So this ratio is constant for a species, but it varies in different species. So in simple words, I can say this is species specific, right? This ratio is what? This is species specific ratio. So purine to pyrimidine 1, adenine is equals to thymine, and adenine plus thymine to cyto plus guanine is variable and is constant for a species. Or in simple words, what can we say? That it is species specific. So that is exactly the Chargaff's rule. Is that clear? That is actually the Chargaff's rule. So here you can see, so this rule applies almost universally in the DNA of viruses, bacteria, plants and animals, but there are certain variations. Just higher plants or animal, DNA is in higher plants and animals, the DNA is rich in adenine and thymine, right? And DNA is relatively poor in guanine and cytosine, right? And moreover, in microorganisms like viruses, bacteria or lower plants, DNA is rich in guanine and cytosine, and it is poor in adenine and thymine. So these were the observations that they have made. So they concluded that difference in this AT to GC ratio, it reflects variation in genetic information, and the phylogenetic, the evolutionary and taxonomical significance is also having, right? Right? We have that such significance. Okay? This ratio is having that significance. Understood? Sure? Are you sure? So this is the Chargaff rule. Okay? This is the Chargaff rule. Understood? So they use this data and the X-ray crystallography data, the photograph 51 thing, and then they did what? They explained the structure of DNA. So see, this is the DNA. So obviously, right, I'm not the expert here who can depict this part. So that's how they obtained the structure of DNA. So I'll mention the points there, but firstly see this. Let's suppose this is the DNA, right? You can imagine it, or you can compare it with the helical staircase. So when I talk about the diameter here, it is going to be 2 nanometer or 20 angstroms, right? 1 nanometer is equals to 10 angstroms. So 2 nanometer is equals to 20 angstrom. Secondly, because they explained what? I told you the most common type of DNA, the BDNA, the right-handed DNA. So here, each turn, when you look at this, this is, right, this is going to be your major groove. This one is comparatively narrow, minor groove. So this one is major groove. Okay, this one is minor groove. So students, one major and one minor groove makes one turn. And here we see that each turn, you can, as I said, you can compare it with the helical staircase. So each turn is having 10 base pairs. When do we say base pair? See, I told you already, in the DNA, sugar phosphate backbone is there. Bases are projected in the center. And from the Chargaff rule, they concluded that adenine always pair with thymine. And how many bonds are there? 2 hydrogen bonds. Right? How many bonds are there? 2 hydrogen bond. And here 3 hydrogen bonds are there. So adenine alone is a base, thymine alone is a base. When they are showing this pairing, then I am calling it as base pair. So if I'm saying base pair, so I'm talking about 2 bases, right? I'm talking about 2 bases. Is that clear? So if I'm saying each turn is having 10 base pairs, means 20 bases are present. How many bases are present? 20. So let's suppose this is one base pair, this is another. So here in between these two, what is the distance? It's 0.34 nanometer or 3.4 angstrom. Okay? Between the 2 bases, let's suppose here adenine thymine is written, cytosine guanine is written. So here, in between these two, imagine it like a staircase, one stair, then next. So basically, one stair and next, then next, then next. So they are having what? 0.34 nanometer, which is 3.4 angstroms. So here, students, as I said, each turn is having how many base pairs? 10 base pairs are there. So if each base pair is having a distance of 0.34 nanometer, you can multiply it with 10 and you will get 3.4 nanometers or 34 angstroms. That's how you can check it. That's how you can check it. So this is the data that they have given us. And moreover, let's suppose now, if I change the information here, right, if I give you a hypothetical situation where I'm saying the distance between 2 base pair is 0.48 nanometer and each turn is having 20 base pair, then how will you calculate the distance of each turn or each helix? 20 into, right, 0.48. That's how you can calculate. Are you getting my point? So obviously that's how you can have the different calculations. So I have changed the distance, I have changed the number of base pairs. So this is how I'm going to obtain the information out of it. So that's what they did. So now let's explain this part properly, okay? So see, DNA double-stranded, it is made up of how many polynucleotide chains? 2. So the backbone will have what? It is having alternate sugar and phosphate groups, or I would say the backbone is sugar phosphate backbone. So nitrogenous bases, they project inward from the backbone. Okay, so 2 strands because they run in opposite direction. That's why we call it as anti-parallel. So one strand is having polarity 5' to 3'. Like let's suppose this is your 5', 3'. So another strand will having this 3' to 5'. So that's what we read. Okay, that is what we read. Now the next to it is base pairing by hydrogen bond. So adenine will always pair with thymine through what? 2 hydrogen bonds. Guanine with cytosine by 3 hydrogen bonds. Done. Then adenine will always, or I would say a purine, which is 9-membered, will always pair with pyrimidine. That's a rule, right? That's a rule. So why is it so? Obviously, so that uniform strand distance can be there. Is that clear? So that uniform strand distance can be there, right? So if it is a purine, let's suppose this, here you can see, sorry, it is a pyrimidine, and here let's suppose you have purine. Now here you have a purine and here you have a pyrimidine. So overall, you'll see the distance is going to be what? It is going to be uniform because 9-membered is always pairing with a 6-membered. 9-member is always pairing with a 6-membered. Am I clear? So that's what you have to remember. So the 2 strands, they coil around each other in a right-handed fashion. So the DNA that they explained, once again, I'm telling you bDNA, and that's what they said. Okay, so there is one more line where it is written helical pitch and base pairing spacing. What does it mean? So one complete turn of helix, it has 3.4 nanometer, or you can say that 34 angstrom. And students, remember 1 nanometer is equals to 1 billionth of a meter, right? 1 nanometer is equals to 1 billionth of a meter. That is 1 nanometer is equals to 10 raised to power minus 9 meter. Is it important? Of course it is important. Okay, it is important as well. Done. So each turn includes approximately 10 base pair. It varies from DNA to DNA because not all the DNAs are right-handed. Let's say we have different types of DNA, A DNA, B, C, D. So it is only your Z DNA. You can remember zigzag from it. Students, it is going to be left-handed. Rest all are right-handed. So you can write it. It is your Z DNA that is left-handed, rest all are right-handed, and yeah, number of base pair per turn can vary in different DNAs. Am I clear? In different DNAs. कदूबा मैं डिटेल में बताओ मैम, बेटा इससे ज़्यादा डिटेल तुम्हारे समझ में नहीं आएगी अगर तुम्हें ये भी नहीं समझ में आ रहा, इससे ज़्यादा डिटेल तुम्हें कोई नहीं बताएगा. ओके चलिए, so this part is clear. Next, base stacking for stability. No doubt hydrogen bond is there that is providing the stability. Moreover, you know, in minor and major groups there are different proteins that are interacting, but here the plane of one base pair will stack over the next. This provides additional base pairing. So this stacking along with hydrogen bond, it will contribute to the overall stability of the double helix. So just say one base pair will stack over another, Just imagine a helical staircase in your mind and imagine the steps, the arrangement of steps. So it is also in such way here in DNA. That's why the more stability is provided here. So the base pairing, right? Because here there is the base pairing and there is the complementarity. What does it mean? That adenine will always show pairing with thymine. Cytosine will always show pairing with guanine. So actually we say adenine is always complementary to thymine and cytosine is always complementary to guanine. So the base pairing will provide is providing a very unique property here, right? And see, they are complementary to each other. So look at the structure. That is the axis of the helix, right? The minor and the major groove. One minor and major groove is making one turn of this helix, right? And here you can see one turn of the helix is 3.4 nanometer, right? So you just have a look quick. Guys, quick. Observe the diagram carefully. So this is 3.4 nanometer. So it is also, one turn is 3.4 nanometer. So it is also known as pitch of helix. So pitch of helix is what? 3.4 nanometer or 34 angstrom, right? 3.4 nanometer or 34 angstrom. So one turn means 360-degree rotation here. Is that clear? Right? So in meters, what is it going to be? In meters, what is it going to be? So you know that 1 nanometer, students, equals to 10 raised to power minus 9 meters, na? So here it is 3.4 nanometer. So that will be 1 nanometer is equals to, so 1 nanometer is equals to what? Yes, please. 1 nanometer is equals to 10 raised to power minus 9 meters, na? So here what should be the answer? Quick. Can we simply write it 3.4 into 10 raised to power minus 9? 3.4 into 10 raised to power minus 9 meter. That's how you can even change the units. Okay? That is how you can even change the units. Right? So 2 bases, 0.34 nanometer. So 3.4 nanometer means 3.4 into 10 raised to power minus 9 meter. That's it. Okay? That's it. So I hope the structure of DNA is crystal clear to you. Okay? Sure? So if you are not able to understand what pitch of helix is, it simply means matlab after one complete turn of double helix, the molecule will rises 3.4 nanometer along that axis. Okay. Done? So that's it. So here you can see the large groove, the small groove, which is making one turn. And as I said, right, so it is the pitch of the helix, right, that after one complete turn, this DNA along that axis will raise by 3.4 nanometer. Okay, students. So here from one of my favorites, right? So from this book, I have added this thing that nitrogenous bases, right? Their namings as well, though we have even discussed it in detail. So you can refer to it later. Okay, so here, as I said, A, B, C, and Z. So it is the Z DNA, which is the left-handed. Rest all are right-handed. So yeah, this is important. You should know it. So B DNA is having approximately 10 base pair. 11 is having, A is having 11, C is having 9.33, and Z is having 12 base pairs per turn. So number of base pairs, yes, they do vary. Okay, they do vary. So what can we conclude from this part? We can have simple conclusion here. Okay. So when you talked about DNA, we said it is going to be double-stranded. So because it is double-stranded, that's why it is like all that things that purine is equals to pyrimidine, adenine is equals to thymine. It will be there for the double-stranded DNA. So it is having C, T, A, G. And in RNA, what do you see? C, U, A, G. Sugar here is deoxyribose. It is ribose. Okay, so mostly it is basically it is single stranded. So that's what we need to know. And now let's answer a few questions and then let's go ahead. Then let's discuss the search for the genetic material. So I'll give you one simple question. Firstly, let's suppose if it is a double stranded DNA. If it is what? If it is a double stranded DNA. So in double stranded DNA, adenine is equals to 30%. Tell me about the thymine, cytosine, and guanine. This is first question. Now in a double-stranded DNA, if let's suppose thymine is equals to 40%, then tell me what should be the adenine, what should be cytosine, what should be guanine. Now, one more thing. So you solve this question. Question number 1, question number 2, and I'm writing question number 3 here. Which one is DNA or RNA and is single-stranded or double-stranded? That's what you have to answer. Quick, solve it. Yes, please, quick. Guys, quick. I hope it's done. It's quite easy. Done or not? Noted? See, here you people can check if adenine is 30%, definitely it's a double-stranded DNA, then the thymine will also be 30. 30 plus 30, 60. So I can say 20% will cytosine, 20% will guanine. So 40, 50, 60, 70, 80, 90 and 100. That's how you can check. So here you will write 40%. 40 plus 40, 80, you're left with 20. So you can say 10% and 10%, okay? 10% and 10%. Now, look at this part. Which one is a DNA or RNA? Obviously, this one is going to be RNA. Why? Uracil is there. Simple. Now, adenine is 20%, but thymine is not 20. It is 10. So it is going to be single-stranded DNA. It cannot be double-stranded. Then, thymine is 20%, adenine is 20%, cytosine and guanine. This is going to be double-stranded DNA. Here thymine, and it is again double-stranded DNA. That is also the way to check it. Is that clear? Is that clear? Sure. So this is all about the structure of the DNA. Okay. Now this is the diagram. You people can clearly see 5' end is having phosphate. Okay. Important to note. 3' end is having hydroxyl. Important to note. Adenine, thymine, 2 hydrogen bonds. They are projecting inward. Cytosine, guanine, 3 hydrogen bonds. Okay, and moreover, the stacking also providing it what? The stability. So 3' end OH, 5' end is having the phosphate. So that's what you have to keep in your mind. That's it. So here you can see the zDNA is going to be the left-handed one. Rest everything is right-handed. So here you can see some differences also, which is important. Just a double-stranded molecule is arranged in a helical structure. It's DNA. It is single-stranded, right? Each strand is a polymer of nucleotides. That is also a polymer of nucleotide, right, students? So DNA में adenine, thymine, cytosine, guanine. RNA adenine, uracil, cytosine, guanine. So DNA stores genetic information. Important. In the starting of the lecture also, I told you this thing. Okay. So this one translates genetic information, or I would say this is good for transferring or passing of genetic information. Understood? Only present in DNA viruses. In viruses, if you ask me the function, so in some viruses, it is also acting like what? It is also acting like the genetic material, okay? So that's it. So this is all about this part. So, so let's discuss the DNA packaging, and then let's talk about the central dogma of the information. And moreover, why RNA and DNA is better? Why DNA is better than RNA? What are the changes that occur in the DNA? Right? All that things we will discuss. So firstly, we'll focus on the packaging. Now, if you ask me about the break, we just started at 2:20. So obviously, you'll get the break at first break at 5:20 or 5:30. Then we will continue. You'll have your first break at 5, hopefully. Then 5:30 will continue. Then 8:30 will 5:30 will start. We'll continue till 8:30, and then again half an hour break will be there for the dinner. So breaks are also packed, and we have to finish this chapter, right? And we have to complete everything. That's why I started early. So let's talk about the DNA packaging. Okay. See, if I talk about humans, right? In our case, the length of the DNA is 2.2, approximately 2.2 meter long, And if I talk about the nucleus, that is so small. And we have to fit in that DNA there. And how can we check the length of DNA? It's simple. You should know the number of nucleotide. So if you know the number of nucleotide, if you know the distance between that two, you can multiply it and you'll get to know your answer, that how long our DNA is. So DNA is a long thread. It's a long thread and we have to pack it in a microscopic thing, right? We have to put it in a microscopic thing. So what's the way out? We will pack it. Like suppose in your almirah, if you do not arrange your clothes well, right, after a certain point of time, even you will not be able to put all the clothes. Or if you have too much clothes, and plus it will not be easy for you to take it out. Now, if let's suppose your almirah, if it is well organized, right, you'll have more space to accommodate other clothes as well, right? Okay, this is what we know. Okay, so now what am I trying to tell you? Now we have to pack that. We have to put this long DNA in that small structure. So what can we do? We can go for the packaging. So I will start with the prokaryotes, right? I'll explain you first, then I'll share the notes with you all. So in prokaryotes, we know it very well, right, students? We know it very well that our genetic material in the prokaryotes, like your bacteria, like your kingdom Monera, the genetic material is directly there in the cytoplasm. It is known as nucleoid. Plus, we know that it is devoid, means without, devoid of histone proteins. It is what? It is devoid of histone proteins. That's what we know, isn't it? See, prokaryotes, this is the cell, this is the cytoplasm that we have, this is the genetic material. We call it as nucleoid, not just nucleoid, you can call it as genophore, right? It is also referred to as naked DNA, right? And sometimes, because here there is no, as per NCERT, no well-defined nucleus in prokaryotes. NCERT, it doesn't say that there is no nucleus. As per NCERT, no well-defined nucleus, right? No well-defined nucleus. So that is also referred as incipient nucleus. Okay, and here, because the DNA, it is double-stranded and it is circular and even haploid. Some basic points I'm discussing here. And what? And haploid. So prokaryotes means you're talking about bacteria, members of kingdom Monera. You discussed that the genetic material is directly there in the cytoplasm. Plus we discussed there, we call it it is devoid histone proteins. There are no histone proteins there, right? It is also referred to as genophore, naked DNA. It is circular, though it is haploid, it is double-stranded. So here it is also referred as prochromosome, right? We also call it as prochromosome, right? Prochromosome. Now, do we need to pack this as well? Of course, we need to pack it. Like here in your syllabus, if you talk about Escherichia coli, right, you'll see the DNA length is also there in the millimeters. So when we have to pack it, here also we need some positively charged protein, and that proteins are known as nucleoid associated proteins, right? They are also the proteins which are present, right, which are required, I would say, for the packaging here. Nucleoid associated proteins, of course, positively charged because our DNA is negatively charged. Why? Due to the presence of phosphoric acid. Now, in the case of prokaryotes, we talk about the amines, polyamines, right? That are also basic in nature, right? Amines are there that also help in the packaging. So you even discuss about polyamines. We also even discuss about nucleoid associated proteins there. So when we talk about the DNA packaging in a cell, just remember, right? Total base pairs in a typical mammalian cell, right? Total base pair in a typical mammalian cell is 6.6 into 10 raised to power, right? 6.6 into 10 raised to power what? Anybody? 10 raised to power 9 base pairs, isn't it? 6 into 6, 6.6 into 10 raised to power, like in a diploid human cell, it's going to be 6.6 into 10 raised to power 9 base pair. And in haploid, you'll simply write it 3.3 into 10 raised to power 9 base pairs. So you are talking about the humans here. Yes, please. You are talking about the humans here. And the distance between two base pairs, we know it very well. It is 0.34 nanometer or 3.4. 0.34 nanometer. So in meters, you can simply write 10 raised to power minus 9 meters. So it is going to be approximately 2.2 meter long DNA. Is that clear? So, and nucleus is 10 raised to power minus 6 meters. I told you, right? I told you that is 1 micrometer in diameter, nucleus. So DNA is long. So even in E. coli, right? In some books, 1.56 is also given. So in some articles, 1.56 nanometer is also given. So you can see 1.36 millimeter. It is not meter, millimeter. So you multiply it with 10 raised to power minus 3 meters, right? Distance between base pair is again same. So that's how you can calculate it even in the case of E. coli. So number of base pair even. Let's suppose you got this in the form of MCQ. How to check the number of base pairs in a DNA? So obviously, see, it's like this. Total DNA length equals to number of base pairs into distance between two base pairs. So if we have to check distance between two base pair, if these two things are given, again you can check it. Here it is in multiplication, here it will be in division. Or the another way out is, obviously the way it is written here, let me show you. So number of base pairs is equals to total DNA length to the distance between two base pairs. So if you know this, you will be able to calculate this. Understood? Yes, beta, these sessions are really very helpful for the CBSE board as well, because there also you have to discuss everything in detail. Okay? Okay? Sure. Done? So packaging in prokaryotes. So prokaryotes is having, as I said, they have single circular double-stranded DNA molecule, means haploid it is. So despite being much smaller than eukaryotic genomes, DNA is still long. As I said, 1.36 in E. coli compared to the size of cell, which is approximately 1 to 2, right? Approximately 1 to 2 micrometers. So in DNA in prokaryotes is also highly compacted. How? By using polyamines. So what these polyamines are? I told you in prokaryotes, P for prokaryotes, P for polyamines. So what are these polyamines, my dear students? So they are positively charged, of course. They neutralize the negative charge on the phosphate backbone, help in the condensation. So here in prokaryotes, always remember, no histone proteins. Histone proteins we only discuss in the eukaryotes. So here you are not going to see the histone proteins. Is that clear? So this ensures that even the long DNA molecules can fit inside a small prokaryote while remaining functionally active. So polyamines, obviously they are not the proteins, they are amines, polyamines. So here when you read this in detail, you get to know there are some nucleoid associated proteins, right? There are some nucleoid associated proteins that help there. Is that clear? That help there. Is that clear? Sure. Chaliye next. Okay, let's go for it. So next part. So in eukaryotes, what's the story? In eukaryotes, we'll go in the steps. Okay. See, in eukaryotes, double-stranded diploid DNA is there. So what about its packaging? What about its packaging? The very first step, right? We will discuss it in 3 steps. Nucleosome model is there. Solenoid model is there. Then radial loop model is there. So in the first case, nucleosome model, we will talk about certain proteins which we call as histones. What are they? Histones. So histones are actually what? They are basic proteins. When I'm saying they are basic proteins, it means they are positively charged proteins. What are they? They are positively charged. So they are positively charged. They are histones. Why? Because they have basic amino acids. Okay, students, what do they have? They have basic amino acid. So overall, if I say basic amino acid, so you have a trick that is Hall, right? It's not its one word symbol, right? Hall is just a trick. It's not one word symbol for these amino acid. They say one word symbol is different. Like when you write alanine, you write ALA. When you write arginine, you write ARG. So that's not what I'm talking here. All I'm saying is that this is just a trick to remember, and that trick is histidine, arginine, because arginine is basically it's a one letter symbol is R, and then L stands for lysine, right? So in the case of histone proteins, the basic amino acids are lysine and arginine. What's that? That is lysine and arginine. It was in 2020, 2021, so it's a repeated MCQ. This one is what? It's a repeated MCQ. Am I clear? So these are the basic amino acids that we see in the histones. So basic amino acids, which basic amino acids are there? Lysine and arginine, right? Lysine and arginine. Now, there are five types of histones that we discuss. How many types? 5 types of histones are there, like H1, H2A, H2B, H3, H4. Is it important? Of course. This one is also important. So H1, H2A, H2B, H3, H4, these are the 5 types of histones that we see here. Now, out of that, right, these 4, they form the core of nucleosome, right? Core of nucleosome. This one is actually the linker histone protein. This one is what? This one is actually the linker histone protein. Okay, so what does it mean? See, you see that there is H2A, H2B, I'm randomly just arranging it, H3, H4. There are 4 histone proteins. Two copies of these histone proteins, they are going to make an octamer, right? Two copies of these histone proteins, this is going to make an octamer. Octa means 8. So basically, 2 copies of H2A, 2 copies of H2B, 2 copies of H3, 2 copies of H4. So in totality, it is an octamer. In totality, it is an octamer. So when DNA coils over it, right, then you will call it as nucleosome, which is basically a basic unit, right? It is actually a basic unit of the packaging here. And moreover, this H1, right, this is the linker histone protein. So linker histone protein is not the part of octamer, but it helps in connecting. So that is it. Is it important? It's very important. So nucleosome is that basic unit of chromatin. We discuss it in cell. Chromatin. Chroma means color. So chromatin is basically DNA plus protein. So how can you see this? You can see it in a different, in this way. So that's how it is. So that's what you can see. Okay. So here you'll see the H1 linker histone protein is also associated with it. Is that clear? So this is your one nucleosome. This is your next. So you can see. So we started with the DNA that is having diameter as 2 nanometer, and here in some references 10 is given, in some references 11 nanometer is given. So that's what it will form, from 2 to 10 or 11. Okay, so octamer is having what? Nucleosome. Octamer is having what? These proteins. Now see, this DNA in between these two nucleosomes, it is the linker DNA, right? It is the linker DNA. And students, this DNA is transcriptionally active. Do you know that? Transcriptionally active means I can use the information of this DNA to make this DNA is capable of making mRNA that will form protein. And this is how the information flow in a living body. DNA forms mRNA, mRNA forms protein if DNA is the genetic material. So this DNA is transcriptionally active, like I can access the genes of this DNA. So this DNA is known as euchromatin. What do we call it? We call it as euchromatin. What do we call it? Euchromatin. U means true. And this euchromatin is lightly stained. When you stain it, you will not say very dark. It is lightly stained. Are you getting my point? So it is transcriptionally active. Right? It is not very tightly packed. Now imagine if the DNA is very tightly packed. I will not be able to access it. I will not be able to open it. I will not be able to make the mRNA. So that DNA, which is transcriptionally inactive. So if DNA is transcriptionally inactive, means the genes that DNA contains, we will not be able to express. So that transcriptionally inactive DNA is called heterochromatin, students, and that is tightly packed and darkly stained. Right? Tightly packed. One second. So that is tightly packed, darkly stained. Arey, what is happening? That's heterochromatin. And this one is what? This one is loosely packed. So that's what you have to remember. Okay, so when it is seen under the electron microscope, so it appears like beads on string, and there are around 33 million nucleosomes and around 200 base pairs are coiled over it. Just here you guys can see, just look at this particular diagram, right? You started with the DNA, 2 nanometer. It is coiling over the histone proteins, right? These are the nucleosomes. So as per the books, it is like approximately 200. In some books, 147 base pairs are given. So this DNA packaging, see, the first level of DNA packaging is the nucleosome model, my dear students. DNA will wrapped around an octamer of histone protein. So this is H1, H2A, I'm repeating it twice, H2A, H2B, H3 and H4. So in octamer, these four are making the octamer. So you can even make your short notes. So here you can see histones are positively charged, right? So which amino acids do they have? They have lysine and arginine. Again, a repeated PYQ. Okay, so these amino acids, why? They have positive charges in their side chains, right? These amino acids are having more than one NH2 group. So more positive charges there. Then positive charge will attract the negatively charged phosphate groups of DNA. So this interaction will help in the binding of DNA. So this model was proposed by Kornberg and Thomas. Next, 5 types of histone. Core, that core will be made by H2A, H2B, H3, H4, right? 2 of each, so DNA will wrap around it. So each nucleosome contain approximately 200 base pair. But if you go for the standard book, students, let me show you. First of all, see, these are the histone proteins. They'll form, slowly, they'll form dimers. 2 copies each, so octamer. So this is octamer when there are only histone proteins. When DNA will coil over it, then you call it as nucleosome. Okay, then you call it as nucleosome, right? right? Understood? Next, see in between linker DNA, okay? Okay, so look at this again. So no doubt in some books of 12th grade level, they mention nucleosome are approximately having 200 base pair, but here, right, if you follow the standard books, right, they mention 146 nucleotide pair, double helix या 147. So you can see beads on a string and this will form the chromatin. So nucleosome will include, right, मतलब these 146 nucleotide pair are there in DNA double helix, and these nucleosome includes, see these nucleosome, they are saying these 200 nucleotide pairs of DNA, right? So this is the core. So nuclease can digest the linker DNA here. So the nanometer of each nucleosome here is 11, right? 11. Is that clear? Matlab, if we release the nucleosome core particle and when we check its diameter, it's approximately 11, right? It's basically 11 nanometer, right? 11 nanometer. So now when you dissociate, this is, see, this is a complete sequence. I hope it's visible. This is beads on the string. In between linker DNA, these are the nucleosome. So obviously it is actually forming this DNA with histones and all. This is forming the nucleosome. Now when I use the nuclease which digests the linker DNA, when I get this portion separately, so I am able to see that the diameter is 11. So we started with 2, we reached to 11. Plus in this released nucleosome core particle, when we dissociated it with some salts, octameric histone core is separated and there are 146 nucleotide pair. So what am I trying to tell you? Just say, see, if this is the octamer, this white part is the octamer. Now when DNA is coiled over it, then you are saying it as nucleosome. So now when you include this part, like this part, then you are saying 200 base pairs. Like linker DNA plus the DNA which is wrapped over this octamer, 200. Now let's suppose if I separate this, right? If I separate this, if I just consider the DNA which is wrapped here, right? So in that case, I'm saying approximately 146 or 147 nucleotide pair. Now this confusion clear, everybody? Now this confusion clear? So when you are taking nucleosome, when it is, when you also consider linker DNA, right, and the DNA which is wrapped over the nucleosome, then you are saying 200 nucleotide base pairs. Otherwise, only that DNA which is just wrapped over the histones, that is having 146 nucleotide pair. Okay. Done. So see, this is the actual diagram. This is the histone octamer. And this one that I'm marking with the red is the wrapped DNA. And the one that I have marked with black. So this is your H1. This is your H1. Can you see that? This is what? This is the H1. Is that clear? Is that clear? Okay. Now, when you talk about the H1, this is not like, it is not given in NCERT, just for your knowledge. H1 is very lysine rich. It is H2A which is having lysine arginine rich. H2B is moderately lysine rich. H3 argin rich, and even in H4 along with arginine, glycine is also there, right? Glycine is also there in the H4 histone protein, right? In the H4 histone protein. Now this beads on string thing has formed, right? Now the second level of packaging is solenoid. It's simple. Just say, let's suppose this is your double helical structure of DNA. Okay, you have started coiling it. right? Say, let's suppose this is one structure you have same same somewhere here, okay? Same here. They say this is one nucleosome in between linker DNA. Now you're further packaging it, further packaging it. That is actually the thing here. Okay, that is actually the thing here. So it is proposed by Finch and Clerk, 1976. So nucleosome, they coil, right? These nucleosome, they actually coil to form a solenoid wire. You might have you have studied that in physics, solenoid. So just like solenoid wire, okay? So it coils to form a solenoid wire, to form a solenoid fiber, and that will have 6 nucleosome per turn. Imagine this solenoid is having approximately 6 nucleosome per turn. So this solenoid, let's say last time it was, the nucleosome is having a diameter of, this one is having a diameter of, as I said, in some articles 10 is given, in some 11 is given, right? So let's go for the 11 mentioned in the foreign author books. So then it will further coil. It is forming the solenoid, that is 30 nanometer. That is 30 nanometer. 30 nanometer means 300 angstrom means, right? 300 angstrom. So this will give rise to 30 nanometer chromatin fiber. So first DNA will coil over histones, then beads on the string, that chromatin is formed, then you are further coiling it, forming a solenoid which is having a diameter of 300 nanometer. Then there is the radial loops or the dangler strings. So then they will further pack. So you know, they'll form some loops like this, right? That solenoids are further coiling, right? They are forming the loops like this. So when you see this chromosome having single arm, its diameter is 700 nanometer. But when you see the chromosome like this, having 2 chromatids, so 700, 700, here you'll see 1400 nanometer. Okay. Okay, so this is the radial loop or the dangler string model. Here you need acidic proteins as well. Here we will also talk about the non-histone proteins, and they are not basic, they are acidic. So you guys can see the packaging of chromatin at higher level. The packaging of chromatin at higher level, it requires additional set of proteins. They are non-histone chromosomal proteins, NHCs, right? And this information is important, that they are acidic. NHC. So NHC stands for non-histone proteins, and they are not basic. Histone proteins are basic, they are acidic. So this is the next level of the packaging. Okay, so each chromosome has 1 or 2 scaffolds at the center made up of these, like these non-histone proteins are there. They're helping in further packaging. They are going to form some radial loops, and that loops will further form some, they will project, they will form some other loops, right? So entry and exit of loops are close together, students. So these loops further undergo the folding, compaction, they'll form the chromosome. So this is actually the diagram that I want to show you. Here you can see we started with 2 nanometer, right? Then this linker DNA and all. So if both of it's considered 200 base pair, if only this part, if only this DNA is considered, then 147 base pairs or 146 nucleotide base pair. So this is solenoid, further packed. This is solenoid. Then it will form these 300 nanometers, these, loops that will further coil, will form radial loops, lateral loops. These radial loops will further form lateral loops, having both the ends closed. 700 nanometers, 700 nanometers, and there you have 1400 nanometers. Just allow me 1 minute. Done? So 2, 11, 330, and then see radial loops on a scaffold. Scaffold means a place like which is providing the site for the attachment. That is also you can say. So that is how it will go. Then chromatid, right? So this is about the complete packaging here. One more diagram. So see, I hope now this diagram, it clears all your doubts. Okay. Done, students? Okay. So now the next point is the difference in the euchromatin and heterochromatin. The NCERT part I have already mentioned you. So euchromatin is loosely packed. Heterochromatin is densely packed. Euchromatin is lightly stained under microscope. Heterochromatin is darkly stained. Euchromatin is transcriptionally active. This is transcriptionally inactive. So these points are given in your book, right? And moreover, euchromatin is rich in functional genes, and in heterochromatin, it contains mostly repetitive DNA, non-coding DNA. Then this euchromatin, in the nucleus, it is located in the inner or in the central region, and in the heterochromatin, it is near the nuclear lamina. Okay, this is important. Next, in euchromatin is comparatively obviously less coiled, so transcriptionally active. Heterochromatin is very compact, right? So in euchromatin, histone modification is there. They are more acetylated. That is why the gene expression is more. They are less acetylated, right? So euchromatin replicates early during S phase, heterochromatin late during S phase. So this is what you have to remember. So euchromatin is present in both pro and eukaryotes, but heterochromatin is only there in the eukaryotes. So this is all about the packaging. So the next topic that we have to discuss is the search for the genetic material, and here we start with the Frederick Griffith, right, students? So up to this part, if there is any doubt, do let me know in the chat section here. Yes, please do let me know in the chat section here. So by 1926, the quest, the search that what is behind this, inheritance, it started. It reached the molecular level. Like it reached at molecular level because people were aware about the laws of inheritance, but what at molecular level is passing to the next generation, that quest has started in 1926. So previous discoveries by Gregor Mendel, Walter Sutton, Thomas Hunt Morgan, which we have also discussed in the last class, it had narrowed down the research. So one thing was clear, that there is something which is basically present in most of the cells, and that too in the chromosome. So then when we talk about this, there are n number of experiments, but we talk about some basic, some main experiments. Here we have the first experiment that we have to discuss is the transforming principle. Which experiment do we have to discuss, students? We have to discuss the transforming principle experiment, as I said, which is given by Griffith, So what he did in 1928, right? Frederick Griffith, he experimented, right? He used the mice, of course. Then he used the bacteria. So when we talk about the bacteria, right? He used Streptococcus pneumoniae, obviously, which causes what? Pneumonia. It is also known as Diplococcus, and it is also known as Pneumococcus. Is that clear, students? Now, when you talk about this particular bacteria, remember, coccus means we are talking about the shape here, spherical, right? Spherical. Are you getting my point? Spherical. Yes or no? So that is the meaning of the coccus here, spherical shaped. Now this particular bacteria is having different strains. You know that in the case of prokaryotes, if we study them at the molecular level, they vary a lot. Okay, and we haven't developed the techniques yet that we can culture all that bacteria in the lab and we can even study all of that. That's why whenever we talk about the number of species on this planet, we go for the eukaryotes. We don't even discuss the prokaryotes. Now when you look at the streptococcus, so as I said, how many strains are there? There are two strains. One is S3, another is R2. Okay, so when you talk about this S here means smooth, and it is actually virulent one. It is actually what? Virulent. What is the meaning of virulent? Virulent means pathogenic. Pathogenic means disease causing. R here stands for rough. It is, of course, it is non-virulent, means non-pathogenic. Are you getting my point? This one is non-virulent and non-pathogenic. So you know that in a bacteria we discuss cell membrane is there, cell wall is there, and then they are also having a glycocalyx, cell coat, which can be having a slime layer or the polysaccharide coat. Now when you talk about this S strain, it is the virulent, pathogenic, means it causes pneumonia that can kill the mice. Now here we see that in the smooth strain, right, such bacterial strains, they form a polysaccharide coat, and actually that is the polysaccharide coat that is virulent. Means when we grow this particular bacteria in the culture, the colonies, they appear smooth, right, having the polysaccharide coat, and here they appear rough, right? So here we see rough colonies, right? Here we see what? Here we see smooth colonies. Here you see rough, here you see the smooth colonies. So why they are smooth? They are having the polysaccharide coat that can cause the virulence. Now, see, when he conducted this experiment, this is what he noticed when he injected the S strain. So S strain, as you know, pathogenic, disease causing. R strain is non-pathogenic, means it is not going to cause the disease. Yes or no? Yes or no? So when this S strain, when it was injected into the mice died. Why? Due to pneumonia, of course, right? Due to pneumonia, of course. This R strain, mice live. Now, in third experiment, what he did? He heat killed this S strain. Due to that heat, what's going to happen? That polysaccharide coat will be destroyed, obviously, right? So that has killed that bacteria. So mice will live because this bacteria, this is the heat-killed S strain, so it will not be able to cause the pneumonia. Now, in the last experiment, what he did? He used heat-killed S strain, which is again non-pathogenic, and also used R strain, which is again non-pathogenic. So if both are non-pathogenic, because this one is pathogenic, mice die. This one is non-pathogenic, non-disease causing, mice live. Heat-killed S strain will destroy that polysaccharide coat, which is responsible for the virulence, right? It has killed basically that bacteria. So obviously mice is going to live. But now here, what's the point, students? Here the point is that S strain is also the heat-killed, right? R strain is what? R strain is live. S strain is heat killed, R strain is live, but both are non-pathogenic. So he was expecting the mice to live, but it died. That is basically the question mark. That how is it possible? Even the heat killed S strain, even the heat killed, right? Even the heat killed S strain, just a minute, guys. So in heat-killed S strain, that is also non-pathogenic. R strain is also non-pathogenic, right? Then how is it possible? Let me silence my phone. Okay, so how is it possible? How is it possible that mice can die? So then he came up with a conclusion and he called it as the transforming principle. Are you getting my point? Transforming principle. So he concluded that the R strain bacteria, right? What he concluded? He concluded that the R strain bacteria had somehow, right? It was somehow transformed by heat-killed S strain. Like heat-killed S strain had transformed R strain bacteria. This is what he concluded. He called it as transforming principle, but he didn't mention that DNA is the genetic material. He said that something from that heat-killed S strain will convert that R strain into colony forming or polysaccharide coat forming bacteria. And do you know this experiment, it also tells us about the stability of the genetic material, that even after the heat it was not destroyed. We'll discuss it further. So he concluded that the R strain bacteria had somehow been transformed by the heat killed S strain bacteria. This point is clear. Then some transforming principle is transferred from that heat killed S strain had enabled this R strain to synthesize a smooth polysaccharide coat, right? That is why it was able to do that, and it became virulent, right? So it happened because of the transfer of the genetic material, but he was not aware of that nature. Okay, so this is about the transforming principle. Then next experiment is biochemical characterization. Again, a previous year question, who did so? Right now, many scientists, they tried many things. Now from that heat killed S strain, they tried to isolate proteins, carbohydrates, nucleic acid, DNA, RNAs, right, like this. Then they checked that what actually is responsible for the transformation. Like, I'll give you an example, then I'll talk about it. Just say, let's suppose if there is a test tube, just giving you a layman example to explain this, right? Just say, let's suppose here you have R strain, live R strain. Just giving you one example so that you can understand what actually is happening. Now I have added the content from the heat killed S strain here. Matlab, this is having the live R strain. Here I have added the components of that heat killed S strain. And let's suppose if I added proteases here. Proteases, what that proteases will do? It will digest proteins. We are aware of it, isn't it? So obviously, protease will digest the protein of this heat killed S strain, but we know that it is the DNA which is the genetic material. They were not aware by then, but we are. So obviously, we know that in this case also, transformation occurs. Just giving you one example. Now again, in another test tube, let's suppose if I add carbohydrates with that, for that heat killed content, heat killed bacteria's content, so that will destroy the carbohydrates, right? That will, again, the transformation occurs. Again, the transformation in that R strain will occur. Then lastly, like, let's say now here if I'll add RNAs in the same setup, if I add RNAs, what is the meaning of RNAs? It is RNA digesting enzyme. What's that? It is RNA digesting enzyme. We know that still the transformation occurs. But if we add DNAs, what is this DNAs? In the NCERT it is given the difference in DNA and DNAs. So it is DNA digesting enzyme. Yes. What's that? It is DNA digesting enzyme. Yes or no? So there will be no transformation. There will be no transformation. Why? Because if DNA is destroyed from that heat killed S strain, then obviously it will not be able to convert that R strain into the S strain. So such types of experiments were performed. So who did that? MacLeod, McCarthy, Avery, Oswald, Avery, MacLeod, McCarthy. So prior means before the work of, because this biochemical characterization is done by them, Oswald Avery, Colin MacLeod, MacLean McCarthy. Before their work, it was considered, protein was considered as the genetic material. Before their work, protein was considered as the genetic material. Now, they worked to determine the biochemical nature of this transforming principle in the Griffiths experiment. So they purified biochemicals like proteins, DNAs, RNAs, right? From that heat killed S cell. From that heat killed S cell, they purified all the things so that they can check which one can actually transform R cell into the S cell. They discovered that DNA alone from S bacteria. They performed the experiment and this is what they concluded, that it was the DNA from the S bacteria that transformed it. They also discovered that protein digesting enzyme proteases, RNA digesting enzyme RNases, they did not affect the transformation, as I told you. So the transforming substance was not a protein or RNA. So because when the digestion was done with the DNAs, it did inhibit transformation. So yes, in the NCERT it is written what is the difference in DNA and DNase. DNA, deoxyribonucleic acid. DNA is the enzyme that can digest the DNA. So it suggested that DNA could cause the transformation. So they concluded DNA is the hereditary material, but not all biologists were convinced, right? Not all biologists, they were convinced. So I hope this part is done. Yes. I hope this part is done. Sure. Then next to it is the unequivocal proof that DNA is the genetic material is given by Hershey and Chase. So that's the next experiment that we have to discuss. So in 1952, Alfred Hershey and Martha Chase, they gave the experiment which is also known as transduction principle and the blendings experiment that provided the proof that it is the DNA which passes from one generation to the next. So that's what we have to discuss. Then we will discuss these points from the notes. So first of all, I'll start with some basic terms, right? Like in the batch also we have discussed it. Transduction. What is transduction? See, this is a bacteria. When a virus is used to transfer the genetic material from one bacteria to another, this is known as transduction. Like see, I hope you all are well aware of the structure. This diagram you will even see in this chapter in biological classification. Even students, we talk about this structure in human microbes in human welfare. So you should know about this diagram in simple words. That's what I want to tell you. So it is what? It's a bacteriophage. What is it? It's a bacteriophage. What is it? It's a DNA, mostly I would say. So actually it's a virus that infects DNA, that infects bacteria. What is this bacteriophage? It is a virus that infects bacteria. So bacteria itself causes a disease. Virus is causing harm to that bacteria. So you can also call it as hyperparasite because it is a parasite on parasite. So bacteriophage. So mostly I would say, you'll say it's a double-stranded DNA virus. Mostly what do we study? That it's a double-stranded DNA virus. Though it's not like always they are like this. No, we have the example of QB bacteriophage, again given in NCERT. There is a QB bacteriophage. That's actually an RNA virus. That's not DNA virus. That is RNA virus. Do read your NCERT well. This information is again from the NCERT. Now, what I want to tell you. So this bacteria, with the help of these tail fibers, it attaches itself to this virus, with the help of its tail fibers, it attaches itself to the bacteria surface. Then it injects its DNA in that particular bacteria. Like here, you have the bacteria, and in this experiment, they have used E. coli, that is your Escherichia coli. Escherichia coli. Now, they have injected the DNA in it. This virus has injected its DNA in it, in bacteria. So you know that what's going to happen. Obviously, this viral DNA will use the host machinery, will make its multiple copy. How? It will incorporate itself into the host DNA. It will incorporate itself into the host DNA. It will use that host machinery that will then start synthesizing its own DNA. Then multiple copies, right? Within this cell, you'll see there will be multiple copies of this bacteriophage, and this will contain the DNA from this bacteria also. Its own DNA and the DNA from this bacteria also. And now when these viruses, when they will get free from this particular bacterial cell, they will infect another bacteria and they'll again pass their DNA. So that is also a way, that this particular virus is passing the DNA of this bacteria to another. So when virus is used to transfer the genetic material from one bacterial cell to the another, this is also known as transduction. That's why this is again a transduction experiment, right? And let me tell you this thing here, students. Let me clarify this thing here, right? And you have to keep it in your mind, right? That already we are aware, right? That in the virus, this outer part, right? This outer part is actually proteinaceous. This is made up of protein, and it is Inner part is having this genetic material which we are calling as DNA. I hope you are aware, and we know that DNA passes, but they were not aware. So what this experiment is all about? This experiment is having 3 steps: infection, blending, and centrifugation. First of all, like 2 setups were there. Setup number 1, setup number 2. So they use the bacteriophage, T2 bacteriophage it was. So in first setup, they grew this virus. Obviously, if we have to grow the viruses, we cannot grow it in a medium where cell extracts are not there, because viruses outside the body, they are non-living. They are just the inert crystalline proteins. So if we want to culture the viruses, we have to put the cell. We have to put the cell extract. Is that clear? We have to put the cell. We have to put the cell extract. Am I clear? Am I clear? Sure. So now what's going to happen here? They grew that bacteria in a culture and they are the radioactive. Just say, let's suppose S35 isotope was there. Which isotope was there? S35 radioactive isotope. Or we can simply say, because we have to trace which one will pass to the next generation. And in this setup, they use P32, right? Radio labeled isotope. Is that clear? Now, in the first step, as I said, infection will be there. Just say now, when we'll grow this bacteria, right? Now, when we grow this bacteria, in what? In what? In the radioactive sulfur. You know that this sulfur, where will it get incorporated? Into the protein. Because we know that we have, in the protein, we have sulfur-containing amino acid. Do you remember that? Sulfur-containing amino acid? Do you remember the sulfur-containing amino acid? Right, let me write it here. Sulfur-containing amino acid. For that, we have a trick. I think I have discussed it multiple times. CMS, that is Chief Minister Sir. Right, Chief Minister Sir. So what does it mean? What does it mean? It means it is cysteine methionine. C stands for cysteine. M stands for methionine. So that is what? That is a sulfur-containing amino acid. So C stands for cysteine, M stands for methionine, S stands for sulfur. So it is the sulfur-containing amino acids. So protein like S35, where will it get incorporated? S35 in the protein coat, and this one will be in the DNA. Are you getting my point? This one in the DNA. Is that clear? Now 3 steps are there, right? Just say for radioactivity, let's go for the red color. So this time you know that radioactivity will be there in the outside part. Here the radioactivity will be there in the DNA. So first step is infection, right? First step is infection. In this first step, what are we supposed to do? In this first step of infection, these radio-labeled, right? These radio-labeled bacteriophages, we want them to infect the bacteria. That is the first step of the experiment, which we are calling as what? Infection. What do we call it, students? Infection. So in the first step, we want these bacteriophages to infect the bacteria. Now, the second step is blending. Right? The second step is what? The second step is blending. Right? From this blending, you can remember from this B, break. This is what you can remember. From this blending, from this word B, you can remember break, right? What can you remember? You can simply remember the word break, okay? So here in this step, right, basically we'll try to break, or we break the contact in between the virus and bacteria. In the first step, we have provided that this sufficient time, right? What we wanted, we just simply want that bacteriophages, they should infect the bacteria. So how can they infect? We are already aware of that they'll pass their genetic material. Then only they can infect the host cell, and they can use the host machinery and can make the multiple copies. Now we have to check that what exactly passed to the next generation, right? So obviously second step comes the blending, where we will break the contact in between these bacteriophages and the bacteria. In between this virus and bacteria, we will break the contact. And the third step is centrifugation, right? The third step is centrifugation. So in the centrifugation at high speed, right, we'll rotate this content and we'll separate the viral and bacterial content. Why? Why? Because we want to check that in next generation, like in bacteria, what was the thing that passed? Like radioactivity. We have to trace the radioactivity here. So in centrifugation at high speed, we'll rotate this. We want to separate this bacterial component and the viral component. You know one thing, right? That virus is having less weight than the bacteria. Bacteria is heavy. We know it. So obviously heavy things, they'll settle down at base. So the base thing, right? In the during centrifugation, the heavier substances, they'll settle down at the base. Now, heavy things, they settle down at the base. Now, so heavy things, they will settle down at the base. So this base part will show us what? This palate part will show us what? Just let's suppose this upper part is supernatant and the base is having the palate. So this palate part is showing us the bacterial component and the upper part is showing us the viral component because it is lighter. So in this experiment, when S35 isotope was used, so radioactivity was there in the viral component only. So radioactivity in this case, radioactivity, because we have to trace it, it was in the supernatant, right? Supernatant shows the viral component. Now here in this experiment, radioactivity, right, when radioactivity P32 was traced, radioactivity was there in the palate. So palate is showing what? Palate is showing the bacterial component, right? Palate is showing the bacterial component. So obviously what we got to know that in bacteria, the DNA of the virus pass. So it is the DNA which actually passes from one generation to another. So that's how they perform the experiment. You first have this look, right? The very first step is infection, right? So see, the genetic material will be passed here, right? Then in the blending, we are breaking the contact because we want to see what has passed to the bacteria. And here, right, we detected the radioactivity in centrifugation method. So no radioactivity was detected in the cell. Radioactivity was detected in the supernatant. So supernatant here is showing what? It is showing the viral component. So it was there only. So it was not the protein which passed to the bacteria. Now here again infection, again blending. So radioactivity was detected in the cell, right? It was detected in the cellular component in the bacteria. So no radioactivity was there in the supernatant. So that is what they concluded. That's how they provided this proof that it is the DNA which is the genetic material. So let's discuss this, guys. So see this, the unequivocal proof that DNA is the genetic material. It came from the experiments of Alfred Hershey and Martha Chase. Year 1952. Is it important? Of course, it is important, right? It is important. Then bacteriophage, that is virus, attaches to the bacterial cell, injects its genetic material inside. So bacterial cell uses this viral genetic material to produce new viral particles. We are aware of this fact. So the main aim of this, to find out what was the aim of this experiment, to check out whether it's DNA or the protein which is the genetic material. So what they used? Radioactive labeling. Okay, so radioactive phosphorus P32, right, and radioactive sulfur S35. So yes, you should know P32 is radioactive phosphorus and S35 is sulfur. Okay, then virus is grown with P32 add radioactive DNA, S35 add radioactive protein. So after the formation of labeled phages, right, means the one which are having the radioactive elements. So 3 steps were done: infection, blending and centrifugation. So in infection, as I said, both labeled phages in 2 different experiments, they'll infect E. coli. Blending, mixture was agitated in a blender. B for blending, B for blender, B for break, so that we can detach the viral coats from the bacterial cells. Understood? Then in the centrifugation, what was done? The virus particles were separated from the bacteria by spinning them in a centrifuge, and after that, this is what we got, that bacteria which was infected with the virus that was having radioactive DNA, it indicated that DNA was the material that passed from the virus to bacteria, right? It is not the protein. So it indicates that protein did not enter the bacteria from virus. DNA is therefore the genetic material that passed from virus to bacteria. So these are the 3 steps here, right? This is what they followed, and that's how they provided the genetic material, that the proof that it is the DNA which is the genetic material. Is that clear? Yes. So just take 1 minute, recall it, and if there is any doubt, then tell me. If there is any doubt, then tell me. Quick. Quick. Yes, please. So next line is that why do we say, yeah, what are the properties that genetic material should have, and why DNA is better than RNA? This is what we have to talk, right? Guys, let's finish this part. Okay, let's practice 1 or 2 questions, and then we'll have a break, and then replication part, the semi-conservative proof and all, we will discuss after this. So, because this is just the theoretical portion, right? This is just the theoretical portion. Theory it is. You can take the notes later. You just understand it. Then there will be a break. Okay. So, in some viruses, we know that RNA is the genetic material. So, this line is given in NCERT. I told you before. TMV, tobacco mosaic virus, single-stranded RNA virus. Remember, where do you discuss it? Biological classification. Remember that red colored DNA in the NCERT and these green colored structure, right? We used to discuss. So tobacco mosaic virus. So that is the virus having RNA as the genetic material. So QB bacteriophage, today itself I talked about it. So yeah, so QB bacteriophage is also what? It is the RNA virus. So usually bacteriophages are DNA viruses, but this one is the RNA virus. So when we can say that that this compound can be a genetic material, obviously it should be able to generate its replica. Why? Because we know that, right? Rudolf Virchow has said, omnis cellula e cellulae. Cell comes from pre-existing cell, and we know that for the continuity of the life, right, even if it is a unicellular organism, multicellular organism, we all reproduce, our cells divide. This is important. So during the cell division, it is mandatory, it is important that our cells, what should they do? They should, right, what should they do? They will replicate. So genetic material also the genetic material should replicate, so that our dotted cells, they can get the equal amount of DNA, isn't it? Now it should be able to generate its replica, number 1. It should be stable chemically and structurally, number 2, right? Structurally, chemical and stable. Why? Because if genetic material, if it will show mutation at a faster rate, so if too much mutations are at a very faster rate, then obviously, right, it is showing that the genetic material is not stable, and that mutation, that so quick mutations, right, that can also result in the formation of a new species very early, right, which is again not good. So it should be stable chemically and structurally, then only it can just store the genetic information properly and then can pass it. So it should provide the scope for slow changes. So we are not saying that genetic material should be mutation free. Mutations should be there, but they should be at a slow rate, because that is what we need for the evolution. Because we know that for evolution, variations are important and mutation is something very important for that. And then it should be able to express itself in the form of Mendelian characters. So when we say this line, so we simply want to say that it should be able to, express itself, right? Just Mendelian characters like genes, yeah, factors, they were controlling a character, then they were passed to the next generation, they were stable enough, right? These were the points that we had discussed there. So it means that like Mendelian characters, what were they doing? They were just controlling that inheritable traits now, right? So the genetic material should have the such properties. So we know that because of the rule of base pairing and complementarity, adenine will go with thymine, cytosine will go with guanine. We know that DNA and RNA, they both have the ability to direct their duplication. Even RNA can form its copy and even DNA can also form its copy. Isn't it? Isn't it? Thank you, Lakshmi. RNA can also form its copy and DNA can also form its copy. Yes or no? Yes or no? They both can do that, right? Right. So other molecules in the living system, proteins, they do not fulfill this criteria. That is why it is the DNA which, so DNA and RNA, they are eligible for this to be the genetic material. Now, as I said, no need to write, you just have to understand it. See, mutability. Why mutability, undergo mutation is important for the genetic material? Why? Because if it will not occur, if such changes will not be there, then how the evolution is going to occur? Right? How do we evolve? For evolution, we know that variations are important, and for that variation, these mutations, the crossing over, it is important, isn't it? Isn't it? So when we compare DNA and RNA, RNA mutates faster. So it is having lesser stability. Lesser stability, mutate faster, it is not stable, and moreover, RNA can also act as an enzyme, ribozyme. So because it is having the catalytic property, again unstable. So that's why with the time, during evolution, DNA evolved from the RNA, right? Because with the time, what happened? We basically moved from simplicity to the complexity. So better stability was required. So that is why, so DNA mutates slower. So that's why it can maintain integrity across generation. We don't want something that just get mutated in the next generation. You take the example of RNA viruses. They mutate at a very faster rate. So they mutate at a very faster rate, they form too much of variants, and the more they form variants, they get weaker. That is also the case. Okay. Then next is expression of trait. The material must be able to express the stored genetic information via protein synthesis, and that's what we see in the case of DNA. We are going to discuss central dogma of information given by Crick. Okay, central dogma, we'll discuss it. That's all, also a PYQ given by Francis Crick, right? That's again a PYQ. So DNA, it is able to, genes on DNA are able to express itself by making proteins. So DNA form RNA, RNA form protein, isn't it? Isn't it? So proteins will decide the trait, the phenotypic expression, right, of a particular trait in an organism. So yes, the genetic material, the DNA is able to express itself. Then the genetic material is chemically stable. Under different physiologically condition, it should be able to survive, right? It should not degrade easily, or it should not change with the minor environmental fluctuations. That shows its stability, right? That is how it can maintain its integrity. Now in the case of RNA, you know that RNA is having ribose sugar, that 2-OH makes it reactive, plus it is also having the uracil. So DNA contains deoxyribose, which lacks reactive 2-OH group. Yes, that 2-OH group in the ribose sugar makes RNA more reactive. That's why during evolution, it was not there in the deoxyribose sugar. So double-stranded DNA, and DNA moreover is the double-stranded, means it is providing more stability, right? And other than that, you know that RNA is having the uracil, DNA is having the thymine, that also provide the additional stability there. So hydrogen bonding between complementary base pairs provide structural integrity. That's what you have to remember. So why do we say that DNA is better? And when it comes to the DNA stability, you can simply keep it in your mind that during Griffith's experiment, even after heat killing, the DNA still was able to transform that R strain into the S strain, which clearly shows its stability. Isn't it? Isn't it? So stability is something very important. It is a very important for the genetic material, right? So Griffith's transforming principle itself, that heat which killed the bacteria, did not destroy some of the properties of the genetic material. So that's a favor for the DNA. Then 2-OH group, which is present in the RNA, which makes RNA more labile, right? RNA more labile and easily degradable. So RNA is unstable due to this group that is not there in the DNA. Plus, RNA is known to be catalytic, right? We know that RNA can also act as an enzyme. And what do we call it? It is called as ribozyme. Again a PYQ, right? Again a PYQ. So that is again, it is making it more reactive. So the DNA is a better genetic material having better stability, and the thymine at the place of uracil is also providing the additional stability. So the RNA being unstable mutate faster. I have even given you the example of viruses. RNA viruses, they mutate at a faster rate. They form their various variants, and the more variants they form, it will make it easier. So that is what we have to remember, and that is something that you have to keep in your mind. So this information is very important. This is the third time I'm mentioning it. See, RNA can directly code for the synthesis of protein. Means RNA can easily express the characters. How the characters express? Right? Gene expression, no doubt we will discuss here. In the last chapter, we have discussed it, that characters they express, the phenotypic expression is there. How is it possible? At molecular level, you see the DNA will be there, that will form mRNA, that mRNA forms the protein. So it is the RNA that expresses the character directly. Right? It is the RNA that expresses that character directly, that RNA forms the protein. Right? So DNA is dependent on RNA for the synthesis. So somewhere it shows that RNA word concept is true, right? So the protein synthesizing machinery, right? We'll discuss it in translation. It has evolved around RNA only. The protein synthesizing machinery, it has evolved around RNA only. So it indicates that both can function as the genetic material, because RNA is forming the protein, and by making the protein, a character can be expressed. So obviously RNA can also act like a genetic material, and DNA, if it has to express the genetic, if it has to express itself, it has to make the RNA first. It can never make the protein directly. So it needs the RNA. So that shows us about the RNA word concept, right? Right? So that is why in the starting itself, I told you, DNA being more stable is preferred for the storage of genetic information. But when it comes to the transmission of genetic information, which one is better? It is the RNA. Isn't it good? What a lovely point it is, right? Sometimes we get the questions, we simply say that if it is written RNA, out of the following, which one will transfer the genetic information? You'll click DNA. No, it is the RNA. So RNA helps in transmission of genetic information, but when it comes to the storage, DNA being more stable, DNA is going to store it. Understood? Understood? So RNA word concept says RNA was probably the first genetic material, because all the life processes, be it the metabolism, translation, splicing, they all evolved around RNA. It is also having the catalytic function. So you know that when we discuss the evolution, this is what we see. Isn't it? This is what we see in the case of evolution as well, guys, right? That obviously if RNA, like there is a protein word concept also. Some people believe protein came first, but majorly it is RNA, because RNA can act like genetic material, plus RNA is also having catalytic properties. And you know that when catalysts are available, they increase the rate of the reaction which is required by them. And also we need it today also. So it had catalytic functions like ribosome and could replicate. So however, due to its instability, it was eventually replaced by DNA as the primary genetic material, because obviously we evolved, we need better things there. So DNA became dominant due to better stability, accurate information transfer, and efficient storage capabilities, and complementary strands are there which will help it to resist the change, right? To resist the changes by evolving a process of repair. So this is what we are going to study in this replication part, right? That how, you know, the DNA repair, this mechanism also evolved with time. Okay, so that's it. So rest we'll discuss after the break. Okay, so break time. So break time there is. So let's meet at 5:45 p.m. So you guys will get a break for half an hour. Am I clear? So for half an hour, you people can enjoy this break. Then we'll see you then. Hi everyone, welcome back. I hope you all are doing good. I hope you all are doing good. So let's get started. And what are we going to discuss now? Now we will start the replication part. Okay, so we are done with the basics of DNA. Like firstly, we started with the nucleic acids. What are nucleic acids? Right, students, the types. What are they made up of? Right, all the bonding part we covered. And now after that, what are we going to discuss? Yes, everybody. So we also covered the experiments and now we are going to discuss their application. Is that clear? Okay. So when it comes to the DNA replication, we'll study that the DNA replication is semi-conservative in nature and that is what we are going to study. But before that, let's talk about this part. Like, see, Watson and Crick, right? Watson and Crick, they proposed the scheme for replication of DNA. So please pay attention here. Watson and Crick proposed the scheme for DNA replication. They didn't tell us the detailed mechanism. They just proposed a scheme. Why? Because they studied the DNA structure. They noticed the complementarity there. And they were like, because we know adenine can pair with thymine, cytosine can pair with guanine. By using this, obviously, we can make a new strand of DNA. Isn't it? Isn't it? So Watson and Crick proposed a scheme for replication of DNA which suggested that two strands they would separate and can act like a template for the synthesis of new complementary strand. Is that clear? For the synthesis of new complementary strand. So after the completion of replication, each DNA molecule, right, after the completion of replication, each DNA molecule have one parental and one newly synthesized DNA strand. So this scheme is basically the semi-conservative nature of the DNA replication. It was proposed by them. So I'll give you an idea. Like let's suppose these are two parental strands, right? We know that complementarity is there. What does it mean? Like let's suppose if this particular strand, let's suppose you separate them, right? Just giving you an example. If adenine is there, thymine, guanine, cytosine, thymine, adenine. So obviously, if adenine is there, in front of that, what will come? Thymine. In front of thymine, adenine. In front of guanine, cytosine. In front of cytosine, guanine. Thymine, adenine, like this. So obviously, new strand can also be synthesized. This is what they noticed. So when we talk about this, so there are three different modes of DNA replication. So conservative mode, the first one is what? The conservative mode. So when you talk about the conservative mode, in that case, what's going to happen? Parental strands, they will open. They'll act like a template. What is the meaning of template strand? Yeah, what is the meaning of template? Like, sometimes, let's suppose if you have to write a mail, right, we Google it now, or in our books, right, in our English books it used to mention the template for this mail, the template for this letter. Like they'll give you an idea that this is what you can write there. Okay, that is a kind of template. Like wedding cards. Okay, so we go to the publisher, they give us different cards, no, that you can opt this, you can opt that, you can go for this, you can go for that. So that is actually a template. So according to your choice, as per your preference, you can just tell them and they can just publish it accordingly. Same is the case here. You have the like DNA strand which is having the basis. When you open that DNA strands, they act like a template. You can use them to copy the information. You can use that to basically add the complementary bases. Now, in the conservative mode of DNA replication, what's going to happen? Slowly, DNA will open, strands will open, complementary to that, bases will be added, but later on, parental strands, they join together and daughter strands, they again join with each other. Parental strands, they join together. So daughter strand, they also join with each other. So this is the conservative mode. Then comes the semi-conservative mode, which we have to discuss in detail. Now in the semi-conservative mode of DNA replication, obviously DNA will open slowly. Each strand will act like a template. And how the daughter DNAs will form? how that daughter DNA will form. So in the daughter DNAs, one strand will be parental, one strand will be the newly synthesized. Here, see, in the daughter DNA, this is the totally the parental DNA. This is the newly synthesized. That is going to happen in what? In conservative mode. You are conserving the parental information as such. Then the new daughter DNA strands are formed. Right here, one strand is going to be parental, right? And the another strand is going to be, it is the newly synthesized, or we can call it as daughter DNA, right? Or daughter DNA strand. Then comes this. This one is dispersive. This one is what? This one is dispersive. Now here, what you people will notice, see, what you people will notice, see this, in the dispersive, some part will be of parental strand, some part will be of newly synthesized strand. So this is what we study, the conservative, semi-conservative, and then comes the dispersive mode of DNA replication. Then comes what? Dispersive mode of DNA replication. So this is what we study here. Is that clear? This is what we study. I hope it is clear in your mind. Now, what's the next part here? See, as I said, now we'll talk about the experimental proof, but again I'm repeating, Watson and Crick, they proposed the model. They were not the one who explained it, right? They just proposed the model. So when it comes to the experimental verification, so for the very first time in the case of bacteria, that experimental verification was provided by Messelson and Stahl. Who provided it, students? Yes, everybody. Who provided it? Messelson and Stahl. Is that clear? Is that clear? Who provided it? Messelson and Stahl. Isn't it? Isn't it? So Matthew Messelson and Franklin Stahl, they performed the experiment in 1958 that basically provided the experimental proof for semi-conservative mode of DNA replication. That provided the experimental proof for semi-conservative mode of DNA replication. Am I clear? Semi-conservative mode of DNA replication. Is that clear? Is that clear? Done. So what they did, see, they experimented on E. coli, right? That is Escherichia coli. They experimented on E. coli. Normally, you know that when you talk about the DNA contains nitrogen. Yes or no? Yes or no? We know it very well that DNA contains the nitrogen, and which isotope of nitrogen is there? 14N. Normally, this is what we have, and it is the light nitrogen. Here we are not talking about the radioactive, we are talking about the heavy and the light. So this is the light nitrogen isotope. Then what happened? They took the E. coli, then they started growing it in a medium which is having, right? They started growing it in a medium. There is the culture medium, and in that culture medium, you know that if we have to, this is the culture medium. Here, if we have to grow the cells, the bacteria, we have to provide the food here. We have to provide the source of energy here, isn't it? We have to provide the source here. We have to provide this energy here. So now, what will you see here? You will see that here for the nitrogen, ammonium chloride was there, and that ammonium chloride was having, the formula is NH4Cl, and here it was having heavy isotope of nitrogen, right? Heavy isotope of nitrogen. So students, it is not radioactive or radio labeled. You just have to remember 14N is light isotope of nitrogen, right? It is the light isotope of nitrogen. And when you people talk about the 15N, this is going to be the heavy isotope of nitrogen. So it is not like radioactive, radio labeled, it is heavy or the light. Is that clear? It is heavy or the light. 14N is there and 15N is there. 14N is there and 15N is there. So now for several generations, they allowed this E. coli to grow here. Overall, in the case of E. coli, if you ask me the generation time, so in the text, 18 minutes is given, or in the diagram, I'm going as per NCERT, 20 minutes is given. So this is what you can note down, right? So 18 and 20 both are given in the NCERT. So now the point is ammonium chloride, they use the heavy isotope. They allowed the E. coli to grow there for some generations. And then finally, when that cells, when they got the 15N nitrogen in their DNA, then they took that E. coli cells. Okay? Why is it so? Because now they have to prove this semi-conservative mode of DNA replication. So now let's suppose this is the E. coli, this is the DNA of E. coli, and this DNA of E. coli, is having what? It is having the 15N isotope of nitrogen. Which isotope of nitrogen? 15N, that is heavy isotope of nitrogen. Then again they shifted this E. coli, right? Again they shifted this E. coli in a medium having again ammonium chloride, NH4Cl, and this time again the light isotope. So this E. coli is having 15N DNA. Now they have shifted it to a medium having 14N isotope. Now let's suppose this is E. coli. Sorry. So let's suppose now this is 15 and 14, and what are they trying to do? Heavy and the light, right? Obviously here we are talking about the density. So they use cesium chloride density gradient centrifugation method, and even before performing this, what they did, they took some bands, right? They isolated this heavy isotope of nitrogen, and then the heavy isotope of this heavy isotope of nitrogen containing DNA, they obtained the band there, right? In another one, they conducted the same for the light isotope. Let's suppose they obtained the band here. It's same like we go for the blood test, right? Doctors say now that the normal value of this particular vitamin or this particular iron is in this and this range. In your case, that value is getting higher or the lower. We have that standard values, standard calculations, standard values, and as per that, we just check whether it is low or the high. So same way, before conducting an experiment, we used to have that standard. So here, they obtained it. The heavy isotope of DNA is making band here. It is heavy, so at the base. It is lighter, so it is like this. So now what they did, like let's suppose this is the E. coli. So after 20 minutes, because this is the generation time of E. coli. This is the generation time of E. coli. So generation time means the time in which cell will divide into 2. This is the meaning of generation time, in which cell will divide into 2. So what they notice, like let's suppose, right, what they notice there, after 20 minutes, they obtained the bands, but band was at this position. It was a hybrid, right? Like it was not at this, which is let's suppose 14. It was not at this, which was 15. It was somewhere in between, right? In between. This is what they checked. Then afterwards, they conducted, like they allowed that E. coli to keep growing there. Then again, after another 20 minutes. So in totality, can I write 40 minutes? Like in your books here, 40 minutes is given. So it's not like that these two daughter cells, they will collect them after 40 minutes. No. Let's suppose you are starting here from 0, then after 20 minutes, then after another 20 minutes, right? After another 20 minutes, they obtain the, right? They again check this, right? They again check this. Now what they found? When they check the bands, again, the same thing. There was a band for hybrid also. There was a band for 14N also. But hybrid means the one which was in between was also there. So hybrid means this is showing 15 and 14N. So that's what they did. That's how they conducted the experiment. So they basically made it clear that after first generation, that is after first 20 minutes, when they isolated the cell, when they isolated their DNA, when they run this centrifugation experiment, the band that they obtained was not at this level, that is it was not showing 15N. It was not at this level, that is it was not showing 14N. So it was somewhere in between. So it indicates that that DNA was not as heavy as 15N, was not as lightweight as 14N. So it was somewhere in between. This is what they proved. Are you getting my point? Yes. Are you getting this? So they kept on performing such experiment, and that's how they concluded. So here you can see 15N DNA, red, after 20 minutes, right? So this is the gravitational force. So heavy DNA settled there. After 20 minutes, this is where they got the band. So green is showing light isotope, 15, and red is showing heavy isotope. So after another 20 minutes, after next 20 minutes, in totality 40 minutes, this is what they got in the generation 2. Two daughter DNAs were having the hybrid, and two daughters DNA, they were just having the 14. Why they are just having the 14? Because now the source of nitrogen is having that same isotope, right? And because here the parental cell was having 15N and then it got the source 14N, so that's why the hybrid was there. So that is how they obtained it. That is how they proved it. So Messelson and Stahel, they conducted the experiment in the case of a bacteria which we are calling as E. coli, right? The prokaryotic cell, the E. coli, and moreover, Tyler and his colleague, right, in the case of eukaryotic cell, they selected a plant cell, right, from the Vicia faba beans, and they performed the experiment with the radioactive thymidine. So this is all about the experiment. I'll tell you this and then we will pick up the important question, right? So see, E. coli was grown in a medium having 15 NH4Cl, right, as the only nitrogen source. This N is stable, but it is non-radioactive. It is heavier than the normal. So it is heavier than the normal, but not the radioactive. Sometimes some teachers also call 15N as radioactive nitrogen isotope. No, it is not radioactive, but heavy only, right? It is not radioactive, but heavy, okay? But heavy. Then DNA, proteins, and other nitrogen containing compounds, they all incorporated this 15N. So the newly synthesized DNA was heavier than the normal DNA because it was having 15N. So the method that they use is centrifugation and in a cesium chloride density gradient. That is again a PYQ. Right, centrifugation. Right, so it is what? It is centrifugation in a cesium chloride density gradient. Is that clear? In a cesium chloride density gradient. So DNA molecules with different densities, they settle at different positions. Is that clear? Is that clear? Done. So after growing in 15 NH4Cl, the E. coli were transferred to a medium containing normal NH4Cl, that is 14 NH4Cl. So then again cells were allowed to multiply. Samples were taken at specific time intervals. So what is it? 20 minutes. So from each sample DNA was extracted. Okay, so DNA samples were then again separated using what? Cesium chloride density gradient. So important it is. Is that clear? Important it is. So this allowed researchers to measure the density at each time point. So by observing the change in DNA density over time, they tracked the light nitrogen was being incorporated into new DNA strands. Right? So parental DNA strand was also there, new was also there. That's why it became hybrid. Right? It became hybrid. Is that clear? So after one generation, you know that E. coli divided once. So each new DNA molecule had one old and one new strand. Then the result was DNA had intermediate density, number one. So after two generations, that is after 40 minutes in totality, DNA was having 50% hybrid DNA because 4 daughter cells will form. So 2 daughter cells will have hybrid DNA and other 2 will have 14N only. So this experiment supports the semi-conservative model because each new DNA strand was having the parental one. So in 1958, year is important, very important. So Tyler and colleagues conducted experiment on Vicia faba beans. So that's a plant. So if it is a plant, it is a eukaryotic cell. And here they use the radioactive thymidine only. Okay. Okay. So this is the common mistake that students make. So let me ask you, who who provided the experimental proof for semi-conservative nature of DNA replication, who provided the experimental proof for semi-conservative nature of DNA replication in eukaryotic cell. So what should be the answer, students? Yes, please tell me what should be the answer here. Everybody quick. What should be the answer, guys? Quick. Exactly. Exactly my point, right? Done, students? So answer is Tyler and his colleagues. So if instead of eukaryotic cell, if I'll mention prokaryotic cell, then if I mention prokaryotic cell, then quick, then instead of eukaryotic cell, if I mention prokaryotic cell, then the answer will be Michelson and Stahl. So we are done here. So now let's start the detailed mechanism of DNA replication. So it is semi-conservative in nature. Okay. So DNA replication. Okay, that's what we are going to start. So what is the mode of DNA replication? We know that it is the semi-conservative mode. Which mode is there? It is the semi-conservative mode, right? It is the semi-conservative mode. So before going forward, right, I would like to ask one question as well. So if I ask you that after see here, on the basis of this, I'm asking you, after 4 generations, how many daughter cells will have hybrid DNA? It's a very simple question, and the answer is going to be simple as well. Tell me, after 4 generations, how many daughter cells will have hybrid DNA? Yes, please. Yes, please. 16, matlab 16 daughter DNAs will have hybrid. Are you kidding me, guys? I'm asking you for the same, right? For the same experiment. I'm asking you this thing for the same experiment. And in that same experiment, what you guys can see, right? What you people can see. You can see one thing very clearly, right? You can see one thing very, very, very clearly here, that here the source of nitrogen is 14N only, that contains 14N isotope only. So obviously in first generation only we got the hybrid. So even after 4 generation, even after 10 generation, you will just have 2 daughter DNAs which will have the hybrid, right? Which will have the hybrid DNA. What you people are talking? Those who are writing 16 or 10, guys, watch the lecture from the starting. Chalo. Now my next question. If after second generation, again, I'm shifting this to 15N. Again, I'm shifting it to 15N. Now my next question. The experiment is same. Now after the second generation, I'm again shifting these 4 cells into the, right? I'm just picking up these 4 cells. I am shifting it again into the 15N medium. Now you tell me after 20 minutes, or I would say after first, after the division, then how many cells will have hybrid? How many cells will have 15 or 14N DNA? Now tell me, right? The same experiment is there. I am saying that after 40 minutes, right? After 40 minutes, we have the second generation, 2 hybrid, 2 14. Now again, I'm shifting it to the 15N. Then what will be the scenario? It's simple. Like let's suppose 14N, 14N, and this is the hybrid. Now you have shifted it, right? Again, I told you the nitrogen source is 15. So obviously when this particular DNA will replicate, now that's going to be the scenario. You can draw it and you can make it. Let's suppose this color is showing 15N. So then so you can see 15N, both will be 15N here. This is going to be the scenario. Okay? This is going to be the scenario. So 1, 2, 3, 4, 5, 6, 7, 8. You will get 8 daughter cells because after this you have shifted it to 15N. So now when this DNA will duplicate, obviously green, 14N parental strand, complementary to it 15N. Here 14N complementary to it 15. It is already 15. So again 15. Right. So here you will see 1, 2, 3, 4, 5, 6. 6 are hybrid and 2 are having 15N isotope. So that's how you can solve these questions just by the common sense only. Okay, nothing else required, common sense. So now we have to discuss the DNA replication, and in detail in our syllabus we have the prokaryotic DNA replication. Now when you people talk about the prokaryotes, see this is the DNA. It is the circular DNA. So can I show it like this? Can I show it like this? Yes, obviously. This is the circular DNA. I told you already. So it's not like that DNA replication can start from any point. No, it will start from a particular location. So the point from where the replication start, we are calling it as a replicon. So the place on the DNA, or the point from where the replication starts. I'll discuss it in a very simple way. So I want you people to just pay attention. Okay. So point from where the replication will start is the replicon. Okay. Now students, in the case of, see, this is a prokaryote. In the case of prokaryote, so as I said in our syllabus, prokaryotic DNA replication is the important thing, and that too E. coli. So in their case, there is only one site from where replication starts. Do you know that? There is only one site from where the replication starts. Okay, so what do we call it? What do we call it? We call it as monoreplicon. We call it as monoreplicon, right? This is the, because there is one location, one point from where the replication will start. So that is monoreplicon. So the site from where it starts, we call it as origin of replication. So how many origin of replications do we have in prokaryotes, guys? How many origin of replications do we have in prokaryotes? How many origin of replication? There is only one, right? And in E. coli, you can call it as ORIC site. This is the name of the site. PYQ alert. Here also PYQ alert. Now, when you talk about the eukaryotes, right? You talk about what? Eukaryotes. The DNA there is linear, right? The DNA there is linear. So there are many replicants, or I would say many origin of replication, many origins of replication. So eukaryotes are what? They are multi or the polyreplicant. Am I clear? First point clear? First point clear? Yes. So circular DNA, only one origin of replication. ORIC site, E. coli, many origins of replication. This is the first thing. Now we know that our DNA is double helical in nature. Yes or no? Yes. Yes. Hannah, this is the point. Hannah, so now see, there you have many origins of replication. Here you have just one origin of replication. So let's suppose this is the circular DNA that we have. In the E. coli students, there is a bidirectional DNA replication. That is why that replication gets completed in that 20 minutes. Okay. If there will be single direction replication, like let's suppose first this DNA strand is getting copied, then the another is getting copied. If it is like that, right, then what's going to happen? It will take more time. It will take double time, right? Now I'm not entering in theta replication or not. So this is the case. So let's suppose this is the origin of replication. So that origin of replication is also recognized by protein. It's not like that all of a sudden the enzymes will get to know, okay, from here we have to start. No, there are proteins for that. But of course, because we have to focus on an exam, so here the discussion will be exam oriented only as per your syllabus only. So basically I want to tell you that if you talk about the site from where the replication can start, firstly, the enzymes, they need to recognize that site, and there are proteins or the enzymes that help in the same process. So let's suppose this is the origin of replication. So if I want to copy the DNA, I have to open it first. Are you getting my point? If I want to copy the information, if I want to see what information that DNA is containing, so I know that bases are inward, so I have to open the DNA, right? So for that opening or the unbinding of DNA, number one, right, what we need here, unbinding of DNA. So that unbinding of DNA, how will that be achieved? That will be achieved with the help of an enzyme which is known as helicase. That's the important thing here. I'll provide you the handwritten notes of mine today. First, let me explain you this topic, then you guys can see. These are the detailed handwritten notes, right? So as of now, I just want you guys to be familiar with this, familiar with this particular topic, and just pay attention, and then I'll provide you the detailed notes. So what is going to happen here? The unbinding of DNA occurs. Who's going to do that? The enzyme helicase, which is also referred as unwindase. Right? What do we call it? What do we call this enzyme, everybody? We call it as helicase, or we can also call it as unwindase. Is that clear? Is that clear? Sure. This is the helicase, and what else do we call it? We also call it as unwindase. Am I clear here? Anybody having any doubt? Sure. Sure? Done? So it is the helicase unbindase, or you can also call it as DNA B protein. Okay. So this particular enzyme, this enzyme also needs magnesium ion as cofactor, number 1, and it uses ATP hydrolysis. Right? It uses ATP hydrolysis to break hydrogen bonds. Now the point is, it can't do it in a single go or one go, as it takes a lot of energy, so results in the formation of a Y-shaped replication fork. Y-shaped replication fork. Done? So what is the name of the enzyme? Unbinding of DNA caused by helicase or unbindase, also known as DNA B protein. So at origin of replication, it will start the opening of the DNA helix. It cannot do it in one go. Why? Because it takes a lot of energy. So that is why it results in the formation of a Y-shaped replication fork. And as I said, it needs magnesium ion as a, as a cofactor. Is that clear? So let me show you this part. Let's suppose this is your Y-shaped replication fork. Can you see that? Like, wait. See, this is your DNA. This is your DNA. This is a double-stranded DNA. I'm just imagine it in this way. This strand is having polarity 3' to 5'. This strand is having polarity 5' to 3'. Right? Now, it's your helicase will move. Let's suppose this is your helicase. It will move like this. It will move like this. When it will move like this, now students, so slowly, it will break these bonds. Right? It will break these bonds, break these bonds, break these bonds. So when slowly it is breaking these bonds, then what is happening? Then this results in the formation of a, right? Then this results in the formation of a Y-shaped replication fork. So because helicase is moving like this, है ना? Helicase is moving like this. So you have to see the polarity also in this direction. This is the direction of helicase. This is the direction of replication fork. So it is slowly opening the DNA helix. So here, this template strand is having polarity 3' to 5', and this template strand is having polarity 5' to 3'. Am I clear? Am I clear? So this is happening. So now one more time. See, this is your this is your circular DNA. Your helicase is coming here. This is the origin of replication. From here, it is trying to open the DNA double helix. And as I said, right, the bidirectional mode is there. So because here you have the bidirectional mode, so here in prokaryotes, you see the replication bubble. There is a formation of a replication bubble here, right? Why there is a formation of a replication bubble? If you can visualize, I'll try to give you the analogy. Like, let's suppose this point is, right? This point is the origin of replication, right? A protein is there which will bring this helicase to this point, that it is the origin of replication. You start breaking the hydrogen bond from this point. So it will use the energy and all, right? Now, it cannot open this DNA in one go like this. No, it cannot. It will go slow, and there is not one helicases. No, it has to open it in both the directions also. More than one is there. So let's suppose this helicase, it is moving like this, moving like this, slowly, because it cannot open it in one go. So now can you see a structure like this will form? A structure like this will form. So this is how helicase is moving. That is how slowly DNA is getting open. So this type of structure is there. So it's like a bubble. It is like a bubble. So where one strand will also be copied, another strand will also be copied. So it is a bidirectional mode of DNA replication. Got it? Got it? Done? So now, when you look at this replication bubble, look at this part. Isn't it looks like this? This part. I'm just giving you some examples so that you can understand. Look at this part. I'm showing you this separately. है ना? See this. Isn't it? So this is the replication bubble. So this structure is also looking like what? Y shape. This structure is also looking like a Y shape. So in replication bubbles, you will see two Y-shaped replication forks. What will you see here? What will you see here? You'll see two. Yes? Done, बच्चे? Done, students? So this is the one replication bubble you can see. Can you see that? Two replication forks, right? This side, this side. So bidirectional mode will be there, number one. Now, let's pick up this, right? Now, let's pick up this part, okay? Let's pick up this particular part. So, as I said, helicase is moving like this. That is how it is. That is how it is simply opening the DNA helix. So because helicase is moving like this, so you have to see the polarity in this direction. So this is 3' to 5'. This one is 5' to 3'. This one is 3' to 5'. This one is 5' to 3'. So this template strand is having polarity 3' to 5'. This template strand is having polarity 5' to 3'. So that's what it is, right? That is what it is. Is that clear? Is that clear? So students, what they do, they just think, right, that this they see this it in this way, 5' to 3', 3' to 5'. No, your helicase is moving like this. So that's how you are going to see the template's polarity. 3', 5', 5', 3'. Understood? Now, when you try to open a DNA double helix, yes? Yes, when you talk about the DNA double helix, see this. See this. You are trying to open it, understand these things. There comes a time when you will feel a kind of tension. See, even you can try it at home. There comes a time, can you see? Here, see, coilings are, right? Coilings are forming. Coilings are forming. So it is obvious. You are trying to open a DNA double helix. At points, coiling will also be there. Plus, both the strands are complementary to each other. Like, let's suppose if here you have A, both are complementary to each other. So when you're trying to open it now, they can re-anneal again, they can rejoin again. So we don't want that. We don't want them to rejoin again. So here comes the role of another proteins, right? Here comes the role of another proteins, that is SSBP, single-stranded binding proteins. Single-stranded binding proteins. In short, you can call them as SBPs, and they'll prevent re-annealing of separated DNA strands. What's their job? They prevent the re-annealing of separated DNA strands without even affecting DNA synthesis, new strand synthesis, या new strand DNA, new DNA strand synthesis, or DNA replication, whatever you want to write. So yeah, they are binded here, but they are not covering any base. Okay, they are binded here, but they are not covering any base. Can you see that? Done? Sure? Are you sure? So it will not cover. They are attached to these separated single strands. They'll prevent the re-annealing, and they are not covering the bases. Like, they are attached in such a way that they cannot affect the DNA replication. Now, as I told you, that supercoiling can also be there. So just to prevent that supercoiling, here comes the role of third enzyme here, topoisomerase. Okay? It removes supercoiling. It removes supercoiling. Are you getting my point? So now in this topoisomerase, what you people will see, there is class 1 and there is class 2 topoisomerase. Class 1, class 2. Class 1 make a cut in single strand of DNA, right? Wherever, like, sometimes it happen, sometimes even you have the wool, if it get, tangled, so you can make a cut in one part there, you can just rotate it and can remove that. Same is the case here. Class 1 topoisomerase, they make a cut in single strand only, right? It can, class 2 topoisomerase can make a cut in both the strands, right? Both the strands of DNA. They can make a cut there, they can rotate that DNA, and they can just remove the supercoiling. So in class 2, what you have to remember, that in prokaryotes, the first topoisomerase discovered was DNA gyrase, right? So it's a class 2 topoisomerase. It's a class 2 topoisomerase. Are you getting my point? So topoisomerase removes supercoiling. DNA gyrase, it is a topoisomerase only, class 2 topoisomerase can make cut in both the strands of DNA. It was the first discovered there, and it also use the energy from the ATP hydrolysis. Okay, so it helps in removing the negative supercoiling, but yeah, it can introduce some supercoilings as well, right? It can also introduce the supercoilings. Are you getting my point? It can also introduce the positive supercoilings and can remove the negative one. So that's what you have to remember. So topoisomerase, two classes are there, class 1, class 2. So it can make a cut in single strand only, make a cut in both the strands, right? So in prokaryotes, DNA gyrase is known as the topoisomerase. So it is the class 2 topoisomerase which uses the energy from ATP hydrolysis. Done? Right? So single-stranded binding proteins, topoisomerase, is that clear? So now we have to synthesize the DNA, okay? We have to synthesize the DNA here, but for that, we will be discussing about certain enzymes. And that enzymes are DNA polymerase, also DNA polymerase. And here we have what? We have DNA polymerase 1. Listen to me, right? It's also known as DNA dependent DNA polymerase. You can also call it as DNA dependent DNA polymerase. It is using DNA strand and making a new DNA strand. That's it, right? DNA polymerase 1, DNA polymerase 2, DNA polymerase 3. So these are the DNA polymerases, right? And they are in prokaryotes. So what are the names here, guys? What are the names here? So it is DNA polymerase, DNA dependent polymerase. So it is the DNA polymerase 1, DNA polymerase 2 and DNA polymerase 3. Is that clear? Is that clear? Yes, please. So it is in the case of prokaryotes. It is in the case of prokaryotes. Is that clear? Is that clear? Sure? Sure? So in eukaryotes, but in eukaryotes you do not have the same, right? In eukaryotes you do not have the same enzymes. You will have DNA polymerase alpha, beta, delta, epsilon, etc. So this DNA polymerase alpha, beta, delta, right, and the epsilon. So this delta, this epsilon, right, because there are two different strands which will get synthesized, right, students? There are two different strands which will get synthesized. So you will see, right, polymerase, this DNA polymerase delta, when you will see, guys, so this is mainly for one, like I would say for lagging strand synthesis. I'll explain what these strands are. For leading strand synthesis, this one will help in repair. So they are they are of different types and they have different role. So in eukaryotes, you do not have DNA polymerase 1, 2 and 3. It is in prokaryotes. It is in prokaryotes. Now, when I talk about the DNA polymerase 1, it is discovered by Kornberg, scientist Kornberg. So it is also known as Kornberg enzyme. Okay. But out of these three, it is the DNA polymerase 3, which is the main replicating enzyme. It is the main replicating enzyme. Okay, this is the main one. So it is also known as replicase. This is the main enzyme. Okay, so whenever it comes to the synthesis, right, be it the synthesis of DNA or it is the synthesis of RNA, synthesis will always be in, right, it will always be in 5' to 3' direction. Just keep it in your mind. Synthesis will always be in 5' to 3' direction. Is that clear? Always. Why? I told you before, let's suppose this is your first nucleotide, nitrogenous sugar, nitrogenous base, phosphate, sugar, nitrogenous base, phosphate. That's how it is. Sugar, nitrogenous base, phosphate. That's how it is. These are the nucleotides. So first nucleotide will join with the next by 3' phosphodiester bond, 3' phosphodiester bond. So here you know that 5' end is having free phosphate and 3' end is having free hydroxyl. So always remember this thing, that whether it is the synthesis of RNA or DNA, it will always be in 5' to 3' direction, right? It will always be in 5' to 3' direction, no matter what, whether you are talking about the synthesis of DNA or you are talking about the synthesis of RNA, it will always be in 5' to 3' direction. Done? So, DNA polymerase 1, 2 and 3. Now, this enzyme is having 5' to 3' polymerase activity I'll explain everything. You just have patience. Do take the notes of this part. 5 dash to 3 dash exonuclease activity. 3 dash to 5 dash exonuclease activity. Am I clear, guys? See, look at the activities here. Firstly, I'll teach you everything. I'll explain everything. Look at the activities here and look at the main point. This activity, right? This one, just keep an eye on it. This is unique to DNA polymerase 1. Okay? This is something which is unique to DNA polymerase 1. Now, what have I used? Synthesis, I told you. See, I am saying synthesis is always in 5' to 3' direction. And I told you that all the three enzymes have 5' to 3' polymerase activity. So, what does it mean? What does it mean? What does it mean? Yes, what does it mean? It's simple. 5' to 3' means this will add the nucleotides in this particular direction. You know that first nucleotide is having free phosphate group. Next nucleotide, its phosphate will be involved, right? Right. Its phosphate will be involved. Done? Done? Done, beta? So what am I trying to tell you? All I'm saying is that 5' to 3' polymerase activity means first nucleotide, then next. So that synthesis is in 5' to 3' direction. Are you getting my point? That synthesis, it is in 5' to 3' direction. That is what I'm telling you. Okay. So obviously all the enzymes are having 5' to 3'. All the enzymes are having 5' to 3' polymerase activity, means they can add bases in this particular direction. Okay. Now, the next point that I have to discuss is 3' to 5' exonuclease activity. Next is what? 3' to 5' exonuclease activity. Which one it is? 3' to 5' exonuclease activity. Done? Once again. Okay. So what does it mean? See, exo means ends. Nuclease, that digests nucleic acid. So this is an enzyme, right, which will break the DNA from its ends, like ends. This is an enzyme that will remove the nucleotides from the ends. Now my point is, what is mentioned there? 3' to 5' exonuclease activity. What is mentioned there, guys? 3' to 5' exonuclease activity. Am I right? What is mentioned there? 3' to 5' exonuclease activity. Okay? 3' to 5'. So it means, let's suppose you have synthesized a strand like this in 5' to 3' direction. You have synthesized the strand. Now, it happens now, like let's suppose sometimes we arrange the things. 1, 2, 3, 4, 5, 6, 7, 8. Then in reverse order we check it. 8, 7, 6, 5, 4, 3, 2, 1. Something like that. That's how you can keep it in your mind. Like 1, 2, 3, 4, 5, 6, 7, 8. Then 8, 7, 6, 5. In reverse order we check it. So the synthesis is in 5' to 3' direction. Right? And in the reverse order, 3' to 5'. Enzyme is having 3' to 5' exonuclease activity. right? 3 dash to 5 dash. Exonuclease activity means this enzyme will move from this 3 dash end towards the 5 dash end, and it will remove the bases. Like if there is anything which is incorrectly attached or something, it is going to remove that. Are you getting my point? Again, I'm saying the DNA repair mechanism, the proofreading mechanism is not given in your syllabus in detail. So we'll stick to the topic here, because here, this chapter, this topic, very interesting, very lengthy. So we have to stick to our syllabus part. So all I'm saying is that DNA, you know that with time, from RNA, DNA evolved, and that DNA also evolved the mechanism of repair, right? We cannot allow the mutation at a very fast rate in DNA. So in DNA, there is a mechanism, of proofreading. That DNA will check whether the bases are added correctly or not, number one. Number two, if bases are added incorrectly, DNA repair is also there. So that's what I'm trying to tell you. So all I'm saying here is that these enzymes will move in 3 dash to 5 dash direction. Are you getting my point? 3 dash to 5 dash direction. It will remove the bases. Okay. It will remove if there is anything incorrectly attached. Now, this activity, 5 dash to 3 dash exonuclease. So what does it mean? It means, yes. What does it mean? It simply means that this enzyme will move in 5 dash to 3 dash direction and will remove the bases. So this activity was not common to all. It was a unique activity, right? And where we noticed that? We noticed it in DNA polymerase 1. Where we noticed it? In DNA polymerase 1, which you also call as pol 1, right? Polymerase 1. So this will help in primer removal, right? I'll tell you. This activity we'll discuss soon. Okay, put a star over it. We'll discuss it soon. As of now, just keep it in your mind that we are talking about the DNA polymerases. In prokaryotes, we have 3 types. Main replicating enzyme is DNA polymerase 3. So it's not like that all the 3 cannot synthesize the DNA. Yes, they can. They can synthesize the DNA in 5' to 3' direction, right? They can synthesize the DNA in 5' to 3' direction. Are you getting my point? They can synthesize the DNA strand in 5' to 3' direction. All 3 can do that. But the point is here, the enzyme has to be fast and accurate. So it has to be fast as well as accurate. And that thing you will see in the DNA polymerase 3. As per NCERT, I'm telling you, this enzyme will add 2000 base pairs per second. Are you getting my point? This enzyme is going to add what? 2000 base pairs per second. So it's not like that, right, students? Yes, please. It's not like that all 3 cannot synthesize the DNA or cannot add the bases. They can. But the main replicating enzyme is this. So its reason is that we don't want, just in final paper, in the NEET examination, it is not like that you have to answer the questions fastly. No. Like speed is important, but it should be speed as well as accuracy. Okay. It should be speed as well as accuracy. Speed as well as accuracy. So here in this case, DNA polymerase 3 is fast and accurate. Its processability is high, right? I just said 2000 base pairs per second it can add, right? Yes. And it is having proofreading ability also by this 3-5' exonuclease activity. Is that clear? It can remove the mismatched base pairs also, right? This is because you have to synthesize 2 strands. One is leading, one is lagging. So it can do that also. Is that clear? Is that clear? So polymerase 1 is slow, its processability is slow, and this enzyme is mainly known for this unique activity. I'll discuss it with you all, right? So this enzyme cannot, handle the long stretches of DNA. It cannot add that bases in the long stretches. Okay, right? Right? Is that clear? Sure. And DNA polymerase 2 mainly helps in the DNA repair. Remember that. Okay? The DNA repair is its main function. Okay? 1 second. Okay, done. So DNA polymerase 1 cannot handle the long stretches of DNA, and as I said, polymerase 2 is mainly considered for the repair. It's not like that ki it cannot add the bases. Its fidelity is high, means it can remove the mismatched pair at a better rate, right? So it mainly plays role in the DNA repair. But yeah, if let's suppose if there is any halt in the polymerase 3 activity, if it is not working fine, then polymerase 2 can act like a backup. Are you getting my point? Then this polymerase 2 can act like a backup. So yes, you can also write them in this way: polymerase 1, polymerase 2, and polymerase 3. That is also a shortcut to write these enzymes. Okay, so this is mainly for the repair, right? This is mainly for the primer removal. I told you already, but I'll tell you what these primers and how that gap filling will be done. So if there is any stalling in the replication fork, this term you might read somewhere: stall in replication fork. It means replication fork has temporarily, a replication has temporarily stopped. Why is it so? Obviously multiple reasons can be there. Maybe there can be DNA damage due to UV radiation, right? Thymine dimers can form. So there are multiple things. Okay, so now, as I said, main replicating enzyme is DNA polymerase 3. That is what I just said. That main replicating enzyme is DNA polymerase 3. Now pay attention. You know what? Just say polymerase 3 is the main replicating enzyme, but do you know it is also having some issues? Let me tell you. So DNA polymerase 3, as I said, it is the main replicating enzyme, but it cannot initiate DNA replication by its own. That's a very important point, guys. It cannot initiate DNA replication by its own. You can write down. It needs free 3-OH group. Are you getting my point? No, you are not getting it. So let me tell you. First of all, see this. This template strand is having polarity 3' to 5', 5' to 3'. Let me write it. Then I'll explain it. Yes. So what am I trying to tell you? I'm telling you a very simple thing here. Right, I am telling you a very simple thing here, that DNA polymerase 3 cannot initiate replication by its own. It is not somebody that will just, pick up the bases and will start adding. It needs the free 3-OH group. I'll give you the example. There are some people, they think, right, they believe that, oh, somebody is doing wrong, we should raise the voice, right, or we should stop that person, but they are not able to. They want to, but they are not able to. They lack that leadership qualities. They cannot start it by themselves. Now, let's suppose I started it. Now you are also having the same emotion, right? I started talking about somebody that person is not doing right. This is inappropriate and this should be stopped. Now, because you were also thinking the same, it's just that you were not able to say that, right? Now I have initiated, you have joined me. Some other people, they have joined me. So I started something, you added to that. Yeah, we also feel the same. It is the case with the DNA polymerase 3. I'll give you another example. Just say, let's suppose, might be you are not able to, like some students, they are not able to crack the NEET examination, right? But if I'll send them to the medical college, they can become the amazing doctors because they are hardworking, they know how to study. But at the first place, they were not able to qualify the exam. Same is the case with the DNA polymerase 3. DNA polymerase 3 is like, I know I have to synthesize a strand, and that I can do in both the directions. Like, I know I have to synthesize a strand anti-parallelly. I can do that. But the point is, I cannot start it. You just give me this point here. Let's suppose ATCG. I'm randomly picking up the sequence, nothing special here. So it's not like that polymerase will be like, okay, adenine is here, I'll bring thymine. No, it needs the free 3-OH group. So to solve this issue, solve this problem, right now there is the requirement of one more enzyme which is known as primase. Okay, there is the requirement of one more enzyme which is known as what? Which is known as primase, right? Which is known as what? Which is known as primase. Now this primase is also known as, it is also called as DNA dependent RNA polymerase. Do not forget its second name. It's DNA dependent. Why DNA dependent? Because it will use DNA as a template, but it is DNA dependent RNA polymerase. DNA dependent RNA polymerase it is. Now this enzyme is going to help. This enzyme will synthesize a short stretch of 10 to 15, or at some references 15 to 20 nucleotides, and actually these are ribonucleotides. Okay, these are ribonucleotides, so we call them as primer. This is the RNA primer. Are you getting my point? So because DNA polymerase 3 cannot initiate the strand synthesis by its own, so the primase will come there. Primase is the DNA dependent RNA polymerase. It uses the DNA as template, and complementary to it, it starts synthesizing, right? Complementary to it, it starts synthesizing what? Complementary to it, it starts synthesizing the nucleotides, right? And it doesn't need free 3-OH group for synthesis, right? It doesn't have any such requirement. It can just simply see the DNA and it can start adding it can add the bases. So 10 to 15 or 15 to 20 ribonucleotides are there that will be added complementary to DNA. So now just imagine, right? Now just imagine, this is the 3' to 5' strand. So primase will come, right? Who will come? Primase will come. It will form a short stretch of RNA here. Primase will come. It will form a short stretch of RNA here. So this is having the polarity 5' to 3'. Now 3-OH group is free. Yes. Now the DNA polymerase 3 will come. Now the DNA polymerase 3 will come, and students, then it will start adding the bases there. Are you getting my point? That's the role of DNA polymerase 3. Right? That it cannot start the DNA replication by its own, but if you will show it the free 3-OH group, it can just pick up and it can start adding the bases. Now, remember, this is the direction of replication fork, because helicase is moving like this. Your polymerase is also moving like this. Here it is synthesizing this 5' to 3' DNA in this direction. Helicase is also moving like this. Polymerase is also moving like this, adding the bases like this. But now there is a problem. What is the problem? Problem is this template strand is having polarity 5' to 3'. It means if I have to synthesize the strand, that will go in this way. Means this side, the DNA strand is going in this direction. But here it has to be in this direction. है ना? So polymerase, which is, and this DNA polymerase 3 is multimeric unit having many subunit. It is moving like this, right? Like this. But it has to do a task in the opposite direction. Like let's suppose I have chocolates in my hand. Here some students are sitting, right? Now if I have to distribute like this, 1, 2, 1, 2, 1, 2, 1, 2, I can do it easily. But now my task is, one student is sitting here, another is there. I have to give them the chocolate like this. One here, another there. One here, another there. And there is a long queue. So you imagine, how is it possible for the enzyme? So the strand synthesis has to be simultaneously, plus one strand this DNA polymerase 3 has to synthesize like this and another strand like this. Means one strand in this direction and another strand is in opposite direction. Don't you think it's a task? It's a task. Of course it is. Definitely it is. So this DNA polymerase 3, what will it do? It will, obviously it needs the primer. So primase will come, primase will form the primer here. Then for the another strand where the synthesis has to be in opposite direction, it will form the primer. But this primer, but this connection will break because enzyme has to move in opposite direction. That is the problem. So next what? Again and again for the synthesis of this particular strand, so yeah, so students, for the synthesis of this particular strand, again and again connection will break. So let me simplify it. Here the template strand is 3' to 5'. So complementary to it, right? Complementary to it, new strand synthesize continuously, new strand synthesize continuously and called leading strand, and called leading strand. Are you getting my point? And called leading strand. So this strand is synthesizing in the direction of replication fork. This strand is getting synthesized in the direction of replication fork. Done? Now, here I'll use a different color so that we can categorize. Here, template strand is 5' to 3'. Here, complementary to it, strand synthesize in opposite direction, of the direction of movement of DNA polymerase, or I would say, one second, I'll tell you in simple words. So 5' to 3' complementary strand synthesize discontinuously. Here strand is synthesized discontinuously and called as lagging strand. What do we call it? This one is leading and this one here is what, students? This one here is known as lagging strand. This one here is known as lagging strand, right? It is synthesizing away from the direction of replication fork. So its synthesis is not in the direction of replication fork, but away from the direction of replication fork. So again and again, connection like DNA polymerase through 3 and its connection breaks. Then again, primase came, form primer. So here strand is in fragments. Okay. So these fragments are discovered by Okazaki students. So lagging strand, these fragments are known as Okazaki fragments. Am I clear? So these fragments are known as what? They are known as Okazaki fragments. So please write down. Quick. Please write down. Quick. Do it quickly. Is it done? Guys, quick. Noted. Quick, quick, quick. Sure. So that is how it is. So in the paper, questions can come like this. Just allow me a minute. Right? Just allow me a minute. So let me show you this part. So let's suppose this is the template strand 3 dash to 5 dash. So template strand is 3 dash to 5 dash. So here complementary to it, which strand will synthesize? Lagging strand. Here template strand polarity is 5 dash to 3 dash, complementary to it, which, oh sorry, my bad, here leading strand 3 dash to 5 dash, leading strand. Here it is going to be lagging strand. So can I not say that the strand synthesis is, we can say not that DNA replication here is, see, DNA replication here is firstly semi-conservative. Indeed, it is semi-conservative in nature. It is semi-discontinuous, discontinuous or semi-continuous, whatever you want to call it, because one strand is synthesized continuously and another discontinuously. So here you can see 1, 2, 3. Let's suppose, so see this. So these are the Okazaki fragments here, right? Okazaki fragments here, right? So here see primer 5 dash to 3 dash overall. This is the direction of replication fork. So leading strand synthesize towards replication fork. Already I have mentioned it, again revising it. Lagging strand, its synthesis is away from replication fork. I hope it's clear, right? They all are PYQ. This is a PYQ. Template strand polarity is a PYQ, right? So which, this is also a PYQ. And here these fragments are Okazaki fragments. Now look at this. Here you have one primer. Here also you have many primers, because again and again correction will break and again they will form. Now don't you think, just say, let's suppose, let's take the example in this way. Don't you think, let's suppose if I just open it and then show you this. Do you think it will work? Do you think it will work? See, this is one fragment, the continuous synthesis, leading. Template strand now you know. Lagging. Look at this, 5 dash to 3 dash. This is another fragment. So see, 5 dash to 3 dash. 5 dash to 3 dash, 5 dash to 3 dash. So don't you think that if we will have DNA where in between the RNA primers are there, don't you think it's little odd? Yes? Okay, just 1 minute. Guys, what kind of debate is going on there? What kind of debate is going on there? And who's saying that I'm not teaching anything from the scratch? Do you even know what is the meaning of scratch? Do you even know the actual concept of this topic? Do you even know what you actually need to understand this topic? Right? Mr. Art Magic, do you even realize that I have divided this topic in multiple sections, and one by one I'm elaborating each and everything in detail, and nobody will explain you this topic in that detail, and not at least in this way that it's a marathon, right? I'm supposed to finish this lecture in 12 hours, but still I'm going slow where the topics are lengthy so that you guys can understand. And this is the kind of debate you guys are, this is what you're doing here in the chat section. So I don't know why do we have the spammers here every time. So see guys, I just have this request from the complete Unacademy NEET English team that why do you people type here in the chat, oh my god, this teacher is better, that platform is better. Then what you do here in our lectures? Why do you come here even? Right? And we have no need to prove anybody that how good we are, how good our content is, because this is for the third time we are starting Game of NEET 3.0, and it is as per the student demand, right? So we all can understand one thing from this part, that students, they need such content, okay? And I think these are the only detailed one shots that are available on the YouTube, that too for free of cost, okay? Where teacher is teaching for 10 hours, 12 hours straight, and whatever topics they are completing in that particular session, they are starting everything from the scratch. They are not just starting a topic with all that, josh and the high energy and everything in detail, and then in the middle they skip. We are teaching each and everything in detail, right? So just pick up your NCERT, start reading each and every point, and then realize that it is not just the NCERT, you are getting way beyond NCERT. So I don't need such stupid students to judge my teaching, but I don't want any kind of debate here in the chat section. If you are able to understand, spend your time, otherwise skip the class, right? Leave the class, okay? सैम भट्ट, मैम वसीम सर की शादी कब है? बेटा मैं वसीम सर की पंडित नहीं हूं ना जो उनकी शादी का मुहूर्त निकालूंगी कि इस दिन है वसीम सर की शादी. हाँ चलिए, so this is what you have to see students. A leading strand, lagging strand. Now see, don't you think we should remove these primers? Yes. Don't you think we should remove these primers? Of course we should. Right? Of course we should. So for now you'll understand the role for primer removal. For primer removal, DNA polymerase 1 is required. Now we will use that unique activity of DNA polymerase 1, right? Done? Done? So for primer removal, polymerase 1 is required, and which activity? It's 5' to 3' exonuclease activity, because here you guys can see that, see, primer 5' to 3', 5' to 3'. So that DNA polymerase 1 will come. That DNA polymerase 1 will remove this primer, right? That DNA polymerase 1 will remove the primer from this part, right? So that is the unique activity of the DNA polymerase 1, and then the gaps are filled. Are you getting my point? And then the gaps are filled by 5' to 3' polymerase activity. So by using that 5' to 3' polymerase activity, these gaps are filled. DNA bases will be added here, guys. DNA bases will be added here. And do you know that here at these points, DNA ligase, which is the molecular glue, will form phosphodiester bond. That molecular glue will form phosphodiester bond. Yes or no? Yes or no? Right? That will form phosphodiester bond, And what's going to happen next? Then the sugar phosphate backbone will also join. Okay. So that's the complete detailed mechanism of DNA replication. Okay, so that's what you have to remember. Look at the diagram here. So here you can continuous synthesis means leading strand. Discontinuous synthesis means lagging strands having fragments, Okazaki fragments. Okay, so it is getting synthesized away from the replication fork. So this stretch will strand synthesize first, then this, and then this. Okay, that's how it is going to work. Isn't it? Isn't it? Done? Okay, so that's what you have to remember, and just have a look of this particular diagram. So see, origin of replication, you can see the replication bubble here. One strand is getting synthesized in this direction, another is in this. So here you know that bidirectional mode is there. Bidirectional mode. So replication fork is having two replication bubbles, right? Replication fork is having one replication bubble is having two replication forks. You guys can see that. So one is look at the helicase. So it is moving like this. Single stranded binding proteins are attached. So this is the leading strand synthesizing continuously. Now look at the lagging strand here. So here, see, one fragment, another. So DNA polymerase 1, it removes the primer, it fills the gap, and then ligase will come to join the sugar phosphate backbone. Any doubt? Any doubt? So one more diagram is there, and not just the diagram. Here you can see this table as well, where enzymes are also mentioned. DNA clamp, there is no need to know that. It's just a protein which prevents elongating DNA polymerase from dissociating from the DNA parent strand. Not so important. Yes, please. Not so important, right? But this is what you have to understand. So this is all about the enzymes, right? The diagram. And now from my handwritten notes, we will revise all the points. So whatever I have mentioned on the board, they are important. You can just simply write it. Okay, so just have a look here. So this is the 5' to 3' exonuclease activity, which is the unique one. So this 3' to 5' activity, it is basically for the proofreading. If there is any base which is attached incorrectly, or if there is any mismatched, mismatch, right? If there is any mismatched base, so that will be removed. Is that clear? Sure? Hanji, any doubts? Done? Sure? Now you know. Yes. Now you know this leading and lagging strand, its role. I wish you should. I hope you know this. Chaliye, so let's have a quick revision. Ready for the revision? Ready for the revision? So let's wrap it up in 5 minutes, and then we'll start the gene, the enzyme, and then we will start transcription. Okay, right? So before our next break, we have to finish the genetic code as well. Okay, then after our next break, we will revise translation, types of RNAs, translation, operance, human genome project. Okay, so in 3 parts we are going to finish it. So let's start with this replicants. So when we use the word replicants, already told you, the replication will start from a particular site or the location or the discrete unit known as a replicant. Bacteria, one replicant, so they are known as monoreplicants. Eukaryotes, multi-replicants. or polyreplicant. So in eukaryotes, when will you see the DNA replication will start? It will start in S phase. Important. So it is the phase where the DNA content will get doubled, but the number of chromosome will remain same. Understood? The DNA content will get doubled, but the number of chromosome will remain same. Now, helicases, it is the ATP-dependent unbinding enzyme, students, which will separate the parent strands, which will hydrolyze the hydrogen bonds. But remember one thing, it cannot do it in one go. So it will move slowly and form Y-shaped replication fork. So it hydrolyzes the ATP. Then helix destabilizing proteins, single-stranded binding proteins, which will prevent re-annealing. Annealing means joining. So the separated single strands, right, cannot join again. Okay, so it will bind to these single-stranded binding proteins. They will bind to the DNA strands without covering the bases, right? Without covering the bases. Therefore, they remain available, right, for the replication process. Is that clear? Then topoisomerase. So in prokaryotes, that is basically the DNA gyrase, and it is the class 2 topoisomerase. So helicase, it introduces a positive supercoil. So topoisomerase, it relaxes that. So there is topoisomerase 1, which cause a break in the single strand. Just let's suppose this is the DNA strand, so it can cut only the single strand. Okay, so topoisomerase can cause double strand break in helix. Done, students? Done, students? So DNA gyrase is a type of topoisomerase which is found specifically in bacteria, and this need the ATP hydrolysis. Then DNA primase, you know that it cannot, no DNA polymerase can initiate the replication by its own. So primase will form a short primer, right, which will provide free 3-OH group to the DNA polymerase 3. Is that clear? Right, so it is the short RNA oligonucleotide segment or RNA primer which will be synthesized by primase, right? So it will help in, right? It will help the DNA polymerase 3 to get the free 3-OH group, and it will be able to initiate the process then. So RNA primer is synthesized by copying a particular base sequence from one DNA strand. Now, primers about 10 nucleotide long, right? So later on, these primers are removed. And now you know that who's going to remove that? DNA polymerase 1 with its 5' to 3' exonuclease activity. How will it remove? With its 5' to 3' exonuclease activity. So in bacteria, there are two different enzymes which, synthesize the RNA primer. Okay, one is RNA polymerase that will act on the leading strand. One is DNA primase actually. Right, another is DNA primase actually that will be on lagging strand. Now let's go ahead. So you know that 3 types of polymerases are there, and the main replicating enzyme is DNA polymerase 3. Then students, polymerase 1, because it is isolated by Arthur Kornberg. So I told you it is also known as Kornberg enzyme. So DNA polymerase 1 can play the role in the DNA proofreading, repair, or the editing, but its main function is primer removal, main function. Right, and main function of repair is done by your DNA polymerase 3. So this activity is unique. That helps in the removal of primer and later on filling the gaps with the polymerase activity. So DNA polymerase 2, DNA repair enzyme, right? And obviously if DNA polymerase 3, if it stops, so then it is used, it continues the DNA replication process. Okay, so ligase, you know that it will catalyze or forms phosphodiester bond between 3-OH group and the free 5-OH group. So it is also known as molecular glue. So initially, I have just mentioned about the enzymes. I hope their function is clear. So now let's see the mechanism here. So you can see the process of separation of 2 strands of DNA is unzipping, unbinding. It starts at a certain point, which is origin of replication. Okay, next. You know that prokaryotes have one, eukaryotes have multiple origin of replication. So helicase, which is also known as DNA B protein, it is responsible for this unzipping and breaking of the hydrogen bonds. Now, helicase, it is unzipper or molecular motor or mobile promoter. That is the name for it. It needs magnesium ion, already told you. It needs the hydrolysis of ATP as well. So it cannot open the DNA in one go. So it will slowly open results in the formation of replication fork. And because in prokaryotes, the bidirectional application is there, so you'll see the replication bubble having 2 replication forks. Then unbinding of DNA duplex and replication from ORI proceeds in both the direction, so it is bidirectional. So this is continuous, this is discontinuous synthesis. So here you can see unbinding of template DNA at a replication fork could be catalyzed by 2 DNA helicases acting in concert, like they are working together, one running along leading strand, another lagging strand. Told you already. Now SSBP, you know that they'll prevent the rebinding, right? Then topoisomerase, you know that helicase can even introduce positive supercoil, which will be removed by that. It relaxes the supercoil, topoisomerase. Then how the new chain will form. Yes, one point that I have missed there, right? Again, that is from the NCERT. Let me revise it here. So now you know that. See this. This is 3 dash, 5 dash. This is 5 dash, 3 dash. Let's suppose here RNA primase will form the primer, and then you know that new strand will form. But what will be added here? Here in surrounding DATPs, that is deoxyadenosine triphosphate, deoxycytosine, cytidine triphosphate, thymidine triphosphate, or guanosine triphosphates are there in the surrounding. So they serve dual purpose. Do you know that? They serve dual purpose. Yes, and already I told you the DNA polymerase 3, the main enzyme, it needs to add the bases 2000, it adds 2000 base pairs per second. I hope you are clear with it. Now, if you look at this, I am saying that they serve the dual purpose. What does it mean? Can you see that they all are triphosphates? Yes, and here I need to add the monophosphates. So what's going to happen? So they will, these triphosphates, they'll act as substrate and also provide energy for polymerization. They act as substrate and also provide energy for polymerization. Are you getting my point? Yes, they act as substrate and also provide energy for polymerization. What does it mean? Obviously, let's suppose here you have A. Complementary to it, you have to add T. Now you have DTTP. Now what will you do with this? Obviously, you want to add it there, right? But it is a triphosphate. So you will use it as substrate and its 2 extra phosphate groups will be removed. So when they will be removed, 2 extra phosphates, right? 2 extra phosphates, they provide energy for polymerization. What will they do? They provide energy for polymerization. Are you getting my point? So it's self-acting as a substrate because it will be added to the chain here, right, to the newly synthesized strand here. But the other important point is 2 extra phosphates will be removed, right? Done. So that will provide the energy for the polymerization. So this is the dual purpose. So here even I have mentioned it, that for the formation of new chain, nucleotides are obtained. So nucleotides are present in the form of triphosphates like deoxyadenosine triphosphate, guanosine, cytidine, thymidine. So they will act like, and see when you write them, it is nucleoside triphosphate. So basically, remember, it's nucleotide or nucleoside monophosphate, but here it is nucleoside. If here you'll write nucleotide triphosphate, then it will be incorrect. So it is nucleoside triphosphate. Am I clear? Am I clear? Sure? Sure. So it is the nucleoside triphosphate. Am I clear? It is the nucleoside triphosphate. Okay. So they are acting like a substrate plus provide energy for the polymerization. Then during replication, 2 extra phosphate groups of all nucleotides, they are separated. Energy released is consumed in DNA replication. Okay, students. So DNA polymerase 3, they cannot initiate DNA strand formation as it needs free 3-OH. Right? Right. So for initiation, DNA dependent DNA polymerase, that is primase, is required. So for primase, you also use the word DNA G. That's what you can write. So primase is also known as DNA G. Is that clear? Is that clear? So this enzyme produces a small strand of RNA at 5' end. So that's what we have to remember. So you know that complementary, the bases will be added. So the template strand having polarity 3' to 5', complementary to it, leading strand will be formed continuously and it will formed in the direction of the replication fork. And here you know that, right, template strand is having polarity 5' to 3' complementary to it. Strand will synthesize discontinuously, so it is the lagging strand. Which strand it is? It is the lagging strand. Done? Done. So Okazaki fragments are there, right? So that's how it synthesizes. You can look at the diagram. So in the diagram itself, I have mentioned all the things. Okay, so now point is why lagging strand forms. So you know that here, because DNA polymerase, it can synthesize DNA in Y' to 3' direction, and it has to synthesize both the strands in opposite directions. So here you can see that it forms because DNA polymerase can only synthesize DNA in the 5' to 3' direction. Only in the 5' to 3' direction. So since two strands of DNA are anti-parallel or they are oriented oppositely, this creates a challenge. So in case of leading strand, DNA polymerase 3 synthesizes the strand towards the direction of replication fork and needs only one primer, but on the lagging strand, this must synthesize DNA away from replication fork, and that too in short stretches. Okay, so that's why the lagging strand form. Done, students? So this is it. So it is important to remove RNA primers from that. So DNA polymerase 1, its unique activity will be used, students, and then the gaps are filled by using the DNA ligase. So this part also I have mentioned, I have discussed. So 3', 5' to 3'. Okay, this is the unique activity. So I think now I have mentioned it thrice. Understood? So this is it. This is all about the DNA application. You can read that book. Each and every point will be clear to you. Okay? You'll be able to understand everything. Done? Sure? Are you sure? Okay? Done? Any doubts? Okay, so this is all about the DNA replication. So, next thing that we have to discuss is central dogma of information and the transcription, right? So, ready? So, any doubts? Any doubts? Quick. Any doubts? If there is any, do let me know. Done, students? Okay, so next topic is central dogma of information. Then we'll talk about the transcription. Okay, so here you people can see genes and enzymes. So here I'll tell you one thing. See, Beadle and Totem. Okay, Beadle and Totem. Right now we don't discuss this experiment a lot, but yeah, you should know this. Beadle and Totem, they proposed one gene, one enzyme hypothesis. Means one gene will form one enzyme. But later on, when the structure of enzymes, it was studied, then we got to know that it is not one gene, one enzyme. So later on, when the structure of enzymes, it was studied. So it was not like we got to know that it is not like that it is made up of one protein only. It is made up of more than one protein, right? Right. So at the end, what happened? See, so Beadle and Totem, they proposed one gene, one enzyme hypothesis while working on Neurospora. What is Neurospora? Neurospora is a fungus that belongs to what? Ascomycetes, and it is also known as, it's a fungi, it is also known as Drosophila of plant kingdom. What do we call it? It is also known as Drosophila of plant kingdom. Okay, it belongs to Ascomycetes. So it states that gene control functional trait through controlling synthesis of specific protein or enzymes formed by later. So it states that gene, what do they control? Functional trait, the characters that appear. Genes are controlling that. How? They make the specific protein or the enzymes are formed by these proteins. But later on, Janowski et al. Means Janowski and his team, they proposed one gene, one polypeptide. That we cannot say that one gene is forming one enzyme only, because sometimes enzymes are made up of more than one protein, right? So we can simply say one gene, one polypeptide. There is one gene is forming one polypeptide. Is that clear? Is that clear? Right? One gene is forming one polypeptide hypothesis, and it states that a structural gene specifies the synthesis of a single polypeptide. So that's what you have to remember. Okay, done. So one gene is not giving you one enzyme, but it is giving you one polypeptide, because you have to remember that enzyme, they can be made up of many proteins as well. So next point that you have to understand is central dogma of information. Next, what we need to discuss? We need to discuss central dogma of information. So it was propounded by Crick. Who gave it? Francis Crick. Central dogma of information. Who gave this? Yeah, who proposed this? Francis Crick. So it tells us about the flow of information. Just see, your DNA will form mRNA. mRNA will form protein. This is what we know. DNA, you know that it's a genetic material, it replicates, right? It increases, it multiplies, or it increases, it forms its replica. So DNA to mRNA, what do we call it? We call it as transcription. mRNA to protein, what do we call it, students? Translation. So this is the flow of information, that DNA will form mRNA. M here stands for messenger, right, students? And mRNA forms protein, that is the translation. So this is the flow of information. It's not like that DNA will immediately form the protein, it will form the mRNA first. So remember, when you talk about the transcription, so transcription in the case of prokaryotes, because prokaryotes, they don't have nucleus, so transcription occurs in cytoplasm, right? It is simultaneously with translation. But in eukaryotes, it will occur in nucleus. Now when you talk about the translation, so what is the site of translation? So it is cytoplasm for both, right? It is cytoplasm for both, for prokaryotes as well as eukaryotes. So transcription, my dear students, in prokaryotes it occurs in cytoplasm, and translation and transcription and translation are coupled there, right? So simultaneously it occurs. But in eukaryotes, the transcription occurs in the nucleus, but the translation will take place in the cytoplasm. So this is the flow of information. This is the central dogma of information. So that is what you have to remember. That is what you have to remember. So keep it in your mind. Now, the another part is central dogma reverse. Okay? So it is also known as Teminism. Teminism. This is the another name for it. So here what is happening, students? Your RNA will form DNA. So it's in transcription, DNA is forming RNA. So can I not say that it is the reverse transcription? Yes. So it is the reverse transcription or central dogma reverse or Teminism. So which enzyme is carrying out this? This will be done by reverse transcriptase. Yes. So here RNA will form complementary DNA, right? This DNA is complementary to RNA now. So this forms complementary DNA, and students, this DNA is single stranded. So this is the central dogma reverse. So the RNA viruses, right? Just say I'll give you the example of retrovirus. So retrovirus may you have RNA as genetic material. You have RNA as genetic material. So here you'll see what will occur. Here reverse transcription occurs. What will occur, students? Reverse transcription, which is known as Teminism. Okay, so take a note quick. Please, I'm back. Done. Okay. So the next topic is transcription. Okay. So next topic is transcription. So though I have mentioned everything on the board, but still you will get the excess of my handwritten notes. You can simply join our Telegram group, right? Unacademy NEET English, and even my personal Telegram group also you guys can join. So here, let's start transcription. So what exactly is the transcription? Just now I told you about the central dogma of information that DNA is going to form the mRNA and then it is going to form the protein. It is the way by which a gene expresses itself. We say the genes, they express a trait, they express a particular trait, but the question is how? How do they express it? Simple. DNA, genes, they are present on DNA, they make the proteins, and that's how they express a trait. Now, when it comes to the transcription, so formation of mRNA is the transcription. M here stands for messenger RNA. M here stands for what? Messenger RNA. And let me tell you, mRNA makes 1 to 5% of the cell's total RNA. Just 1 to 5%. It is not maximum. Maximum is ribosomal RNA. It is minimum 1 to 5% of the cell's total RNA, and this mRNA is even the least stable RNA. That's what you have to keep in your mind, that it is what? It is the least stable RNA. Now, what is the next point that you have to understand here? See, so as I said, DNA forms the mRNA and the mRNA forms the protein. Messenger RNA, 1 to 5% of the cell's total RNA, right? And it is the least stable RNA. Now, when it comes to the transcription, so when it comes to the mRNA, mRNA is just like the primary transcript of DNA. Means it's like you have a book, you're taking it to a shop for the photocopy. So that photocopy will have the exact information that that DNA is having. That book is having. Same is happening here. mRNA is just like the transcript, the photocopy of the information which is present on the DNA. But what is going to be the difference? You know that mRNA will have the uracil, not thymine. That's what you have to understand. Okay, one by one I'll clarify it. So when it comes to the, so this is what you have to remember. Now when it comes to the transcription, so transcription cannot start from any part of the DNA. If you think that transcription can start from any part of the DNA, no, that's not true. That is literally not true. Transcription starts from a point which is known as transcription unit. From where the transcription starts, students? It starts from transcription unit, right? It starts from what? It starts from transcription unit. So when you talk about the transcription unit, so here, this part, you see, you'll call it as promoter. Let me highlight that so that you can see that. This one is terminator. These are the three things that we study in a transcription unit. I'll elaborate it. Just allow me some time. And here, in between, you will see structural gene. What do you study or see? You'll see the structural gene. So I'm repeating this again. In the transcription unit, what are the things that we are going to see? We will talk about a promoter, then structural gene, which has to be transcribed, and then the terminator. So I believe it is clear from the words here. Promoter, something that will promote the transcription or will start the transcription. Terminator will stop it, and the structural gene that has to be transcribed. Isn't it? Isn't it? Isn't it? So that's what we have to remember, right? That is what we have to remember. So we have the promoter here, we have the terminator here, we have the structural gene here. So keep these three things in your mind when you're talking about the transcription unit. This point is clear? This point is clear, right? Now, the another thing, see, you have, this is the structural gene. Now, I'll elaborate it. See, not both the strands of DNA will get transcribed. Not both the strands of DNA will transcribed. Why? Let me tell you. Right? Let me tell you this. See, this is the DNA. P for promoter, T for terminator, and SG I'm using for the structural gene. Now look at this DNA, this transcription unit very carefully, okay? So obviously, if let's say here you have ATCG, randomly I'm writing the sequence I told you before. See, let's suppose this is a sequence, right? On one of its strand. So if it is A here, the complementary to it T. T complementary to T, it will be A. This is the DNA only, not the RNA. G. That's what we have. Yes or no? That's what we have. So this is the promoter, terminator, structural gene. If this strand of DNA is having ATCG, this will have TAGC. That is how it is going to be. That is how it is going to be. Now, whenever there is the synthesis, right? I told you synthesis will always be in 5' to 3' direction. Be it RNA or it is DNA, synthesis will always be in 5' to 3' direction. Are you getting my point? Are you getting my point? Yes or no? Yes or no? So it will always be in 5' to 3' direction. Now, what am I trying to tell you? See, here in the structural gene or in the transcription unit, because you have two strands of DNA, and as I said, only one strand will get transcribed. That's what I just said. And that strand is called template strand. Having polarity 3' to 5'. That's what you have to keep in your mind. See, so here, this is the template strand here. Can you see that? 3' to 5'. So in the structural gene, this is the template strand. It is also known as non-coding strand. It is also known as minus strand. Are you getting my point? It is also known as antisense strand. Now when you look at this, it is also known as non-template strand. And if I will say in NCERT exactly, the main point written is coding strand. So it is also known as non-template strand, coding strand, plus strand, and then it is also known as sense strand. Then it is also known as sense strand. Is that clear? Are you getting my point? Template strand is having another names. Non-coding strand, negative strand, yeah, minus strand, anti-sense strand. This is the non-template coding plus strand or the sense strand. Yes, this is actually the naming. If it is coding, that doesn't mean it will get coded. No, it is a template strand that will get coded. So this is a confusion. I have seen students making mistakes here. So kindly pay attention. So only one strand of DNA will get transcribed, and that strand is called template strand, having polarity 3' to 5'. And other strand of DNA is called coding strand. Are you getting my point? Another strand is called coding strand. Is that clear? Another strand is called coding strand. Along with that, you can also call it as non-template strand. You can also call it as plus strand. And as I said, sense strand. So these are the names here. And why is it so? Why only one? That we will discuss. See, now as I said, this 3' to 5', template strand is having polarity 3' to 5'. So don't you think complementary to it, the RNA will form in 5' to 3' direction? Don't you think so? So RNA will form in 5' to 3' direction because this one is the template strand. So now when you look at this part, just see. If there is A, here, what should be there? U. It will not be T, because in RNA we don't have thymine, but instead of that, uracil is there. So if it is T there, then A, then G, C, U, A, G, C, U, A, G, and C. This will be the sequence on the mRNA, right? This will be the sequence on mRNA, right? Template strand. Now look at it very carefully. See, that is the sequence on mRNA. mRNA should be in the direction, synthesis is always in the direction 5' to 3'. The template strand is in the direction 3' to 5'. Complementary to it, you have 5' to 3' strand synthesis. So now when you look at the sequence, see, TAGC, TAGC, TAGC. Here, UAGC, UAGC, UAGC. So basically on mRNA, the sequence of bases will be as such, will be similar to coding strand, but only at the place of thymine, uracil will be there in mRNA. Uracil will be there in mRNA. Right? So look at this. Done? So you people can see this, that on mRNA, the sequence will be similar to the coding strand, but only at the place of thymine. In coding strand, uracil will be there in mRNA. So that's why the strand is called as the coding strand. Because see, mRNA is having the same sequence just like this coding strand, right? That's what you have to remember. Now, for a moment, just imagine, okay? Now, for a moment, just imagine if both the strands will get copied. Chali, I'll use the next slide here. See, template strand 3' to 5', coding strand 5' to 3'. So let's say here, see, this is the DNA that we have. Now you know that if, let's suppose, both the strands will get copied, this strand will have polarity 5' to 3', and here you will see 3' to 5'. So look at it very carefully. Here what will you see? This is what you are going to see. This is what you are going to see. This is our actual mRNA. Yes or no? This is our actual mRNA. Yes or no? Yes or no? Right? So now let's suppose if both the strands will get copied, then what will you see here? So T, A, U, C, G, A, U, C, G. One second. G. That's what you will see. Now see, both the DNAs, they are also complementary to each other, na? They are also complementary to each other, right? They are also complementary to each other. So they will result in the formation of a double-stranded RNA. So why both the strands are not get copied? It is very simple. See, we know that 3 bases, right? 3 nitrogenous bases, they give us 1 amino acid. This is the codone. Listen to me. This is the codone, right? 3 nitrogenous bases, 3 nitrogenous bases, they give us 1 amino acid, right? So we say it is the triplet base codon, right? Triplet base codon. So just imagine if both the strands of the DNA will get transcribed, if both the strands will show the transcription, then what's going to happen, students? See, this strand will give us a different protein, the information for a different protein, and this strand will give us the information for a different protein. So it's like same strand, same part of DNA, but it is giving us the information for the synthesis of different amino acid, and that to result in the formation of different proteins, which is not right, which is inappropriate, isn't it? So we will not get the proper information, right? We will not get the proper information here, number one. Number two, if both the strands will get copied, the RNAs that will form, they will also be complementary to each other, right? They are also complementary, so that will result in the formation of a double-stranded RNA. Then what will be affected? The translation, right? Then what will be affected? Double-stranded RNA is there now. Then how are you going to read it? So the translation part will be affected. So that's why only one strand will get synthesized, will get copied, and that is your template strand. Other strand is the coding strand, and that will have the same sequence like mRNA. Only at the place of thymine, uracil will be there. Now, see, in the transcription unit, here, just this point to this promoter, here you will see the start site, the site from where actually the mRNA will form. Now see, if this is the start point, guys, listen to me very carefully. If you go towards this direction, this is upward, this is downward. Now, just like integers, you know, now here, if it is here 0, this is in minus. So in the case of DNA, that is how we, mention the position. Just let's suppose if I'm saying that minus 10 bases from start point, means I will consider this as the start point. Minus is indicating upward direction, minus 10 bases. Plus 10 bases means I have to move this side. So this is something that you have to understand. This is a start point, upward direction and the downward direction. This point is clear. Now, another point that you have to understand, that in transcription, here in DNA, generally references are made with respect to coding strand. Do you know that? Generally references are made with respect to coding strand. What does it mean? This is another common mistake that I have seen students making. See, I'll tell you. Let's suppose I'm facing the camera. When I'm facing the camera, I'm saying my right side is having a door, my left side is having the window. Are you getting my point? Are you getting my point? I'm facing the camera, then I'm saying my right side is having the door, my left side is having the window, because I'm facing the camera. Now, if I'll face away from the camera, again, I'm saying my left side is having door, my right side is having window. Initially, I said my right side is having door, my left side is having window. Now I'm saying my right side is having window, my left side is having door. Why? Because earlier I was facing the camera, now my back is facing camera. I'm facing away, right? I'm looking away from the camera. But the door is at its place only, the window is at its place only. There is no change in that. It is about my reference point. Like sometimes we say my right side is having a shop, my left side is having the road. Now if you'll turn, it will be opposite to that. That is the point that I want to tell you. If I consider the reference of coding strand, see, so this is a structural gene. You know that this is going to be your coding strand. Okay. And this one is what? This one is your template strand. So, if we take the reference of coding strand, done, then promoter is at, this is coding strand. Where is promoter? Promoter is at 5' end. Am I right? if I take the reference of the coding strand, where is promoter? The promoter is at 5' end. And terminator is at 3' end. Am I right? Terminator is at 3' end. But, so generally the references are made in accordance with the coding strand. But if we consider the reference of if we make the reference of template strand, then promoter is at 3' end and terminator is at 5' end. Are you clear with it? Yes. Yes, read it. So that's the another point that you have to remember. So now we have discussed. Yes, please. Done? Done? Sure? Done? Sure? So that's how the reference will be made. So this is about the RNA, DNA and why can't we synthesize, right, both the strands of DNA. Now the next point is the process, the mechanism. Okay. So obviously there are 3 steps: the initiation, elongation and the termination. So firstly we'll talk about prokaryotes. Okay, so now you know that the transcription will start from in the transcription unit. But before that, yeah, I would like to mention one more thing. See, when you talk about a gene, right, a gene or the DNA, when you talk about the gene or the DNA, I'll see this. I'll mention it here. I will not write it. I'll mention it here. So what is a gene? Gene is defined as the functional unit of inheritance. Gene factor. Factor was a term used by Mendel. Then later on, Jonathan called it as gene. So gene is basically the unit of inheritance. And when it is gene, right, it's difficult to define it in terms of DNA sequence. Because DNA sequence is basically coding for the tRNAs, mRNAs. It is also acting as a unit of replication. It is also, taking part in crossing over. There are different things. So if we have to define gene in terms of DNA sequence, that will be little difficult, right? So the segment of DNA, which signals or which makes the polypeptide, because now we are saying one gene, one polypeptide. So one gene, so which is making polypeptide, we use the word cistron for it. So if I have use the word cistron. If I have used the word cistron, right? If I'm calling something as cistron, so it's simple that it is that segment of DNA which is making polypeptide. Okay, so it is that segment of DNA which is making polypeptide. Is that clear? Is that clear? So it is that segment of DNA which is doing what? Which is making polypeptide. Okay, so cistron, you remember that segment of DNA which makes polypeptide. Am I clear? Which makes polypeptide. So this is the another concept that you have to understand. So prokaryotes. In prokaryotes so in prokaryotes, you people see P for prokaryotes, polycistronic mRNA. In eukaryotes, we see monocistronic mRNA. One is polycistronic, another is monocistronic mRNA. One is polycistronic, another is mono. So polycistronic mRNA. This mRNA will give you the information for synthesis of more than one polypeptide, right? Gives information for synthesis of more than one polypeptide. So here you will see there will be more than one structural genes as well that will make polycistronic mRNA. And now in the case of eukaryotes, what you will see, monocistronic mRNA. Monocistronic mRNA. So here you will get the information for the synthesis of one polypeptide. So this part is important. Do not make the mistake here. So prokaryotes, when you write the prokaryotes, remember they are monoreplicant. I have seen students confusing these terms. And polycistronic, they are monoreplicants and polycistronic students. And when you people talk about the eukaryotes, they are multi-replicant, and monocistronic. Yeah, polyreplicant and monocistronic. Important information. Is that clear? Okay. So now let's talk about the prokaryotes. So here, transcription in prokaryotes. That's what we have to talk about. Transcription in prokaryotes. So here students, firstly the site is, you know that it is going to be cytoplasm, right? It is going to be cytoplasm. So let's suppose this is the DNA. See, before going forward, so here which enzymes are we going to discuss? You'll talk about RNA polymerase. Yes, RNA polymerase. We can also call it as DNA dependent. Yes, please. DNA dependent RNA polymerase. Just primase was also the DNA dependent RNA polymerase. This one is also. Right. Here you'll talk about sigma factor. Helps in initiation of transcription, or I would say the start. S for sigma, S for start. Then rho factor, rho helps in termination. That is, it will help in stopping the mRNA synthesis. Done. Then here we'll talk about a Tata box in promoter region, which we call at Priv-now box. Priv-now box. We also call it as Priv-now box. So these are the terms which are associated with your transcription in prokaryotes. Now, students, if we have to open the DNA double helix in the transcription unit, which enzyme are we going to use? If we have to open the DNA double helix, then which enzyme are we going to use? Please tell me, which enzyme are we going to use if we have to open the DNA double helix? Quick. Yes, quick. Guys, quick. Hanji, quick. Huh? Anybody? So remember, it is the RNA polymerase, right, that helps in opening DNA helix during transcription. So it has been asked as a question in NEET 2020, right? So it is a PYQ of NEET 2020, that enzyme that unbinds DNA helix during transcription. So what should be the answer for this question? What should be the answer for this question? This is going to be RNA polymerase. So yes, it happens, beta, that when we just read the enzyme that unbinds the DNA helix. So if you miss this point, that we are asking it during transcription. We are asking it during transcription. So in transcription, it is RNA polymerase. It is not the DNA replication. So it's a NEET 2020 PYQ, and that's what you have to, right? That's what you have to remember. Okay? That's what you have to remember. Understood? Understood? So RNA polymerase help in opening DNA helix during transcription, or it also can't open DNA in one go, as it takes a lot of energy to break hydrogen bonds. Is that clear? So that's what you have to remember in the transcription here. Now the second part, see, there is a sigma factor, and this sigma factor, it is in prokaryotes only. You are not going to find it, not in eukaryotes, right? So it is in prokaryotes only, not in eukaryotes. Why? Because eukaryotes have some other factors. Remember this. Because eukaryotes have some other transcription factors. Obviously, that's not the part of your syllabus. But if I have to tell you, transcription factor D, A, B, G, H, right? They are the transcription factor. TF, G, A, right? TF, D, A, B, G, H, like this. That is how they are used. But here in prokaryotes, you will have the sigma factor, S for sigma, S for star. It helps in the starting of the transcription. So this factor, because see, your RNA polymerase is again the multimeric enzyme. So sigma factor is one of its subunits. So this factor takes RNA polymerase to the promoter region, and in the promoter region also, site from where it has to open DNA helix. So in the case of prokaryotes, what you people are going to see, in this promoter region, there is a Tata box. So can I say that it is upstream to the start point, right? Remember promoter, then start point. So upstream to the start point in the promoter region, here you will see a Tata box. Why do we call it as a Tata box? Because it is having 6 base pair, TA, TAA, T, right? These are the 6 base pairs which are present there. They are thymine adenine rich, and you know that thymine and adenine is attached with the 2 hydrogen bonds, so it's easy to break it, right? It is easy to break it. So here it is also known as priv now box. Here it is also known as priv now box. What do we call it, guys? We call it as priv now box. Am I clear? Priv now box. Is it clear? So priv now box, where this RNA polymerase will come, it will start the opening of DNA helix, and then from the start point, it will just start making the mRNA. So it's not like that in one go immediately it starts the mRNA formation. Many times mRNA form, it gets separated, then finally it forms. So sigma factor will only help in initiation. It will not help in elongation. Once the RNA polymerase start the mRNA synthesis, it will leave. It's like there is a baby, right, who's going to the school, then that's the first day of that baby. So you'll drop that baby to the school, you'll wait for some time whether that baby is crying or not, and then you'll come back because that baby will eventually get adapted to that. This is a normal thing, general thing. So the sigma factor will also carry that RNA polymerase to that promoter region, to the starter box, right, so that it can just open up the DNA helix first and then can form the mRNA. So Tata box there is known as Privnow box. It is having 6 base pair. So in prokaryotes we call it as Privnow box, but in eukaryotes it is known as Goldberg-Hogness box. In eukaryotes, what do we call it, people? We call it as Goldberg-Hogness box. Is that clear? So that is again a Tata box. It is having the 7, right? How many base pairs do we have here? Here you will see the 7 base pair, right? Here, what will you see? You will see the 7 base pairs. How many base pairs are we going to see, guys? We are going to see the 7 base pair. And what do you call it? You call it as the, yes, please. You call it as the Goldberg-Hogness, sometimes simply the Hogness box. Okay? Sometimes simply the Hogness box. So I believe this is clear. So it is having 7 base pairs. Understood? It is having 7 base pairs. Understood? So it is TA, T. These are that 7 base pairs. These are the bases. You know that complementary to it, base pairs will be there. Understood? Sure? Is it clear? Sure? Done? Any doubts? Any doubts? So it is also the or TA, T, A, T, 7. TAT, okay? That can also be there. So this is what you have to remember. So now come to this diagram of the NCERT. See this. So sigma factor, even in your book you can write it, only helps in initiation but not in elongation. Are you getting my point? It will only helps in elongation, initiation but not in elongation once RNA polymerase start making RNA, it leaves. Are you getting my point? It leaves. So here you can see sigma factor will carry this RNA polymerase to the promoter region, and to be more precise, in the Tata box there. So Tata box may it will bind, it will open the DNA helix. Once it will start that mRNA from the start point, and then sigma factor will leave. So sigma factor only helps in initiation but not in elongation. Once RNA polymerase start making RNA, it leaves. So then see, they are moving, it is getting opened. Then when it comes to the termination, so rho factor can help. So if it is rho factor that is helping in termination, so that is going to be rho dependent termination of mRNA, but it can be rho independent as well. Am I clear? It can be rho independent as well. So rho independent termination is also there. So here a here loop also there. So a kind of hair loop like structure, like a kind of, in the mRNA you'll see the codons will be like that, the kind of, you know, hairpin like structure will form, and the termination will occur. So please tell me this diagram. Is it clear in your mind? Yes. Guys, right now, see, let me finish this, then you will have the break. Yes, please. Yes, please. Yes, please. Is that clear? So here the termination will be there. Okay? Now guys, let me finish this topic and RNA processing part. Then we'll talk about it. Is that clear? Done? So this is all about the transcription in what? In the case of prokaryotes. Now I'll discuss the same in eukaryotes as well. I'll discuss the same in eukaryotes as well. So see, already told you no sigma and rho factor there. There are other factors. Plus, in eukaryotes, you will talk about there is RNA polymerase. Yes, 1. There will be RNA polymerase 2. There will be RNA polymerase 3. So that is what you all will see here. Are you getting my point? RNA polymerase 1, 2 and 3. See, it is the RNA polymerase 2 that will form hnRNA, which means heterogeneous nuclear RNA. It is also known as immature RNA. It is also known as pre-mRNA, right? RNA polymerase 1. See, because it is in the case of eukaryotes, 28S ribosomal RNA, 5.8S ribosomal RNA. Done? RNA polymerase 3. From 3 you have to remember tRNA plus it forms 5S ribosomal RNA plus it forms small nuclear RNA. That is snRNA. Are you getting my point? That's what you have to discuss here. Is that clear? Is that clear? So, no sigma and rho factor here. RNA polymerase 1, 28S, 5.8S ribosomal RNA. 2 will form hnRNA, heterogeneous nuclear RNA, immature RNA, pre-mRNA, RNA polymerase 3, tRNA, 5S ribosomal RNA or this. So yes, they definitely ask question from this part. So this is something which is very important, right? Which is very important. So here you don't have one type of RNA polymerase. Here you have, right here you do not have one type of RNA polymerase, but instead you have 3 different types. Now, the another point is, see, in the case of eukaryotes, you know that DNA is in the nucleus. So if DNA is in the nucleus, then obviously transcription, it is also in the nucleus. Are you getting my point? Transcription is also in the nucleus. But what is the another funda or the another point here? let me tell you, in eukaryotes, there is the concept of split genes which are having evolutionary significance. What do you see there? In the case of eukaryotes, you see the concept of split genes. So what is the meaning of split genes? In eukaryotes, we'll see that DNA is having a region which gives DNA is having region which give information for DNA is having region which gives information for synthesis of protein, and that region regions are also interrupted by regions which do not give information for synthesis of proteins, right? Which also gives information for the synthesis of, which do not give information for the synthesis of proteins. So let me elaborate it. Okay, let me elaborate it. So in DNA, regions that give information for protein synthesis, we call them as exon, or they are the coding regions, and non-coding regions are known as introns. Firstly, you tell me, are you clear with the term exons or introns? Are you clear with the terms exons and introns? So let's suppose if this is a gene, just giving you one example, right? Just giving you one example. Let's suppose here you have the coding regions. In between you have the non-coding regions. So if I have to give you the example, you can see exon, exon, exon. In between you are having intron, intron. So this is basically a split gene. Yeah, these are the split genes. So split genes are the genes which are having a part which is giving the information for making protein, and there is another part which is not giving the information for making protein. So coding region, right? So again I'm repeating because might be some students are knowing it for the first time. So coding regions are exons, and non-coding regions are introns. Are you getting my point? So this part is having, this is actually having an evolutionary significance, right? It is having what? It is having an evolutionary significance, the presence of the exon and intron. Now, it's not like that. Almost all, like in eukaryotes, mainly split genes are there, but genes for histones you understand, right? And interferons are non-split genes, right? They are the non-split genes. Are you getting my point? Are you getting my point? So in eukaryotes, mainly split genes are there, but the genes for interferons and the histones are non-split genes. So split genes means where the coding regions are interrupted by non-coding regions, introns. So obviously if that genes will form, right? So such DNA will form mRNA having exons and introns. Don't you think so? Now I'll use the term exon and introns. So make yourself familiar with this term. Exons mean the regions which will give the information for the synthesis of protein. Introns are the regions which will not give the information for the synthesis of proteins, right? Yes? Understood? Yes? Understood? Understood? So that's what you have to remember. So it will form mRNA having exons and intron. So such mRNA is heterogeneous nuclear RNA or hnRNA. So we call it as hnRNA slash immature RNA. So this is not even the mature RNA. So this is something that you can learn here, that in eukaryotes transcription will occur in the nucleus, and here what do you see? What will form? Just a minute. Let's suppose this is nucleus. So here what you see, what will form? So hnRNA forms here. hnRNA forms here. So that is an immature RNA. That is an immature RNA, and that RNA is having exons and introns, means coding regions and non-coding regions. So I believe this part is clear to all of you, right? I believe this part is clear to all of you. Yes? Sure? Understood? Sure? Any doubts? So please write it down. Quick. Quick, guys, just take a note. Quick, guys, I'm waiting for you all to write it down. Quick. Noted? One minute. Okay. Beta, session duration. Session will go till the chapter completion. Okay. So this is what we have to remember. Okay. So 3 different types of RNA polymerases are there. So just have a look. Now we'll talk about the processing. So in eukaryotes, 3 different types of polymerase. Polymerase 1. Yeah. So here in my notes here, I just missed. This is what I was thinking, that I missed something. It is 18S ribosomal RNA. That's all. Okay. So here you can see that 28S, 18S, 5.8S, RNA polymerase 2. It will form your hnRNA, heterogeneous nuclear RNA. It is the immature RNA. It is the pre-mRNA. It is not even the mature RNA. Why? Because in DNA, split genes are there, and that split genes contain the coding and the non-coding regions. That split genes, they contain what? They contain the coding as well as the non-coding regions. So because they contain the coding and the non-coding regions, so obviously in the mRNA, right? Obviously in the mRNA also you will see the same things. Yes or no? Obviously in the mRNA also you will see the same thing. What same thing you are going to see? mRNA will also be interrupted like this. It will also be having the coding and the non-coding regions. Am I clear? That mRNA will also be having the coding as well as the non-coding regions. Am I clear? Am I clear? So yes, that is why it is very important to process that. Are you getting my point? That is why it is very important to process that mRNA. Is that clear? Is that clear? Sure? Just 1 minute, guys. Having some issue with the mic. Just a minute. Okay, so RNA polymerase 3, tRNA, 5S rRNA, and nuclear RNA. So I believe you remember that much, that in the nucleus there is nucleolus. What do we have? Nucleolus. So nucleolus is referred to as the ribosome factory. So actually it is in the nucleolus your rRNA forms, rRNA and proteins forms, right? So but here you will have 28S, 18S, 5.8S ribosomal RNA, but when it comes to the 5S ribosomal RNA, that will form here in the rest of the nucleoplasm, because it will be formed by RNA polymerase 3. So that is something very important. Now in eukaryotes, you know that Tata box is there having 7 base pairs. Here too we have mentioned the bases. So I told you about this as well. It is Goldberg-Huggins box. Then I told you that there is the concept of the split genes as well. So genes that contain non-functional ya non-coding part, they are called introns, along with the functional or coding part that are known as exons. So these are the split genes. Most of the eukaryotic genes are the split genes, but the point that you have to remember is that histones and interferons, they have the non-split genes, right? Histones and interferons, they have the non-split genes. Am I clear? What do they have? They have the non-split genes. Is that clear? Sure? Is that clear? Done? Okay, done. So most eukaryotic genes are the example of split genes, but the genes that form histones and interferons, they are non-split genes, right? And then the split gene represent an ancient or the primitive feature of a gene, right? It is having the evolutionary significance. So mostly prokaryotic genes are non-split. So here the mRNA, it will be mature only. That is why immediately with the transcription, translation starts, right? That is why that events are simultaneous. Am I clear? That's why that events are what? They are simultaneous, right, students? So transcription and translation can be coupled in bacteria, right? So in eukaryotes, because split genes are there, so you will see the RNA processing is required. So I believe this part is clear, and you are not going to see anything like that in prokaryotes. So this is the DNA. This is forming 5' 3' HNRNA, that is immature RNA, right? So it is heterogeneous nuclear RNA. Heterogeneous nuclear RNA. So it is also known as immature RNA. Am I clear? So if I want this RNA to be right mature, so what I have to do? So now there comes a process, RNA processing. What has to be done here, students? RNA processing. What has to be done here? RNA processing. Is that clear? So it will be there in eukaryotes only. In eukaryotes only, not in prokaryotes, right? And it involves 3 steps. Are you getting my point? It involves 3 steps. There will be capping, there will be tailing, I'll explain it, don't worry, and there will be RNA splicing. So what will be there in the RNA processing part? There will be capping, tailing, RNA splicing. Capping, tailing, RNA splicing. Let's discuss again. Capping, tailing, RNA splicing. Capping, tailing, RNA splicing. That is something we have to see. Again, I'm repeating. Capping is there, tailing is there, and then comes what? Then comes the RNA splicing. Capping is there, tailing is there. Then comes what? Then comes the RNA splicing. Am I clear? Capping, tailing, RNA splicing. That's what you have to remember. Okay? Okay. So capping, tailing, and then comes what? Then comes the RNA splicing. Now, let's understand this. RNA splicing I'll start later. I'll start with the capping and tailing. Just see. That's the mRNA that we have. Again, I'll repeat the same thing. Just let's see. That's the mRNA. That's the mRNA that we have. See. Let's suppose this is exon, this is intron. So yes, we have to remove these introns. We have to rejoin the exons. This synthesis here also will be in 5' to 3' end. So when it comes to the capping, what will happen at 5' end? An unusual base. Why unusual? Because normally base is what? They are monophosphate. An unusual base 7-methyl guanosine triphosphate. 7-methyl guanosine triphosphate will be added by the enzyme guanyl transferase. Which enzyme is going to do the same? It is going to be your guanyl transferase. Are you getting my point? So at 5' end here, you'll see 7-methyl guanosine triphosphate will be added with the help of enzyme guanyl transferase. So that's how you can write. That's how you can write. And at 3 dash end, what will be done? Tailing will be done. Tailing will be done. So here at 3 dash end, what is the keyword here? Please pay attention. Poly. Poly means many. Many adenylated residue means so many. You can say that 100 to 200. So here at 3 dash end, polyadenylate residues. Sorry. So here at 5 dash end, polyadenylate residue will be added in a template independent manner. That's the keyword here. In a template independent manner. Yes, students. In a template. So here at 3 dash and polyadenylate residues will be added in a template independent manner by enzyme polyadenylate polymerase. By enzyme polyadenylate polymerase. Right? That is polyadenylate polymerase, PAP. PAP. And what is the keyword? The keyword here is template independent manner. Okay? The keyword here is template independent manner. So in template independent manner, like, see, normally this is what we have studied. Normally, this is what we have studied, that there is going to be a DNA, right? So RNA. So if you have to make the RNA, there will be the DNA template, complementary to it, bases will be added. But when it comes to the addition of these, right? When it comes to the addition of these A here, so it is in template independent manner, right? It is in template independent manner by the use of enzyme polyadenylate polymerase. So this is capping, this is tailing. Tailing will have so many adenylate group. Capping will have 7-methyl guanosine cap that help this mRNA to bind with smaller subunit of ribosome during translation. That is also the role here. That's also this particular cap helps. Okay, so first thing is this, and this capping and tailing. Why is it important? This capping and tailing. This capping and tailing protect mRNA from nucleases, okay, which are in the cytoplasm. So it is going to protect this mRNA from that. Okay. Now the next part is, see, RNA splicing. So how will it be achieved? It is basically the removal of introns and then joining of exons. Are you getting my point? It is basically the removal of introns and joining of exons. This is what RNA splicing is. So spliceosomes are required for it, right? So what are these spliceosomes? These are snRNPs, means small nuclear ribonucleoproteins and some snRNAs. They are together. There are some other proteins also. So they are together forming the spliceosome. Okay. So small nuclear ribonucleoproteins, and these are pronounced as SNRPs. Okay. Not SNPs. You call them as SNRPs. Small nuclear ribonucleoproteins, pronounced as SNRPs. So what's going to happen? These spliceosome, right? See, you know that you will only see them in eukaryotes. Why? Because splicing is there in eukaryotes only. Okay, so there is a PYQ on the basis of that, and that was spliceosomes are found in yeast, bacteria, let's suppose E. coli. One more option was there, I don't remember that, but theek hai. Let's suppose I'm writing Pseudomonas here. So obviously spliceosome are found in yeast, because yeast, though it is unicellular, but it is the eukaryote. Okay, it is the eukaryote. So spliceosomes are found in eukaryotes. They help in the removal of introns. So wait, let me show you. So when you see this particular diagram, see this. Introns are there. So in the NCERT itself, you can write non-coding regions, coding regions, so that you don't forget. This cap help mRNA, and this is what the NCERT reading it, to get attached to 40S subunit of ribosome during translation. Understood? Then here, these structures, their naming can come in paper. These are spliceosomes. Understood? Right? So they remove introns and help in the joining of exons. Am I clear? So that is it. So let me show you the spliceosome here. So see this. These are small snRNPs. They join to this 5' splice site or 3' splice site of these, introns. Then they'll cut it. Detailed mechanism is not required here. So they remove this intron in the form of a lariat. Like in Hindi, lariat is basically the jabuk, like that. So introns that detaches and splice itself, connect to make a, right, detaches and splice sites. So see, intron are removed, this side and this side they'll join. You'll have the mature mRNA. Introns, previously it was thought that they were junk, but most are used in other processes. So snRNPs detach from the introns and are used for splicing. So look at this diagram. You can see that how, these snRNPs, some other proteins will bind, and then this is how they are going to make it. So here I have mentioned this process in detail as well. You can simply see this. So RNA processing, capping, tailing and splicing is required. So that also protect mRNA from nucleases. Understood? Then here you can see in capping an unusual nucleotide is added at 5' end of hnRNA by guanyl transferase enzyme. I told you about 7-methyl-guanosine triphosphate here. In tailing, the adenylated residues, right, 200 to 300, but in a template independent manner by the enzyme polyadenylate polymerase. Then when it comes to the RNA splicing, so introns are removed, exons are joined in a defined order. This is done with the help of spliceosome. And as I said, small snRNP, small nuclear ribonucleoproteins, right here basically these are SNRPs. So small nuclear ribonucleoprotein is having snRNAs with some proteins. So see, some other proteins and snRNAs, they are forming spliceosome, right? They are forming spliceosome. Is that clear? So these spliceosome use energy of ATP to cut the RNA, release the intron, join exon to produce functional or the mature RNA. So this RNA in cytoplasm form protein by translation. So this is what the RNA splicing is. And here you can see, so basically SNRPs, snRNPs, how do we pronounce them? SNRPs, right? So they contain some proteins and small nuclear RNAs which will join there, make the cuts. So in the lariat formation in this way, so see, excised lariat, this is the removal of introns. So that's how it will be eliminated. So this is the final RNAs. You can see that. So this is the monocistronic eukaryotic mRNA having 7-methyl-guanosine cap. This is the coding region of the mRNA, and right after this cap you'll see some untranslated regions, right? You'll see some untranslated regions at 5 dash end, just before the coding region and just after the coding region. Or I would say, let's suppose if here you have the start codon, just before start codon you'll see, after stop codon you'll see UTRs, untranslated regions, right? And in the polycistronic mRNA, there also 5 dash or 3 dash UTRs are there. So this RNA splicing, right? So this removal of intron is a primitive feature, or we can call it as reminiscent of antiquity. Means basically it is like ki you're talking about something primitive, right? So it's a primitive feature, or we can also call it as reminiscent of antiquity. So, and process of splicing represents the dominance of RNA word. So this is it, guys. So I hope this is done. So let's have break. So break time. Let's meet at 10. So again, you'll get the break for half an hour. I thought I'll finish the genetic code first, but theek hai anyhow. So after this, we have to finish 3 main topics: genetic code, translation, operance, actually 4 main topics, HGP. Right? DNA fingerprinting we will discuss once we'll complete the Hi guys. Sorry for being late. I was just having my dinner. So, and wow, lucky you. You got a nice break. So, how are you all? Done? Ready to finish the chapter? Yes, everybody. Ready to finish the chapter? Okay, so let's get started. So, now what are we going to discuss? Now we are going to discuss about the genetic code. Okay, so transcription done, replication done, basics done. So now let's talk about the genetic code. So you know that we have the DNA will form mRNA will form protein. That's what we have. DNA will form mRNA will form what? mRNA is going to form the protein. This part is clear. Now, see, what is this mRNA? Is it the polynucleotide chain? Yes or no? Is it the polynucleotide chain? Yes or no? Yes or no? Right? And when you talk about the protein, what is it? It's a polymer of amino acid, or I would say it's a polypeptide chain, or it's a polypeptide. Right? It's a polypeptide. Yes or no? It will not take much time, genetic code. Only the translation topic will take the time. So hopefully by 1 we'll be free. Okay. Done? Yes. So polypeptide, polynucleotide chain and the polymer of polypeptide. See, and DNA itself is what? This is also a polymer of nucleotide, or I would say this is also a polynucleotide chain. So the point is very simple. See, mRNA being a polynucleotide chain is forming the polypeptide chain. Are you getting my point? See, when DNA forms the mRNA, we can easily explain it because it is also a polynucleotide, it is also a polynucleotide. But when it comes to mRNA to protein, so scientists, they were really confused that how a polymer of nucleotide can form the polymer of amino acid. Okay, so this particular relationship, right, that how a polynucleotide chain is forming the polypeptide chain, it is explained by genetic code, right? So this relationship is explained by genetic code. So the term genetic code is given by George Gamow, right? So the term genetic code is given by George Gamow. So he was the physicist, right? He was a physicist. So he suggested that these bases in combination, they form the amino acid. Is that clear? So the relationship, right, between this polymer of nucleotide and the polymer of amino acid is explained by the genetic code. And who gave this term? The term was given by the George Gamow, and he was a physicist. He suggested that these bases can form the amino acid but in combination. So let's start this part, okay? And there are other scientists also who contributed. We will discuss about the Hargobind Khorana, Marshall Nirenberg. We'll also talk about the Severo-Ochoa enzyme. So one by one, let's understand that. Now see, when you talk about the mRNA, how many bases do we have? We have 4 bases. How many bases do we have? 4 bases, U, C, A, and G. Now, when you talk about the protein forming amino acid, what? When you talk about the protein forming amino acid, then you talk about 20 amino acid. Yes or no? 20 amino acid, but these are not the only amino acids. We have around 200 to 300 amino acid, but the protein forming amino acids are 20. So 21st amino acid is also there, not given in NCERT. Even the 22nd is also not given in NCERT. So 21st is selenocysteine and 22nd is pyrrolysine. So selenocysteine and pyrrolysine. Is that clear? So 21st is selenocysteine and 22nd is pyrrolysine. Done, students? Yes, and you have to remember this. Now, what's the next part? So what are these 20 amino acids? See, try to remember the names. Alanine, arginine, asparagine, aspartic acid, cysteine, glutamic acid, glutamic acid, glycine, isoleucine, leucine, lysine, methionine, proline, phenylalanine, done. Serine, tyrosine, threonine, tryptophan, and then comes the valine. So these are the 20 amino acids that we have, right? So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So one is missing. Which one is missing? Histidine is missing, right? H. Yes, so here we are going to write histidine. So these are the 20 amino acids, okay? So these are the 20 amino acids. If you can remember them, very good. Try to remember the 3 alphabet code and even 3-letter code, and even try to remember the 1-letter code as well. Like I'll give you the example I told you earlier. Arginine denoted by A, okay? Arginine, asparagine, arginine denoted by R. Right, it is denoted by R. So this is what you can try to remember, because this part is also there in the biomolecule chapter. So we have how many amino acids? 20. So 20 amino acid means alanine, arginine, asparagine, aspartic acid, cysteine, glutamine, glutamic acid, glycine, histidine, isoleucine, leucine, lysine, methionine, proline, phenylalanine, serine, threonine, tyrosine, tryptophan, and then we have what? We have the valine. So these are the 20 amino acid, and as I said, there is the 21st amino acid as well, right? 21st amino acid is also there, that is known as selenocysteine, and 22nd is pyrrolysine. Am I clear? So these are the amino acids. Now, how many bases do we have? We have 4 bases. How many bases do we have? We have 4 bases here, isn't it? So it's U, C, A and G. So let's suppose if we take 4 bases, right? So if we go it in the, consider it in this way, 4 bases means 4 amino acids, right? Not possible. Now, if we take combination of 2 bases, if we do what? If we take combination of 2 bases, so first position can be filled by any of the 4 bases. Second position can also be filled by any of the 4 bases. So then in that case, you will get 16 codons. So which will code for 16 amino acid? Again, the data is insufficient. Now if we take the combination of 3 bases, see, first position can be filled by any of the 3 bases. Next position also. So how many codons do we have? 64 codons. So they are sufficient. Right, so if we take the combination of these 4 bases, right, and we take 3 bases in 1 codon, so obviously it can give us the 20 amino acids. So simple, 64 minus 20, 44 codons are left. Out of these 44 codons, students, this is the information that you really need to remember. 3 codons are the stop codons. We are left with 41 codons, okay, because here 3 are stop codons, and then after that, out of these 41, they are again coding for these 20 amino acid. So here we can say one amino acid can have more than 1 codon, isn't it? One amino acid can have more than 1 codon, isn't it? Yes or no? Yes or no? Yes or no? Is it clear? Now, let's talk about this. So let's talk about the genetic code. So I told you, it is the relationship between the sequence of amino acids in a polypeptide chain, that is in a protein, and the nucleotide sequence of DNA or mRNA. This is what the genetic code is, number 1. Number 2, it is the sequence of nucleotides on mRNA molecule which contains information for the synthesis of polypeptide chain. Obviously, the sequence on the mRNA, the basis, their sequence is going to decide which amino acid will be there, right? And how that amino acids are going to join and they'll form the polypeptide. Then term was given by George Gamow, important it is. So discovered by Nirenberg, Mattei and Khurana. They were the one who discovered the genetic code, right? So here 20 types of amino acids are there. I told you about 21st also. What is it? Please repeat it. Selenocysteine. What is this 22nd? This is pyrrolysine. Right, this is what? This is pyrrolysine. So selenocysteine and pyrrolysine is there. Understood? Now let's move to the next part. So here what do you see? A codon. It is the nucleotide sequence on the mRNA which will code for the particular amino acid. So all the points are also mentioned here. So as I said in our official Telegram group, you guys can, from that official Telegram group, you guys can download this PDF. Now, in 1954, George Gamow, right, he suggested that if we want to code the amino acid, the 20 amino acid, the combination of bases can be there. So I already told you. So it is the triplet codon, right? So triplet base, if we consider three bases, the combination of three bases, we'll get 64 codons. And out of that, you know that 20 to directly are giving us amino acid. 3 are the stop codon. They stop translation, right? They stop protein synthesis, I would say. And here, they are also known as nonsense codons. Okay, then these 41, they'll code for the amino acid. So one amino acid can have more than one codon. Is that clear? Is that clear? Now let's discuss the scientist discovery, which is again a previous year question. So here you can see Hargobind Khurana. So Hargobind Khurana developed the chemical method by which we can make the by which he was able to make the homopolymers and copolymers, right? With defined combination of bases. If I have to give you the example, like for an example, he took all the same bases, like one base multiple times. So he checked, it has given him the same amino acid, जैसे phenylalanine, so on. Right? This is just the example I'm randomly giving you. Then he took what? Cu, two bases and their combination. See, CuC will give you one amino acid. UCO will also be giving you one amino acid. CuC will give you amino acid. UCO will also give you amino acid. So in this way, how many amino acid you will see here? You'll see the, how many amino acid you will see here? You'll see the 2 amino acid. So if I talk about CuC, what's that? CuC, that will code for leucine. And what about UC? UCU will code for serine. So leucine, serine like this. Okay. So just giving you one example. So he developed the chemical methods which helped in synthesizing the RNA molecule with defined combination of bases. Right? With what? With the defined combination of bases. So this part is clear, everybody. Yes. So defined combination of bases. Then Marshall-Nirenberg cell-free system for protein synthesis, right? Like obviously with the help of this discovery, the scientists, they were able to make the protein even without taking the help, like without considering the cell. In a cell-free system, in a cell-independent system, they started making the protein and it helped them to decipher, to decode this genetic code. And then we talk about the Severo-Ochoa enzyme, because that is again a very important discovery. Why? Because here, see, here we talk about this enzyme, right? Polynucleotide phosphorylase. So it helped to, this enzyme was helpful in polymerizing the RNA in a defined sequence, the sequence which we want to take in a template independent manner. So with the help of enzyme, RNA can be formed, right? There is no need for the template. There is no requirement for a, DNA sequence or the mRNA sequence for that. So in a template independent manner, it was possible. Now, we'll discuss about Hawley. So, Hawley, he discovered tRNA. We'll discuss it. So, Hawley, Khorana, and even Nirenberg, right? You know that. See, Khorana, he synthesized the polymers or homopolymers. Then, in the case of Nirenberg, Marshall Nirenberg, cell-free system for the protein synthesis, and even Hawley. So, in the NCRT, they didn't mention this Hawley part, but we will discuss it in tRNA. So, Hawley, Khorana, and Nirenberg. Hawley discovered this tRNA structure. They jointly got the Nobel Prize in Physiology and the Medicine in 1968 for what? For their interpretation of the genetic code and its function in the protein synthesis. Is that clear? Yes, everybody. Is that clear? Quick. Sure? So, Robert Hawley isolated tRNA, worked on tRNA and explained how they facilitate the translation. It was his contribution. Am I clear? Yes, everyone. Am I clear? Okay. So let's talk about the properties of the genetic code. But before that, let's learn how to make that. Right? How to make that. So everybody, kindly pay attention because I'll teach you the tricks to remember this as well. Okay. Done? So you can arrest gangster. That's the trick that you have to remember. What's the trick? The trick is you can arrest gangster. This is the first trick that you have to remember. So for making this particular table, in this order only, you have to write it. Okay, so you can arrest gangster. That's what you have to mention. So you remember one more thing that you have to consider here. See, this is the mRNA, right? When we know that synthesis is in 5' to 3' direction, right? Synthesis is in 5' to 3' direction. So same way when you study the mRNA, do you know that how do we study? How do we read it? Again, in the 5' to 3' direction. What does it mean? If this is the mRNA, we'll consider first 3 bases, then next 3, then next 3, then next 3. Means there should be no overlapping, right? No punctuation, nothing at all, no comma, nothing at all. And when you see this, we know that it is triplet. So that's how it proceeds. So this will give us the information of one amino acid, the second, the third, the fourth, like this. Are you getting my point? But while making this table, what you have to remember, you have to write the bases in this way only. You can arrest gangster. So this side is showing 5' and the first base actually. This side will tell us about the second base. Again, here in same sequence, you have to write you can arrest gangster. This is the third base. And here again, the same sequence, UCAG, that is you can arrest the gangster. Okay, but here, how you need to write? But here, not like this. Here we have to write it in this way. UCAG, 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 UCAG. So it should be UCAG only because if you will change it, right, if you will write it UACG, then it will be incorrect. So up to this part, all clear? Tell me quickly. Done? Sure? So UCAG. So see, the first base is U here. The second base is also U here. And In the last, UCAG. Done. Then, first base is C here. Second base is U here. UCAG. Then, first base A, second is U, then AU. 4 times write AU, then UCAG. So, GU, 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 UCAG. That's how you can make it. One more example. Like see here. What will be the first base? The first base will be C. Second base C. Then UCAG. First base U. Second is C. UCAG. First base A. Second is C. GC. UCAG. Now come to this part. First base U, second UA. See this. Second base is UA. So UCAG. That's how you can make this complete table. One last example. So here what should be the first base, guys? A. Second base G. So first two bases AG, AG, AG. Last UCAG. So that's how we make it. Okay. That's how we make it. Is that clear? So for this part, the trick is very simple. You'll remember it like this. Sir, pro, thar, ala. What you have to repeat? I'm just picking up the initials here. Sir, pro, thar, ala. Sir, pro, thar, ala. That's what you have to remember. Sir, pro, thar, ala. Sir, pro, thar, ala. So it is serine. It is proline. It is threonine. And what's that? It is alanine. So see, you know this, right? Sir, pro, thar, ala. Then here, you can remember, this is phenylalanine. Fill, E, ill, leaf in my pocket. For this, fill, leaf in my pocket. So 2 to 3 times, if you will make it, you will be able to remember that. That's C. This is from the book. So fill, leaf in. Acha, fill. I just picked up. Okay, fill, leaf in my pocket. Fill, L. L you have to remember. So fill, leaf in my pocket. So try to remember these tricks for 2 columns itself. So fill, leaf in my pocket. In my. So now it's indicating that I need a cup of tea. In my wallet. Pocket nahi, wallet. Fill, leaf in my wallet. Phi will tell me about phenylalanine. Leucine. In, isoleucine. From I you will have isoleucine. My, methionine. Very important here, this methionine, because it is the start codon. Wallet, valine. So isoleucine, valine, very easy to remember. Here, sir, pro, thar, ala. Sir, pro, thar, ala. Serine, proline, threonine, alanine. So if you are able to remember these two, then for the rest of it, I have the tricks again. So you can see that fill, right? First two, phenylalanine. See, leucine. 1, 2, 3, 4, 5, 6. Then isoleucine 3. Methionine 1. This is valine. Right? Valine. Sir, pro, thar, ala. These two, tyrosine. Then see, the 3 stop codon. I'll come to this. Histidine, glutamin, asparagine, again lysine. So, and aspartic acid, glutamic acid, see, serine. So 4 here, but 2 here. Same for the arginine. Right? Same for the arginine. So that's what we have to remember. And how can we go for it? I have mentioned it in my notes as well. Fill, leaf in my wallet. So you can remember this. Fill, leaf in my wallet. So methionine. So it is methionine. So this methionine is the initiator, or I would say, what's that? It is the start codon. It is the initiator as well as the start codon. Done. Sir, pro, thar, ala, serine, proline, threonine, alanine. Column 3 is having tyrosine, stop his gun, Asia like aspartic glue. Tricks are little weird, but they help. Tyrosine stop his gun. Asia like aspartame glue. So tyrosine, stop codon, histidine, gun. GN is there. Glutamin. It will be Asia, ASN, asparagine, like lysine, aspartic. So that's why I have mentioned aspartic here so that you don't get confused with the asparagine. Then glue, glutamic acid. Right, glutamic acid. Then for column 4, the trick is sister stop troubling arginine. Right, sister, cis, cysteine, troubling, stop codon, then troubling, tryptophan, arginine, ARG. Sister stop troubling arginine. Sir, arginine love glycine. Right, sister stop troubling arginine because arginine love glycine. So that's how, if you practice them, you'll be able to remember. I told you how to make it and I even shared the trick. So fill leaf in my wallet, sir pro thar Allah. Right, so tyrosine stop his, right, tyrosine stop his gun. Asia like aspartic glue. And then sister stop troubling arginine. Sir, arginine is in love with glycine. That's how you can remember. So give it a try. Give it a try. You'll be able to remember this. Now we will discuss the most important thing, that is the properties. So already told you that there will be triplet base and this genetic code now, it is universal. It is what? It is universal. In the bacteria also it will, the same codon will code for same amino acid. In humans also, in viruses also. So it is universal, right? Then it is triplet. No overlapping, no punctuation. So basically it is commaless. Then there are few exceptions. After that, see, there are two main properties which you have to understand and the one that you have to remember. So if you are preparing for the boards, then you definitely get question from this part. So unambiguous or non-ambiguous nature of genetic code. Unambiguous or non-ambiguous nature of genetic code. And the another important property here is degeneracy of genetic code. The another important property here is degeneracy of genetic code. So I have seen students getting confused here. So unambiguous nature. Ambiguity means something which is doubtful, but it is non-ambiguous, unambiguous. So while reading the question, do not be like that if it ambiguous is written and you just click it. It is non-ambiguous or unambiguous. What does it mean? It means that one codon will always code for same, or I would say a particular amino acid. Like for an example, your UUU will always code for phenylalanine. Are you getting my point? PHE. It will always code for phenylalanine. So if you pick, right, if you pick this, then CU will always code for serine. Okay? Always code for serine. Are you getting my point? Then will always code for proline. This is what I'm going to tell you, that this is the unambiguous nature, non-ambiguous nature. Now, what is the meaning of degeneracy? Here it says that one amino acid can be coded by more than one codon. Right? One amino acid can be coded by more than one codon. That's what I'm trying to tell you here. One amino acid can be coded by more than one codon. So the simplest example that we have just read, phenylalanine. Then you have other examples also. Jaise, see, so it codes for proline. Can you see that? It codes for what? It codes for proline. So more than one amino acid can code, yeah, one amino acid can be coded by more than one codon. Yeah, more than one codon can code, more than one codon can code same amino acid. So that's what you have to understand. That's what you need to differentiate here. So unambiguous nature, it says that no matter, right? No matter what, if this codon is coding for phenylalanine, it will always code for phenylalanine. Degeneracy says that more than one codon can code for same amino acid, or one amino acid can be coded by more than one codon. In any way you can remember this. This is the degeneracy. And why degeneracy occur? Why degeneracy? So students, degeneracy occurs due to the third base, which is also referred to as wobble base, right? So it is one wobble hypothesis, which is again propounded by Crick itself, right? As per this, right, the first two bases will decide the amino acid. First two bases, they show Watson and Crick base pairing, like C will form bond with G and A will form bond with T या फिर U, right? Or या फिर U, right? But when it comes to the third base, it is the third base which is the vowel base, right students? It is the third base which is the vowel base, and first two bases are going to decide the, right? First two bases are going to decide what? First two bases are going to decide the amino acid. This is the vowel base. So this degeneracy is due to this vowel base. Is that clear? This third base, that base at the 3' end, right, which shows this non-Watson and Crick base pairing, right? Third base is kind of can pair with anything. So this is what we have to remember. Is that clear? So who gave this vowel hypothesis? As I said, it is propounded by Francis Crick. Who gave this? It is given by Francis Crick. Is that clear? So first two bases will strictly follow the Watson and Crick base pairing, and the third base, okay, and the third base, the codon at the third, the third base, it can pair loosely with the first base of anticodon. Is that clear? So there is a flexibility here. That is why even we have 20 amino acid, 20 tRNAs, but still all the codons are studied for that amino acid. Is that clear? Right? So for studying the 61, because of this, right? Because of this, so third base is vowel base, right? It doesn't attach very tightly with that, you know, anticodon. So due to this, 20 tRNAs help to study 61 codons, because actually there are 61 codons for 20 amino acid, and tRNAs are also 20. So first two bases, right? First two bases are strictly going to show, what? Watson and Crick pairing. But when it is the third base, right, that third base will loosely join. Is that clear? Is that clear? Okay. Right? Okay. Done? So I will just tell you this when I'll discuss the tRNA structure, because then I will show you codon and anticodon. So this vowel base, it is the third base and it is at 3' end, right? But this vowel base on tRNA, it will be at 5' end. So we will discuss it then. Okay, so I believe this property is clear. So there are exceptions as well. Matlab, there is a special case also, right? There is a special case also that I would like to mention. Jaise AUG is the start codon, I told you already. But sometimes you know that AUG codes for methionine, but sometimes, you know, GUG can also codes for methionine if it is at the first position. So we will discuss that. So see, the salient features is genetic code is triplet in nature, means it is having 3 adjacent nitrogenous bases that will specify one amino acid in a polypeptide. Matlab, it is having 3 adjacent nitrogenous bases that will give one amino acid. Example, codon is triplet. So this is the thing that they have given. Just in mRNA with 99 bases give information for 33 amino acid. So 99 bases divided by 3, 33 amino acid. Same. If let's suppose you have 999 bases, you can divide it with 3. So that will give you 333 amino acid. Are you getting my point? So that's how you can check it. It is universal. codon will specify same amino acid in all organisms from viruses to human beings. Few exceptions are there. Then it is commaless and non-overlapping. First three, next three, next three, next three. That's how we have to proceed. So which one is the initiator codon? Again, you can just highlight this part. Initiator codon or the start codon. Initiator codon or the start codon. So this is AUG. Done. And when it comes to the stop codon, which will stop the translation, which is also known as nonsense codon, right? Then here you will have, that's how you can write. So it will be UUU AG, AAG, AGA. That's how you can remember. UUU, matlab you have to write this in this format. UUU AG, AGA. So this UAA, UAG, UGA. So UGA is opal, this one is amber, and this one is ochre. So these are the three stop codons, and this is amber, ochre, opal. Important, it's very important. Is that clear? So UAA, ochre, UAG, amber, UGA, opal. So stop codon, nonsense codon, initiator or start codon. That's what you have to remember. Got that? That's what you have to remember. So here itself you can write only for methionine and tryptophan. Right? Matlab, 2 amino acids. These 2 amino acids are coded by only one codon. Yeah, these 2 amino acids are having only one codon. Jaise AUG is for the methionine. Do mark it, beta. Right? AUG is for the methionine and UGG is for the tryptophan. So rest all other amino acids are having more than one codon. Okay, so only these 2 are having one codon, rest all are having more than one codon. Is that clear? Sure? So initiator and terminator codon clear. So as I said, this is something that I have mentioned earlier. So AUG is the start codon and rarely it is GUG. And this GUG will code for the start codon if it is at the first position. So GUG codes for valine. So mainly, who's the initiator codon? It is AUG. In the NCRT or even in the PYQ, it is mentioned that, yeah, even in the NCRT and in the PYQs, it is mentioned that which amino acid is having the, which codon is having the dual function. So AUG codes for methionine and AUG is also the start codon. Okay. Okay. So it is having dual function, codes for methionine, also act as initiator. That's a PYQ. So AUG codes for methionine in eukaryotes and formyl methionine in prokaryotes. Another important point. So no need to make the like notes, even if you print it out and then from that if you extract the short notes, more than sufficient. So sometimes GUG function as start codon because it codes for valine, but if it is at start position, it will code for methionine. So it's a kind of special case. So obviously it is showing the ambiguity here, but still it is believed that it is the special case only, right? So terminator codon, stop codon, nonsense codon, as I said, UUU, AG, AGA. Ochre, amber, opal. This is important. So there are only 61 codon that will code for, right? There are only 61 codon that will code for amino acid. Rest 3 are the stop codons. Okay? Right? So non-ambiguous nature, one codon specify only one amino acid and not any other. So here we discuss the GUG, right? That it normally forms valine, but if it is at the start position, it can code for methionine. And degeneracy, you know that more than one codon for same amino acid. So serine, 6 codon, leucine, arginine, right? So they have 6 codons. So only tryptophan and methionine are specified by single codon only, right, students? So again, we have also discussed that degeneracy is due to the last base in codon, and that last base is at 3' end. It is the wobble base and it show wobbling, that is loosely attached to anticodon. anticodon. But first two nitrogenous bases are important, right? They strictly show Watson and Crick pairing. Watson-Crick base pairing. Understood? So this is the vowel hypothesis. Am I clear? This is the vowel hypothesis. Understood? So there are certain exceptions. So different codons, as in paramecium and some other ciliates, terminator codons can be UA and UGA. The terminator codons can code for glutamine. In mitochondrial genes, AGG and AGA codes for arginine but function as stop signal in human mitochondria. Normally they codes for arginine, but here they work like stop codon. So UGA, which is a terminated codon, it codes for tryptophan, while UA code for isoleucine. So these are few exceptions. If you can remember that, very good. If you cannot, then again it's all right. So the next topic, or I would say not the last वैसे, because after this the operin and HGP. So I'm repeating this again. DNA fingerprinting will be discussed in biotech, okay, because electrophoresis and all will be discussed by then. Done? Any doubts? Any doubts here? So those who are preparing for the CBSE boards, let me tell you, genetic code is one of the most important topics. Okay? Genetic code is one of the most important topics. Great. So next topic is the translation, but before translation, let's talk about the different types of RNAs. Okay. So mainly we have to discuss about the tRNA, because already I told you about the mRNA, and let's discuss mRNA, rRNA, and then tRNA. Okay. Done. So again, you are going to find it very easy, right? You're going to find it very easy. So I'll start with a quick revision. See, when you talk about the different types of RNAs, what are we discussing here? We are discussing different types of RNA, right? Different types of RNA. That's our topic. Yes, students, that's our topic. Tell me quickly. We have to talk about what? We have to talk about the different types of RNA. So that's our topic. So let's discuss that. So when you discuss about the different types of RNA, you know that RNA is also the genetic material. You know the changes. So we will discuss about the genomic RNA or the genetic RNA. And you know it very well. Where do you see this? Right? When RNA is working like the genetic material. So in RNA virus, RNA is the genetic material. So students, if it is, so then it will be the genomic RNA, because here, which role is it playing? It is basically, yes please. So it is basically doing what? It is basically acting like a genetic material, isn't it? Sure? Okay. Done. So what sort of viruses they can be? Just say reovirus. So reovirus is having double-stranded RNA. Retrovirus mein, what do you discuss? Single-stranded RNA. So these are the RNA viruses. So here RNA is acting like what? RNA is acting like the genetic material. Am I clear? RNA is acting like the genetic material. Am I clear? Now, we have the non-genomic, or I would say non-genetic RNA as well, which will play the other roles. Okay? So non-genetic RNA. So here you will talk about mRNA stands for messenger RNA, transfer RNA, ribosomal RNA, isn't it? These are the three types that we have. So when you talk about the messenger RNA, that is mRNA, you know it very well. It is the least stable, already discussed with you all, form 1 to 5% of cells total RNA, right? Forms directly from DNA and then helps in making protein and then provide information for making protein. Done? And then provide information for making protein. Am I clear? I hope it is. Then the second category that we have is ribosomal RNA. So you know that. So this is the most stable, comma, make around 80% of cells total RNA, form ribosome with some proteins, Am I clear? Am I clear? So it is going to form the ribosome with protein. So 80% most stable form of RNA. Am I clear? Sure. So what's the next part here? See students, in prokaryotes, you know that which ribosome do we have? 70S. So larger subunit, you should know about this because we have to discuss it in NCERT. So 50S and 30S subunits are there. So this 50S is the larger subunit. This one is the smaller subunit. Both the subunits join only at the time of protein synthesis. So this is made up of 20S ribosomal RNA, 5S ribosomal RNA, and this one is made up of 16S ribosomal RNA. Write down. It is important. Quick. Yes, please. Done? Sure? So 23S, 5S ribosomal RNA and 16S. So this one is made up of approximately 34 proteins. This one 15 proteins. 21 proteins, because total here is, see, total here will be 34 plus 21. Obviously, it is going to be 55 proteins. So that's what you have to remember. Am I clear? Am I clear? This is important. Kindly write down. Yes, please. Kindly write down. Done? So, in eukaryotes, you know that in the cytoplasm, yes, one more point I would like to mention. It is 80S, and you know that mitochondria and the chloroplast, they contain what? They contain 70S. They contain what? They contain 70S, right? Subunit. And even on the endoplasmic reticulum, the rough endoplasmic reticulum is having 80. So larger subunit is 60S. This one is 40. So this one is made up of 28S ribosomal RNA, 5.8S ribosomal RNA, 5S ribosomal RNA, and this one is made up of 18S ribosomal RNA. So these two, you know that RNA polymerase 1, this is synthesized by RNA polymerase 3, and this one is again synthesized by RNA polymerase 1. Do we need to remember that? Definitely. And here you'll see approximately 80 proteins, right? Right? Approximately 80 proteins will be there. Just say here approximately 49 proteins, or here somewhere around 33 proteins. So total will go 81 proteins. So it is a recent PYQ, and in that NTA's option 80 proteins was the correct answer, 79 or 80. So you can check it. It's a recent PYQ. Okay, it's a recent PYQ. So just do it quickly. So this information is important. Okay, this information is important. So magnesium ion done, students. So it is the magnesium ion that helps in the attachment of these two subunits. Am I clear? Sure. Is it sure? Clear? Okay. So before starting tRNA, one thing just skipped my mind. So let me discuss it here again. Sorry for this, but just 1 minute. So see, remember, in the case of, right, in transcription also, yes, in transcription also, obviously it is transcription, RNA bases are there. So you have that CTPs, GTPs, UTPs, ATPs. So just like DNA replication, they also serve dual purpose. Okay, they also act like substrate and also provide energy for polymerization, right? I just forgot it, so I'm including it. Also provide energy for polymerization. Okay, so I already explained it in the DNA application, because these are the triphosphates. Okay, so one phosphate, like monophosphates, are required in this polynucleotide chain. So rest two phosphates, they'll release the energy that will be used for the polymerization. So this was the point that I just forgot, and all of a sudden when I discuss this enzymes wala part from this mRNA thing, from this ribosomal RNA thing, then it came in my mind. So do remember this. Number of proteins are also important. That's a PYQ. And then it is the tRNA. So I already told you Holly discovered it. So let me explain it a bit. It is 5' end, it is 3' end, having free OH group. Okay, here, right, two-dimensional structure, 2D structure of tRNA. tRNA means transfer RNA, right? So tRNA means transfer RNA slash, it is also known as soluble RNA slash, it is also known as adapter RNA. Are you getting my point? You can call it as transfer RNA. You can call it as soluble RNA. We can also call it as, yes please, we can also call it as adapter RNA. And you know that RNA polymerase 3 will synthesize it. And moreover, it is the smallest RNA. It is the smallest RNA, and it makes 10 to 15% of cells total RNA. Are you getting my point? 10 to 15% of cells total RNA. Is that clear? Is that clear? Sure? Okay. So Holly, I told you about that scientist now, presented its 2D clover leaf-like structure. So it looks like a clover leaf. So clover leaf-like structure, and the diagram that I'm drawing there is 2D structure. When you talk about its 3D structure, so that 3D structure is what? It is inverted L shape. Both are PYQs. Are you getting my point? Both are PYQs. Now, look at the diagram. See, here 3' end will have OH and mostly 5' end. This base here is guanine, mostly. Now, when you look at this side is known as amino acid acceptor arm. First, you remember these few points, then it is known as amino acid acceptor arm. Means amino acid has to join there. tRNA is an adapter. Why? Because this tRNA, as per the codon on the mRNA, will bring the charged amino acids from the cytoplasm and will help in the protein synthesis. That's why we are calling it as the adapter RNA. So this is the amino acid acceptor arm. Opposite to this is the anticodon arm. And this rounded part, it is referred as anticodon loop. What do you call it? Anticodon loop. Now students, when you see this part, this is DHU loop. Which loop it is? DHU. DHU stands for, yes, you have to remember, dihydrouridine, an unusual base here. So it is the dihydrouridine. And this one is T, psi, C. So T here, you must be thinking, ma'am, in the ribosome, in the RNA, we don't have T. So this is actually ribothiamidine. It is referred as ribothiamidine. And what is this psi? Psi means pseudouridine. It's the pseudouridine. So this is what you have to remember, right? And this structure is variable arm. Its function is not so clear, but this arm helps to classify tRNA, helps to classify tRNA into two classes, that is class 1, class 2. Like in two categories, we can put this tRNA on the basis of that. Okay. Okay. So arms, these arms are formed. See, these are the arms now. So here you see complementary base pairing. So arms are formed when complementary base pairing is there. Okay. Arms are formed when complementary base pairing is there. And in the case of loop, no base pairing, no complementary base pairing, single bases are there. So where you'll see the single bases, you'll see the loop. Otherwise, you'll see what? You'll see arm. So it is the two-dimensional structure, right? It is the two-dimensional structure. And amino acid acceptor arm, it will join here with 3' end. Opposite to it, this is the anticodon arm, which will join with the mRNA. Dihydrouridine loop, it will join with the enzyme. T psi C will join with the larger subunit of the ribosome. So yes, the structure of tRNA should be very clear. And yes, here you write it. So it is, there is a sequence here that we always see, that is CCA. That is CCA. So now that's what I'll discuss. Okay. So you look at this side. Do not get confused. So now in this diagram, they have formed the variable arm towards this side, है ना? Right? This side is showing the variable arm. So obviously here you will see this is going to be the T psi loop, psi C loop. This is the DHU. So just consider this part here, okay? Just consider this part here. So once again, five' end having guanine and free phosphate. This is 3' end free OH. And here you always see a sequence which you call at CCA. Why am I calling it as CCA? See, first of all, chalo, one by one I'll start. I said that this is the amino acid acceptor arm. That's what I just said. That's what I just said, amino acid acceptor arm. So students, right, this is the homework for the translation. Jesse, this is the basic structure of an amino acid. Here it can be H, it can be R. So sorry, here it is H only, and here it can be R or it can be H in the case of glycine. So this is the basic structure of amino acid. So this amino acid, right, this is the basic structure of amino acid. So this amino acid will, with its, right, this amino acid with its COOH group joins the tRNA joins the tRNA at 3-OH, at 3' end. That's how it is going to be. So amino acylation of tRNA it is going to be, right, amino acyl bond will form here. So it is a COOH group, right? So amino acid with its COOH group joins the tRNA at this 3' end, which is having the OH. This is the first point that you have to remember. That's why it is amino acid acceptor arm. Now, see, 5' to 3', I told you this is going to be the reading direction. So that's why here we are calling it as CCA. So what are you going to see? You'll see that in tRNA, we find, in tRNA, what do we find? We find a sequence CCA, right? So you can even remember it as ACC, but actually the reading frame is 5' to 3', so we call it as CCA. What do we call it? CCA. Now, opposite to this, right, this part is what? This is the anticodon arm, and then this is the anticodon loop. That's what I told you. So anticodon arm, or this part is the loop. So this side, it joins with what? It joins with mRNA. So it is having the code, it is having anticodon of mRNA. What does it contain? It contains anticodon of mRNA. So I'll give you the example. So you take it in this way. Just take it in this way. This is your mRNA, 5' to 3' end, 5' to 3' end. So just look at it. Here, let's suppose you have AUG, है ना? AUG. So you know that complementary to it will be UAC. So this is 5' end, this is 3' end. Okay, just keep it in your mind. So that's how you are going to write UAC. I know you will be confused. Remember, 5' end will join with the at 3' end. This one will join here. This one will join here. Let me explain it. Just say, let's suppose this is your mRNA, 5' to 3'. Okay? This is your mRNA. Now, if I consider tRNA as a straight line, just say this is 5', this is 3'. So you know that that's not going to happen. That here, if it is AUG, so here you will get UAC. 5' will not join with 5'. A will join here. A complementary to U, U complementary to A, G complementary to C. That's how it is. So you can say if this is, let's suppose this is 1, 2 and 3. Here 1, 2 and 3. That's how it is. 1 will join with 1. A will go with U, U will go with A, G will go with C. So that is how it is. Is that clear? Or in another way, how can you write it? This is your mRNA, AUG. This is 5' and 3' end. Okay, so that's the tRNA. This is 3' and 5' end. So it is UAC. Done? UAC. So see, that's how you can write. Okay, so codon and anticodon. So that is what I was talking. That see, if you talk about the mRNA, first 2 bases, can you see that? These first 2 bases, they strictly show Watson-Crick base pairing. Are you getting my point? They strictly show Watson and Crick base pairing, but this base, this is the vowel base. So this will be loosely fixed. It will not show that Watson and Crick base pairing. So on the codon, because mRNA contains codon, tRNA contains anticodon. Are you getting my point? tRNA contains anticodon. This one contain codon, this one contain anticodon. So this 3' base at the codon, and this is the 5' end of the 5' base at anticodon. They show the vowel base. Jesse, there is one base inosine that can join with uracil as well as A. It can join with both. Imagine. So this is what the vowel hypothesis I was talking about by then. Okay. Chaliye. So next, this is your dihydrouridine loop. So how do I remember? D for down. I'm feeling drained. We need charging, right? We need energy, E, energy charging, enzyme. So remember, this will join with enzyme. So what's the name of enzyme? It is amino acyl tRNA synthetase. It is amino acyl tRNA synthetase enzyme. So we have 20 amino acid, 20 amino acyl tRNA synthetases, right? So each amino acid is having its unique, right? It's unique. What? It's unique amino acid. Done? Chaliye. So we are left with this T-Cy-C, pseudouridine. So it will bind with larger subunit. It binds with larger subunit. So this is the role of all the enzymes and CCA. Remember that. Understood? So if there is any doubt, do let me know. Okay? Sure? Chaliye. So if this is clear, then the next part, right, which is translation, you'll also be able to understand that. So see this, right? The bond here. So amino acid with its COH group binds here at 3' end. At 3' end, this 3' end. Okay, so let's go for the translation. So translation means the protein synthesis. Yes. So what's the time? Okay. So let's talk about the translation. So you know that whether it is prokaryote or eukaryote, the translation will occur in cytoplasm, right? In both prokaryote and eukaryote. The site is what? The site is cytoplasm. Now, what is the requirement? What exactly do we need for this? Right? We need ions, ATPs, done. But you need mRNA. You need tRNA. We need amino acid. We need enzyme. Right? 20 different enzymes for 20 different RNAs. And this is amino acyl tRNA synthetase. What's that? This is amino acyl tRNA synthetase. That's the name of the enzyme. Am I clear? It is amino acyl tRNA synthetase. Is that clear? Sure. Is that clear? So it is amino acyl tRNA synthetase. Now students, for the translation, what is required? The first step, the very first step, that is again the PYQ, it is the charging of amino acid. Okay, it is the charging of amino acid. This is the very first step. We know that the initiator codon is methionine. It is formyl methionine in prokaryotes and simple non-formylated methionine in eukaryotes. The very first step is in the amino acid. See, all that things are in the cytoplasm. Even your amino acid is in the cytoplasm. Now there is the requirement of ATP, right? Now, with the help of this enzyme, and this enzyme is unique for all the ATPs. Like it is, we have 20 amino acids, we have 20 these enzymes. So what is the name of the enzyme? The name of the enzyme is amino acyl tRNA synthetase. So with the help of this enzyme, this amino acid will get charged. How come this amino acid will get charged? You have 3 phosphates here. So amino acid with the AMP and this enzyme complex will form. Okay, that's the very first step of translation. Okay, so this is again a PYQ, that which exactly is the first step of translation. If we want the protein synthesis to start, the very first thing that has to occur is the charging of tRNA. What's that? It is the charging of amino acid. It is the charging of amino acid. So amino acid with the help of ATP and this amino acyl tRNA synthetase, right? So 2 phosphates will release. So AMP, AAAMP, amino acyl tRNA synthetase, which I'm simply referring as enzyme there. That's the name of the enzyme. So they will form the complex. They will form the complex. So if you have to see this, you can see it in this way, guys. Just say, let's suppose if this is believed to be the enzyme, let's suppose if this is the structure of your enzyme, so here you have your amino acid and your AMP. That's what I'm going to tell you, amino acid and AMP. Now, what's the next step? Because all the things in the written format is mentioned, you just note down the reaction. Second is the charging of tRNA, which is again loading of tRNA. Just here, this is the charging of amino acid, which is also known as activation of amino acid. Done. So now here what will happen, loading of tRNA, or you can also call it as amino acylation of tRNA. Done. So look at this part here. Let's say here you have your amino acid and AMP. Okay? Now this enzyme will join to the dihydrouridine site of tRNA. You know, na? This is the enzyme. So let's say this is a site of the enzyme. So this enzyme, what this enzyme will do? Okay. See this. This enzyme, can you see this enzyme? See, this enzyme, it is having the site, tRNA is having the site for the attachment of this enzyme. So see this. It is moving there, right? They are getting attached. This is fun, students. Trust me. See this. So they were separated, they joined. Now, what is the role of this enzyme? This enzyme will make sure that this amino acid should go there, right? Should join here. Its COH bond should join here. Actually, that is the function of this particular, enzyme. So your amino, you can also write it in this way, amino acyl AMP. Can you write that? This is amino acyl AMP enzyme complex. So amino acyl means it is your charged amino acid. You can also write it in this way, plus your tRNA. Now, this enzyme is like, just say, let's suppose your 2 friends are having a fight and you are like, I want my friends to have patch up. So you are that mediator. One friend, another friend. You convince this friend, you are bringing them closer. They are having the bond again and you are just out of it. It is something like that. So amino acyl AMP enzyme complex, so its own tRNA. Then, see, AMP and the enzyme will be removed. So it will become amino acyl tRNA complex. Like that, amino acid will go to this tRNA. Enzyme will be separated for the next amino acid and even the AMP also. So this is something that you can understand, that amino acid will come to this 3-A, this at this end. So this is the charging of tRNA. This is the charging of tRNA, and this tRNA is the charged tRNA. So once again, I am repeating it. When it comes to the initiator tRNA, so it is going to be for prokaryotes, my dear students, for prokaryotes, it is going to be formyl methionine, formilated methionine tRNA, and initiator code tRNA for eukaryotes is going to be simple methionine tRNA. It is not formilated. Am I clear? It is not formilated. Is that clear? Sure, it is not formilated. Understood? Now after this, your DNA is charged. Now obviously the initiation will start, right? And again, it varies in prokaryotes and eukaryotes. So prokaryotes mein for initiation, you people will see there are three, like ATPs, GTPs, all are required, right students? ATP, GTPs, all are required. So for initiation, you need IF1, IF2, IF3. So what are they? They are the initiation factors, right? They are the initiation factors which you need here, right? These are the initiation factors. They are nothing but they are the proteins only. IF1, IF2, IF3 is there. Am I clear? Am I clear? Right? And you know that we need 50S, 30S subunit, GTP is required, Mg2 positive is required, and these initiation factors are required. Am I clear? Am I clear? Now, understand this part, right? In eukaryotes, it is different. See, imagine, this is your mRNA, 5' to 3' end. This mRNA will bind with 30S subunit. Now, you know it very well that 30S subunit is having 16S ribosomal RNA. I believe you remember that. I believe you remember that. Right. So, here, what's the keyword? That this mRNA is having what? It is having a sequence called Shine-Delgarno sequence, which is also known as, in simple words, SD sequence at its 5' end. Done? Because if they have to attach, there should be something complementary to each other. In 16S ribosomal RNA of 30S subunit, right? What will you see? Here, at 3' end, entire Shine-Delgarno sequence is present, which is complementary to SD sequence. Are you getting my point? So that's how they bind here. That's how they bind here. Is that clear? So mRNA and the smaller subunit, this is what they have. Okay. In eukaryotes, the subunit is 40S, having 18S ribosomal RNA, but that 18S ribosomal RNA is having complementary sequence to the 7-methylguanosine triphosphate. Okay. So here, that's how it works. And obviously in eukaryotes, there are other different things also. So you can see, this is your mRNA, 5' 3' end. Let's say here, your 30S ribosomal subunit is getting attached. Now you know how. Now guys, your charged tRNA will come. Who will come? Your charged tRNA, and you know that it is having the methionine, formyl methionine, I would say, right? What does it contains? It is having the formyl methionine. Is that clear? Sure. So this step, right? This step, last one, this step, it requires initiation factor 3, right? It requires magnesium ion. Now when you move to this step requires what? This step requires GTP, initiation factor 2, right? GTP and initiation factor 2 and even initiation factor 3 also play the role here. And of course, how can we forget? This binding cannot be without magnesium ion. So that's how it comes. Okay? This is the smaller subunit. Now students, when you talk about the larger subunit, it is having 3 sites: E site, P site and A site. How many sites do we have in larger subunit? 3. And the smaller subunit is somewhere like this, right? So remember, E stands for the exit site here. Kindly pay attention. Then I'll explain it again. P stands for the peptidal site here. A stands for the amino acyl site here. Am I clear? So exit site, peptidal site, amino acyl site. Exit site, peptidal site, amino acyl site. I'm repeating this again. Exit site, peptidal site, amino acyl site it is. Is that clear? Now you can see this. Exit site, peptidal site or amino acyl site it is. See, here there is one more groove as well in the larger subunit. This is one more groove as well. This groove protects the polypeptide chain. Okay, I hope you are able to visualize. I'll send some animations in our official Telegram group as well. So it protects the polypeptide chain and provide it a passage. So through this groove, it will just go out. This part is clear. So E for exit, P for peptidal, A for amino acyl site in the larger subunit. Now in prokaryotes, what happened? In prokaryotes, I just told you that first of all, there is 30S subunit, 50 and 30S. S stands for Swedberg constant. It is the sedimentation coefficient. 1S is equals to 10 raised to power minus 13 seconds. It is telling us about what? Right, about the settlement of the particle. Sedimentation coefficient it is, right? So 1S is equals to 10 raised to power minus 13 seconds. So this is the 16S ribosomal RNA. This is 30S. This is mRNA. So mRNA is having Shine-Dalgarno. 16S is having anti-Shine-Dalgarno. They'll join. IF3 and magnesium ion is playing the role here. Then the charged tRNA will come. Now after this, the larger subunit will come. So obviously magnesium ion is required here. So larger subunit has come and it is in such a way that this charged tRNA, do you know that? It is in such a way that this charged tRNA, it comes at P site. So basically when larger and smaller subunit they join, they are forming this P site. So this is E site, this is A site. So what we can conclude here that first amino acid come to, first charged tRNA, come to P site, that is peptidal site, right? Peptidal site, initiation. This is initiation, right? So as per the codon on mRNA, next tRNA, right now I'm not talking about the eukaryotes, at the end I will discuss it. So as per the codon on mRNA, next charged tRNA will come to A site. Am I clear here? Sure. A site. Now you just listen to me and I'll tell you how will it work. So this is the 5' end, this is the 3' end. That's how the subunit will come and will bind here. Am I clear? Am I clear? Everything is written in the notes. You just understand it. Okay. Now, when this happen, you know, this methionine at the P site, right? This formyl methionine at the P site. Okay, what will happen to that? Now we have entered in the elongation part because we have the charged tRNA at A site, right? Yes? Now what's going to happen? See, this is the formyl methionine here. When this thing will occur, what will happen? The bond between COOH group of amino acid at P site breaks, है ना, and it gets free. This will, when this, and it gets free. And it gets what? It gets free. Am I clear? And it gets free. Yes? Done? Now, see, if this group, it will break the bond with this tRNA, matlab this amino acid will be free, right? This amino acid gets free. Now, amino acid at P site, which is free now, है ना? Which is free now, will form bond with the amino acid present at A site tRNA. Obviously, that's the elongation that we are discussing. That's what? That's the elongation that we are discussing. Am I clear? That's the elongation that we are discussing. Right? Because here also you have the amino acid. So who's going to catalyze this? Before telling you who's going to catalyze this, these are the elongation factors that will be used in the prokaryotes there. Right? About eukaryotes, I'll discuss the differences later. So I'm just focused on the prokaryotes right now. Okay? These are the EF, means elongation factors. Am I clear? This means elongation factors. Am I clear? Now, this peptide bond, who's going to make this peptide bond? This peptide bond is formed by 23S ribosomal RNA present in the larger subunit, of course, called peptidyl transferase. Matlab this is going to catalyze this bond formation, and you know that here RNA act as an enzyme. So obviously, so it's ribozyme. So it is the ribozyme that will catalyze this bond formation here. So what is going to happen, beta? Your mRNA and the smaller subunit will join by the Shine-Dalgarno, anti-Shine-Dalgarno sequence. Charged tRNA will come. Then the larger subunit will come. It will be at the P site. It is having the amino acid. This is the initiation. Then comes the elongation. Elongation factors and GTPs are going to play the role here. So the next tRNA will come at the A site. When the next tRNA, the charged tRNA comes at the A site, as per the sequence on this mRNA, right, this amino acid present at the tRNA of P site, it breaks the contact with its COH group will be free. So it will form bond here with this amino acid. And who's going to catalyze this bond? 23S ribosomal RNA, which is the peptidyl transferase enzyme, which is the ribozyme there, right? Now, after this, what is going to happen? After this, what is going to happen? So obviously, the tRNA at P site, it is free, and this tRNA at A site is becoming dipeptide. Yes or no? Yes or no? So here comes the role of another enzyme. But tell me, this first part is clear? This first part is clear? Sure? Any doubt here? Yes, please. Okay, done. Now there is the role of elongation factors are also helping. Now here comes the role of translocase. This enzyme, by using GTP, slide the ribosome. It will slide the ribosome in 5' to 3' direction. Guys, it's not the mRNA that is moving. It is the ribosome that is sliding. Right? Just say, let's suppose I was at this place. When I was at this place, in front of me door was there. I slide a bit. Now, in front of me, I have the camera. Are you getting my point? Are you getting my point? So that's how it is. So when there is the sliding, what's going to happen? You can see that see this. Due to the sliding, and sliding will always be in 5' to 3' direction. So this is E, this is P, this is A. So when there is the sliding, the amino acid which was at P, the tRNA which was at P site will come to E site, and one which was at A site will come to P site. And now you know that the next site is again free. Now the next charged tRNA will come. Then again the process will repeat. The amino acid will keep on adding, and that chain, that polypeptide, growing polypeptide chain will come out by the groove. So when this tRNA come to E site, from E site it will just take the exit. So that's how the ribosome will slide with the help of translocase. It needs the GTP. It will keep on sliding. So once it approaches any of the stop codon, just say, let's suppose here you have UAA. This is the stop codon that you have here. So once it approaches the stop codon, then with the help of some release factors or the dissociation factor, it will separate. The ribosomal subunit will dissociate. So that is actually the process that is helping. That is how these sites are also moving. So I know some of you grasped it, but some of you are still confused. So don't worry. Now we will have one more revision of it from my notes so that you don't get worried. So see, right, these diagrams that I have made, they'll clarify all your topic. So that's how it works. This is what you have to understand, right? And now there are few points which I would like to highlight regarding eukaryotes. In eukaryotes, you do not have no elongation factor like TU, TSG, but right, other factors are there, obviously, other elongation factors. Secondly, in eukaryotes, the charged tRNA first binds with (40S) subunit. Yes, you heard me right. Here it is different. It will bind with 40S subunit and form pre-initiation complex. Done. Then it binds to mRNA, and that at 5' cap due to this 7-methyl guanosine triphosphate. And then this will slide over this mRNA, this pre-initiation complex. And finally, guys, there is a Kozak sequence in which start codon is there. There it will move. Wait, let me show you the diagram first. So see this. In the case of there is the eukaryotes pre-initiation complex form. What forms? Pre-initiation complex. Just your smaller subunit and your initiator or the start tRNA or charged tRNA will join. Then students at 5' cap, then they will join. Some cap binding complexes are there. Then they join with the mRNA. Just in prokaryotes, mRNA and smaller subunit, then tRNA. But here, tRNA, smaller subunit, then mRNA. So it will scan. This pre-initiation complex will scan the mRNA. In this Kozak sequence or consensus, there is the presence of the start codon from where the the translation will start. Am I clear? And here you can see at 5', 3' end, this is the one polypeptide amino acid now. So it will break the bond here, and then it will move to the tRNA, which is at the A side. So that's how it keeps on elongated. So I have added many such amazing diagrams here. So this is the difference. So obviously the factors also vary. So let's discuss it from the notes itself, and that will be like a quick revision for you, and then we are going to pick up our topic operon. So by 1 or even before 1 we will be free, and we have completed all the topics there, right students? We have completed all the topics there, right? Yeah, mutation, gene mutation, point mutation, a little bit of it we have. Mutation in the principles of inheritance we have covered, but a little bit of that we'll discuss. But by 1 a.m. we will revise it. So formation of protein from mRNA is the polypeptide synthesis. So the order and the sequence of amino acid, who will decide that? Obviously the sequence of bases on mRNA. So amino acid will join by a bond. This is the peptide bond. That's how it makes polypeptide. It is unidirectional, and where the protein will form, of course, ribosome. So we have already covered this larger and smaller subunit part. So larger subunit is having groove for the protection and passage of polypeptide. It is having site A, amino acyl site, right? And the enzyme peptidal transferase and a binding site for tRNA as well. So tRNA will also have the tRNA binding site. Enzyme peptidal transferase is also there. So smaller subunit is having point for attachment of mRNA. Along with the larger subunit, it will form the P site or the peptidal site. Then when we move ahead, this is the raw material, the amino acid, mRNA, tRNA, and amino acyl tRNA synthetase. So there are 20 amino acid, 20 specific tRNAs, and there are 20 specific enzymes as well. 20 specific enzymes as well. Understood? Then let's move to the next part. So as I said, the first part is the activation of amino acid or the charging of 1 a.m. 1 a.m. We'll finish it by 1 a.m. Okay. So activation of the amino acid. So there are 20 types of amino acid. I told you already. So in the presence of ATP, they will join with their related tRNA. So this is the reaction. Amino acid plus ATP, amino acyl tRNA synthetase. So amino acyl AMP enzyme complex will be there, and 2 pyrophosphates they will release. Then there comes the charging of tRNA, loading of tRNA, or the amino acylation of the tRNA. So here you know that specific activated amino acid recognized by its specific tRNA. So amino acid will attach to tRNA, and AMP and enzyme will be separated. Okay. Okay, so this amino acyl tRNA is the charged tRNA. Now they'll find their way to the protein. So there is a separate amino acyl tRNA synthetase for each kind of amino acid. So you can see COH group here will bind to this. This is something that you have to remember, okay? Okay, so this is the charged tRNA. Now, in the prokaryotes, we have just read initiation factor 1, 2, and 3 required. In eukaryote students, there are total 11 initiation factors. This is not IF1, IF2, IF3, but in eukaryotes, how many initiation factors are there? 11. How many initiation factors are there? 11. These are the subunits. So, GTP and IFS, they promote the initiation process. So, mRNA is having Shine-Dalgarno sequence. I told you at its 5' end. And 16S ribosomal RNA of the 30S subunit is having anti-Shine-Dalgarno sequence at its 3' end. It is also written. But in eukaryotes, you know that smaller subunit, right, of ribosome is recognized by the 7-methylguanosine cap. So they are complementary to each other. Is that clear? They are complementary to each other. So 30S subunit plus mRNA, IF3 magnesium ion we have discussed. So when this mRNA join with this formyl methionine tRNA complex, IF2, IF3 magnesium ion, and 1 GTP is required. Done. So larger subunit will come once you know this forms. Once this forms, then the larger subunit also comes. Okay. Okay. Done, students. Then let's move to the next part. There are 3 sites there. So starting codon will be at the P site. then the next codon will come at the A side. So next tRNA with new amino acid always attaches at the A side. So methionine is non-formylated in eukaryotes, formylated in prokaryotes, again important. So elongation mein you have EFT 2 plus G. That's how I remember. Okay, right? But in prokaryotes. So the link between amino acid of P site of tRNA is broken and the tRNA of P site is discharged. So COH of the P site of amino acid bache, that will become free. Right? Now it will form the bond with the amino acid at the A site. So peptidal transferase synthetase, 23S ribosomal RNA, which is again the ribozyme, that helps in the formation of this peptide bond. Right, students? So the ribosome will slide with the help of translocase by using the GTP. So the tRNA at P site will come to E site, the tRNA at A site will come to P site. From E, it will take exit and the next tRNA will come to A site. So that is how the elongation will continue. Ribosome will slide over in 5' to 3' direction. Right, student? So translocase enzyme will help there and EFG factor in prokaryotes will help there and GTP will provide the energy. So we have used the GTP 2 times when we have to make the starting wala complex and for the translocation, and 1 ATP we have used for the charging. Isn't it? Isn't it? So incorporation of an amino acid will need 1 ATP and 2 GTP molecules. See, that much energy is required for, right, that much energy is required for what? For putting these mRNAs. Yes, right, for what? For basically putting these amino acid there in that chain. Am I clear? Then termination ke liye 3 nonsense codon or stop codons are there, right? Stop codons are there. So linkage between last tRNA and the polypeptide chain is broken by 3 release factor. Release factor 1, 2, 3, and the GTP is required. So release factor 1 is for UAG and UAA. Release factor 2 for UAA, UGA. This is what you can see. So mRNA, we have already discussed its structure. This is 5' end, this is 3' end. So students, see, from start codon it will start. Let's suppose if it is the stop codon, it will stop. So always remember in the mRNA, just before the start codon, you will have UTRs. After the stop codon also, you will have UTRs. So they are the untranslated regions. They will help in the they increase the efficiency of the translation. So that is it. This is all about the topic. This is all about the translation. Now we will talk about the gene expression and we will also talk about some of the inhibitors. Right? So when you people talk about the inhibitors here, see, tetracycline, you might have heard about it. This is the antibiotic. So how will it act? So it inhibits the binding of this charged tRNA to the ribosome. How the streptomycin will work? It will inhibit the initiation of translation and cause misreading. How erythromycin will work? It inhibits the translocation of ribosome along the mRNA. So if there is any doubt, please ask me. Then let's go. Done? Sure? Sure? Great. So regulation of gene expression, right? Operant concept we have to start that we only study in the case of prokaryotes. But before that, there are some other points which we need to discuss, right? So this concept is not lengthy. After this, what will we discuss? HGP. So yes, by 1 AM, we will be able to finish it. And we have covered all the topics. DNA fingerprinting will be discussed in the biotechnology part. Okay? Okay. So if I say that if the chapter will go for 11 hours, right? And this chapter, it's worth giving time. So 1.5 hours is for the break only. So you can simply say, yeah, you are just having 9.5 hours for this important chapter. 20 marks. This is important. So regulation of gene expression. So how a gene is going to express itself? By making proteins. We know it very well. In the case of eukaryotes, if you want to regulate the gene expression, there are multiple levels. Transcription, translation is different. RNA processing is also going on. So there are multiple steps where you can alter that. See, the mechanism which stimulates the expression of certain genes and inhibit the other is the regulation of gene expression, number one. Right, it occurs at various level in eukaryotes, right? So this expression results in formation of polypeptide, and as I said, in eukaryotes this can be stopped at many levels. Just a transcription level is there. When primary transcript, pre-mRNA, immature mRNA forms, then, and even not that. After that, when we do the splicing or the processing thing, at that level also we can regulate it. When we transport that mRNA from nucleus to cytoplasm, and then translational level. But in eukaryotes, prokaryotes, the transcription and translation is simultaneous, so there is nothing like that. So here we talk about, right? Here we talk about certain what? We are going to talk about the genes here. Constitutive genes are there, non-constitutive genes are there. Constitutive means which are always active, their expression is always required. They are the housekeeping genes, means we always need the polypeptide that they are making. So they are expressed constantly because their products are constant needed for cellular activity, right? Because their product is constantly needed. Then, जैसे genes for glycolysis, genes of ATPase enzyme, non-constitutive, smart or luxury genes. We don't need them always. Right, their expression is required in some specific situations only. So they are switched on or switched off as per their needs, right? So if you have to switch on something, just say, let's suppose if the light is switched off, right, then I will do what? By default it is off, I'll switch it on. But if by default it is on, I will switch that off, and by default if it is off, I have to switch that on. So if it is off, right? Just say, let's suppose it's off, right? It is off. I have to switch it on. This is the inducible operand. Yeah, inducible way. You are inducing it, right? It is like that now. So inducible means switched on. And how will it be switched on? In the presence of inducer. And it is required for the functioning of gene activity. But when it is the repressible, by default it is on. You have to switch it off. Repress means to stop. Induce means to start. That's what you have to remember. So these are the two things that we have to discuss in their expression. And basically, what exactly do we need to talk about? We need to talk about the operand concept. So when you talk about the operands, they are in the case of prokaryotes only. You do not discuss them in eukaryotes. Number one, Jacob and Monod, they studied the E. coli. They found it there. They explained it. Number two. Number three, what exactly is the operand? It's again the segment of DNA. That segment of DNA, which is having one regulator gene, the promoter is there, the operator is there, and that many structural genes. So it's a kind of group of genes. Have a look. In 1961, year is important definitely. Two French microbiologists, Francis Jacob and Jacques Monod, they proposed a mechanism called operand model for regulation of gene regulation in E. coli. Again, PYQ, scientist name, year, everything. Then, what exactly is the operand? Told you clearly, it is the unit of bacterial gene expression, right? We'll define it in different ways, right? So, it is the unit of bacterial gene expression. What is it? It is the unit of bacterial gene expression and regulation, which includes, it is a unit of bacterial gene expression and regulation, right? Which includes structural genes and regulatory sequences recognized by regulatory gene product. I am repeating this again. Bacteria, it is a unit of bacterial gene expression and regulation that will include structural gene, regulatory sequences, and there are some regulatory gene products. Then, all genes in an operand are expressed as a single unit. They have a single promoter and is required to initiate and regulate the transcription of all structural genes. So in operand, you do not have one gene, you have many, but all the genes are having single regulator, single promoter, which will control their expression. So how can we define it? This is how we can define it. That's why I told you, you focus on understanding. Wherever you lag, you tell me. PDF will be shared in the Telegram group, so notes will be provided. Don't worry. And moreover, all other things I have already mentioned, right, as well. So handwritten, pre-handwritten notes, even in the live class, whatever notes we have made, everything you'll get. Now, an operon is a part of genetic material, right? You can even join, guys, my official Telegram group, my even my personal Telegram group, Love Biology by Ambika as well. So here you can see an operon is the part of genetic material which act as a single regulated unit having one or more structural genes. So it's the part of genetic material or DNA having single regulated gene and one or more structural genes. So an operator is there, promoter is there, regulatory genes are there, and many structural genes are there. Is that clear? Is that clear? So there are two types of operon. This one is the part of your syllabus. One is inducible. Again, you have to induce. You have to start the gene expression. Normally, that cell is not expressing. Normally, that bacteria is not expressing that gene, right? You need to start it. So by default, the expression of that gene is off and we have to switch that on. So inducible operon, we discussed the lac operon in the E. coli. In our syllabus, we have negative regulation of the inducible operon. One by one, I'll explain. So repressible means it is in switch on state normally. It continues to synthesize and metabolize, right? When this metabolize, when the product is in its maximum value, then we have to switch off the expression of that gene. So it is the tryptophan operon of E. coli. So in inducible operon, what is happening? Normally switched off and begins to work only when the substance which need to be metabolized by it is present in the cell. So I'll give you the example. This is C. Right now I'll just stick to the NCERT part here. So remember, you are talking about the E. coli, right? Normally E. coli feeds on glucose, but let's suppose if in the culture medium, glucose is not there. Matlab normally E. coli feeds on what? Glucose. So genes, or I would say the protein or the enzyme for digestion of lactose are not required because it feeds on glucose only, right? So such genes, their expression is not there, not expressing itself, isn't it? They are not expressing itself. That's what you have to remember. Now what happened? This is the culture medium. In this culture medium, where your E. coli is, there is no glucose, but lactose is there. But lactose is there. So to metabolize, because E. coli has to live, so to metabolize lactose, it has to switch on the expression of certain genes. The expression of, I would say, lactose digesting genes. So what do we have to discuss here? We are discussing the inducible operon here. So we are discussing what? We are discussing the inducible operon here. Which operon are we discussing here? Inducible operon. Now, what exactly is the lactose? Lactose is a disaccharide, right? Lactose is a disaccharide having glucose plus galactose. It is also known as the milk sugar, right? Now what's going to happen? Everybody repeat. Say PPO-ZYA. Everybody just say PIPO-ZYA. Everybody repeat PPO-ZYA. Quick. It's PPO-ZY. It's PPO-ZY. I here, it means regulatory gene. Done. This P is the promoter. It is the promoter of this regulatory gene. Right? Of it, this regulatory gene. Okay. Here, P is promoter. So P is promoter to which RNA polymerase should bind. Done. O is operator. Right? O is operator. Just a minute. Let me close the door. To which the product of regulatory gene binds, and this is going to control the expression here. Right? One more thing. This promoter and operator are overlapping. Right? They are overlapping. This Z, Y, A. These three are what? These are the structural genes. What are they? These three are the structural genes. So Y code for beta galactosidases, and lactose itself, because it is also referred as beta galactoside. Y codes for permeases and A codes for trans-acetylases. Am I clear? Yes? Am I clear? So P here, I here means regulatory gene, right? P is the promoter of this, so it is PPO. This P is promoter to which RNA polymerase should bind. O is operator to which the product of regulatory gene binds. P and O are overlapping. Z will give you beta galactosidase. This is permease. This is trans-acetylase. So this complete slide is containing all the previous year questions. Right? All the previous year questions are there. So noted? So noted? So this is what we have. So in the default state, what is happening? Right? When there is no glucose in the medium, right? So what is happening? As I said now, PPO, Z, Y, A. So no lactose in the medium. When there is no lactose in the medium, so what exactly is happening here? See, this regulatory gene, it is forming mRNA. This regulatory gene is forming mRNA. That is forming the protein. Actually, students, this is a repressor protein. Matlab it is repressing it. This is a repressor protein. Which protein it is? Repressor protein. And this protein binds to operator. This protein binds to operator, due to which RNA polymerase can't bind to promoter. So by default, there is no transcription and translation. There is no transcription and translation. So no transcription and translation will occur here. Are you getting my point? So that is going to happen here, right? That is going to happen here. mRNA repressor protein binds to operator. So here you can see, so here regulatory gene forms repressor protein. So it is the negative control of, या negative regulation of inducible operon. So it is the negative control of inducible operon, negative regulation of. So if repressor forms activator, which it can do, then it is the negative, sorry, then it is the positive, then it is the positive regulation of inducible operon, or positive control of inducible operon. So this part is not in our syllabus. This positive control is not in our syllabus. Negative control is. So it's not like that ki only positive control is there or negative cannot occur. Both are there, but in our syllabus only negative regulation is given. So now look at it. If regulatory gene, sorry, if this mRNA is forming a regulatory protein and if it is binding to the operator, so, and because promoter and operator, they are overlapping. So if here you have this repressor, so obviously my polymerase cannot come to promoter, it cannot bind to promoter, it cannot start the transcription here. So what should be our agenda? We have to remove this operator, this repressor from the operator, right? So who's going to help here? Lactose itself, and to be more precise, it is the allolactose that acts like an inducer. An inducer, right? This itself is going to start it, right? So I'll tell you what exactly is going to happen here, then I'll take your doubts. Okay, so let's see again. As I said, PPO ZYA, right? So it is beta galactosidase permease transacetylase. I'll discuss their function later. So now if, see, this is your repressor. I don't want this repressor to bind there. Okay, I don't want this repressor to bind there. So I need somebody to attract it, to free this operator side so that my polymerase can come to promoter. So it is like what? Right? It is like what? Like, let's suppose you want to break the relationship of two people. You are planting a third person so that this relationship can break. Same. So here, to be more precise, this allolactose will act like an inducer. What will it do? Right? It binds to the repressor. What will it do? It binds to the repressor and will stop it to bind to operator, right? So this is the RNA polymerase. So then RNA polymerase will bind to promoter, and the transcription translation of these structural genes start. Transcription translation of these structural genes start. Am I clear? This will start. Am I clear? This is going to start here. That's the case. So they will form obviously polycistronic mRNA and then the enzymes will be obtained. This is how it is work. So because here the regulatory gene is forming the repressor, that is why it is the negative control of inducible operand. So do we have positive also? Yeah, but that is not the part of our syllabus. Okay, that is not the part of our syllabus. Is that clear? So transcription translation will occur. Now see this. Z form beta galactosidase. Now when this beta galactosidase will form, it breaks lactose to glucose galactose, है ना, to glucose and galactose. So this will get energy, yes or no? Then the second case is this Y, it is forming permease. It is forming what? It is forming permease. So permease allows the lactose to enter in cell, or I would say make cell permeable for lactose entry. I know for lactose entry. Then the function of A is not that clear, but it, yeah, as you can see, the name is saying trans-acetylase. So it transfer acetyl group. It basically helps in detoxification. If in simple words I have to quote it, so it helps in detoxification. It helps in detoxification. Is that clear? It helps in what? It helps in detoxification, right? So Y will form beta galactosidase that will digest the lactose, will break it into glucose and galactose. Permease. Y will form the permease, and that permease allow the lactose to enter into the cell, make the E. coli membrane permeable for lactose. And A is the trans-acetylase, which will transfer acetyl group in different compounds. Basically, it is for the detoxification. Now, here the question arises that, ma'am, how can lactose act like an inducer, right, if permease is required for its entry into the cell? And yeah, it is a fact. If permease is not there, if let's suppose there is a question that if permease is not there, then the lactose cannot enter in E. coli. But how this lactose, which is forming then allolactose, is entering? It's very simple. Some expression of permease is always there in E. coli, very less expression. And due to that very less expression, what's going to happen? Even due to that very less expression, what is going to happen? Some amount of lactose will enter and then it will activate the system. So it is like that. So promoter and operator regions are overlapping. Product of regulatory gene interacts with operator. Right, so it is in absence of inducer where repressor is joining to the operator and no transcription translation. When the inducer is there, right, it will bind with repressor, it will inactivate it, and then you know that RNA polymerase will bind to promoter. Always remember, it will bind to promoter, not to operator. So polycistronic lac mRNA will form, beta galactosidase, permease, trans-acetylase will form. So the diagram has to be very clear. So it is the negative regulation. So inducible operand remains in switched off condition. It begins to work when the substance to be metabolized with is present. Then lactose, you know that milk sugar, it's a disaccharide, right? So beta galactosidase can break it into galactose and lactose. So we need beta galactosidase to digest it. Now, in inducible operand, we have the negative regulation. Why? Because here the LAC-I or the I gene, it produces a repressor. So it is the negative regulation. If I gene will form a activator, then it will be the positive regulation. So here you can see it produces a repressor protein that binds to the operator gene and stops the operator gene functioning. Right? So repressor has two binding sites, students. It can bind with the operator gene. It can bind with the inducer. So promoter gene, it's the site for the binding of RNA polymerase. Then operator, right? Both are overlapping. So it directly control the synthesis of mRNA over its structural gene because it will interact with the product of regulatory gene. So in switched off case, repressor is present on operator. So promoter and operator regions are overlapping. So there are 3 structural genes, students, right? So they will form polycistronic mRNA that will form the polypeptide. So Z will give you beta galactosidase, permease, and then transacetylase. So it protects the cell to build up from the toxic material, right? Formed by beta galactosidase enzyme and acting on the sugar other than lactose. So it will help in that. So all these 3 are required for the metabolism of this lactose. So when the sugar lactose is added to the culture, few molecules of lactose will enter in bacterial cell. Why? Because the expression of permease, a little bit of it is always present. Even the operin, if it is not working, still permease is there in E. coli. So that's why some amount of lactose can enter. Then few lactose molecules are then converted to active form allolactose, which act as an inducer and binds to the repressor. So when inducer and repressor complex forms, it will not be able to join with the operator, then RNA polymerase will bind with the promoter and the transcription, guys, it will start. So this is it. Then this is the repressible operin. That is the only example. The example that we have to study is tryptophan. So tryptophan operin in E. coli is the example of repressible operin, again negative regulation. So by default it is switched on, right? By default it is switched on. So we have to switch it off. So it is not in the right syllabus, but still you should have the idea here, little bit of it. So there are 5 structural genes, A, B, C, D, E, basically E, D, C, P, A. So they are forming these enzymes that can help in the tryptophan synthesis. So here regulatory gene is again forming the repressor, so that's why it is the negative regulation. But the repressor that it is forming here, right, this repressor is inactive. It is not capable to bind to the operator and to stop it. So it needs what? It needs the tryptophan, because tryptophan is itself in the high concentration. So this tryptophan, it acts like a co-repressor. So when inactive repressor and this co-repressor binds, the repressor gets activated. So it will bind to the operator. Then the RNA polymerase will be removed and the process, this transcription translation will stop. So that is it. The last topic that we are going to discuss is Human Genome Project. I feel so good when I finish a chapter and when I feel like that I have justified everything. So last time in the human reproduction also, right, last time in the human reproduction chapter also, we discussed it in detail, but as I said, there are so many extra things that we can discuss there. But last May, where implantation and all the things were there, we just focused on the NCERT part. The paper will come from that only. Even human health and disease, I know, that was the mishap. Principles of inheritance and variation. So there only pedigree analysis part is left. Rest everything is done and dusted. And today MBOI in detail we have discussed, each and every point we have covered. Even I have covered the NCERT, like DNA polymerase uses 2000 base pairs per second. Everything we have covered, right? So now we will talk about the Human Genome Project, very important it is, and then we'll give you a little bit of idea of the mutation. Gene mutation mein you have already studied. I'll just talk about the frame shift mutation here, that's it. And then this will be finished. Okay, so I hope you are enjoying it. So now we are going to start our last topic. So let's get started. So firstly, just have a look. Okay, so I feel so good when I justify that chapter. Okay, done. So it will go for around, I would say, 10 hours, 40 minutes or the 11 hours. So as I said, 1.5 hours is for the break. So rest you can use for the revision. Okay, so we will discuss the HGP. Just allow me 1 minute. Chaliye, so let's start, guys. So Human Genome Project. Why? It is very important. See, again, it's the NCERT part that we have to cover. It was a very ambitious project where what was done, the sequencing of human genome, like which bases are present on which chromosome, yeah, which DNA, like which genes are present on which chromosome, their sequence, their position, everything was considered. Okay, so it was launched in year 1990 and it was called a mega project, right? Financially, right, if I talk about the finances, a lot of money was required for this particular right, for this particular project. Just say if I have to tell you this part here, just allow me 1 minute. So the requirement was simply कि human genome, it is having 3 into 10 raised to power 9 base pair approximately. We know that 3.3 into 10 raised to power 9. So cost of sequencing of one base per base pair was $3. So it was approximately for the 9 billion US dollars, right? So if the, like, let's suppose if from this, from the human genome, if we take out all the information, the bases and all, so you know what is required in the typed format, right? The books are required. So basically that book, each book will contain 1000 letters and each book will contain 1000 pages. Matlab 1000 pages, each page will have 1000 letters and such type of 3300 books are required. So imagine it was that immense. Okay, so the enormous amount of data expected to be generated were also, it was necessitated to use of high-speed computational devices, because you know that humanly it's not possible to take a record of each and everything. So imagine 3300 books with 1000 pages, with each page is having 1000 letters in it. Okay, so with this a new science emerged, which is bioinformatics, right? Even if you want to go for it, you can. It takes time, but then it will be very, right, then this, like, it will take time, you'll take some time to get settled in this career, but then, right, you'll be able to earn a lot of money. So bioinformatics, it emerged. Then what were the goals of this HGP? To identify all the approximately 20,000 to 25,000 genes in human DNA. It was the goal to identify 20,000 to 25,000 genes in human DNA, number one. To determine the sequence of 3 billion chemical base pair that are making up human DNA, to store this information, to improve the tools for data analysis, to transfer this technology to other sectors and industry, and to address all the ethical, legal or any social issues during the project if they may arise. Okay, so when it comes to this, it's a 13-year project coordinated by US Department of Energy, National Institute of Health, right? The Wellcome Trust US became a major partner there, and then Japan, France, Germany, China and others. So the project was completed in 2003. Imagine 1990 started, completed in 2003, right? So knowledge about the effects of DNA variation among individual, it can lead to the revolutionary new ways. Why? To diagnose the disease. Just say, let's suppose there is a healthy individual. If we sequence the data of that individual, there is a person with some disorder. I can compare their data, right? I can compare their sequencing. Then I can figure out where actually the difference is. So we can figure out that, okay, that is the point. Yeah, that is a gene where the mutations are there. That's why this disease is occurring. So that's why this project was very, very, very important. Is that clear? So not just humans here, you know, many non-human model organisms were also there. So question can come from this part, like bacteria, yeast, C. elegans. It's a free-living, non-pathogenic nematode. C. elegans in short form. So it is Caenorhabditis elegans. So yes, you should know this. So even the bacteria, yeast, C. elegans, Drosophila, plants which will include rice and Arabidopsis, they have also been sequenced. So this information is again important. Then next to it, what was there? Methodology. So again, that is again a PYQ. One way was that, see, one way was this key, let's, you know, copy the entire human genome. That will include coding, non-coding sequences, everything. So that comes under sequence annotation. Now we know that DNA that forms mRNA, that mRNA expresses itself, they form the proteins. So expressed sequence tagging, ESTs, where to identify all the genes that are expressed as RNA. Matlab whatever RNAs are there in the cell, that RNAs, what do they show? That gene has expressed it, gene has formed the RNA. So one technique was that sequence that RNA only, and other was sequence annotation to sequence the whole set of genome. That will contain coding, non-coding, everything. Understood? Now the next part, for sequencing, total DNA from a cell is isolated, right? The complete DNA for sequencing, the complete sequences, then converted into random fragments of smaller sizes. And then, see, this part is related with the biotech as well. So usually I discuss HGP and DNA fingerprinting both there, but HGP still we will discuss there, but DNA fingerprinting in the biotech. So they just use the enzyme, they cut the DNA into fragments. Then they use that fragment because it is not possible to, make the copies of the entire human genome. So they cut that into fragments. The fragments, they fix that in the suitable host or a suitable vector that will multiply, that will give us the copy of that fragment. So vectors that were used was BACs, YACs. Again, important PYQ because they can accommodate the fragment of large size. So BAC stands for bacterial artificial chromosome, and YAC stands for yeast artificial chromosome. Got that? So these were the two that were used in the Human Genome Project. Again, a PYQ, right? So cloning resulted into amplification of each piece of DNA. Like we got the multiple copies, so then can be sequenced with the yeast. So BAC and YAC, told you already, they are the PYQs. But why them? Because they can accommodate the DNA of large size. Then the fragments were sequenced using automated DNA sequencer because that is not possible to write each and everything. So automated DNA sequencers were used. And who made that technique? The another PYQ, Frederick Sanger. So that method was developed by Frederick Sanger. So these sequences were then arranged on the basis of overlapping regions present in them. So all the chromosome, the DNA which is present on the same chromosome, that is having the overlapping genes, some overlapping sequences. Even if you are going to cut that into fragments, right? Even if you are going to cut that into fragments, right? Then still, even after cutting that into the fragments, like in different fragments, now we have to check that whether that piece of DNA or the gene belongs to the same chromosome or not. So we will see that just say the genes or the DNA which is on same chromosome, they will have some overlapping regions. So we can just take out that fragment, we can just arrange that overlapping regions, and we can just mention the sequence there. So basically the point is key overlapping regions, they are the portions of DNA where two or more genetic, because word is itself explaining that it is overlapping region. So two or more, one second guys, sorry for this. So where two or more genetic elements, they share the common same nucleotide. Like suppose this is a chromosome, the DNA is present on the same chromosome, you are cutting it, right? You have different fragments. Then you want to check whether it belongs to the same chromosome or not. You will find out some overlapping regions. Are you getting my point? You are going to find out some overlapping regions. That can be a gene, exon, some regulatory sequence, anything can be there. Okay? 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 Done? So this required generation of overlapping fragments obviously for sequencing. Then alignment of these sequences, again humanly was not possible. So specialized computer-based programs were there, and these sequences were then subsequently annotated, right? Then they were annotated that this particular sequence belongs to this chromosome, that chromosome. Is that clear? Is that clear? Sure? Are you sure? Any doubts? Any doubts here? Sure? So annotation is just like the labeling, the identification and labeling, okay? Okay, it is just like the identification and labeling of these regions in the DNA sequence. You are identifying them, labeling them. This belongs to this chromosome, this belongs to that. So I believe this part is clear. Then after moving, after this, what was done? So sequence of chromosome 1. Matlab the project was completed in 2003, but the sequence was completed in 2006, right? So this was the last of the human 24 chromosomes. So this is purely NCRT. Each and every line is important. So you know that we have 22 autosome, X and Y chromosomes are there, right? So another challenging task was to assign these genetic and physical maps on the genome. So this was generated using information on polymorphism. Poly means many forms of restriction endonuclease recognition site. जैसे our DNA is having the sites which can be recognized by restriction endonucleases, the special type of endonucleases which make the cuts at specific site. So we used the information on the polymorphism of that site. The repetitive DNA sequences were noted. These were named as microsatellites. So they helped, right? They helped to arrange them. Is that clear? So read it again and then tell me what is the point which is not clear to you. Quick. Yes, everybody, quick. We are about to finish it, so please be quick. Done? Done, students? So here what is said that how they were mapped by using the information on polymorphism of restriction endonuclease recognition site. Polymorphism means different forms in different individual. How can this occur? Obviously due to mutation. Let's suppose my and your recognition sites, they were same. No, they changed. Why? Because of mutation. So that mutations will help us to, you know, differentiate as an individual, because my recognition sequence is different, your is different, because there is a, you know, some change that can occur due to the mutation, right? And there are some repetitive DNA sequences as well. Just we are saying microsatellites, right? So these microsatellites are what? These are the short tandem repeats. Short tandem repeats matlab short you understand na? Tandem matlab which is repeated, right? In a sequence something is repeated. Just microsatellites mein you will see this is 1 to 6 base pair sequence which is repeated many times, right? So they are also polymorphic. They are also polymorphic, right? So every individual will have different number of repeats. Just let's suppose if I have to give you the example, let's suppose if it is GATA, it is the sequence. This is the repetitive DNA sequence, right? So it is having, you know, less base pair, bases or base pair. So it is GATA. So in my case, GATA, maybe it is repeated a number of times. In your case, it can be different, right? It can be different. So these microsatellites, they are highly polymorphic, students, and they are basically non-codings, right? They are non-coding regions. They are not giving you any information for the protein, but help to differentiate one individual from other. There's a GATA, if I'm saying that n number of times if it is getting repeated. So in different individuals, it will be different times that it will be repeated. So in forensic sciences, they play the role, right? Right? So we also talk about the mini satellites that we will discuss in the DNA fingerprinting, that are actually VNTRs. What is the meaning of VNTRs? It is variable number tandem repeats. Variable number tandem repeats. So in this case, what you people will see? In this case, you'll see that around 10 to 30 nucleotides, they are repeated. Okay, 10 to 30 nucleotides, yeah, 15 base pair. You can say that if let's suppose there are 10 to 30, it can be a 10 to, not 10 to 30, it is more, it is 10 to 100. So you can say, let's suppose 15 base pairs are there that are repeated 30 times. So again, it is unique to every individual, right? It is different in every individual. Am I clear? Right? Am I clear? And where do you find these? You will find them in near telomeres. What are telomeres? You know that they are the ends of chromosome. Okay? They are the ends of chromosome. Am I clear? Am I clear? So all the individual will have different number of repeated units, right? So that's why it is going to help. So microsatellite, they are short tandem repeats. Mini satellites are variable number short tandem repeats. Done? Done? So in DNA fingerprinting, mainly it is the VNTRs, Okay, so mutations can cause that. So in genetic mapping, they play the role, isn't it? So that's how it works. Next is salient feature. Human genome contains 3164.7 million base pair. Important. Each and everything is important for the CBSE board as well as for the NEET aspirant. Average gene is having 3000 bases, but size can vary greatly. So again, next PYQ, largest human gene is for the dystrophin, right? Gene being dystrophin at 2.4 million bases. The total number of genes, which was estimated at 30,000, it was much lower than, this is the total number of gene. Estimate was of 80,000 to 1,40,000, right? And almost all 99.9% nucleotide bases are exactly same. There is a difference of 0.1%, and that thing evolved DNA fingerprinting, also known as DNA typing, students. Right, that helped in DNA fingerprinting or DNA typing. So the function of over 50% of the genes, right, even of the discovered gene is, unknown. It is not clear, and less than 2% of the genome will code for the protein. And repeated sequences, they are actually making up the large portion of our human genome. So this is it. So then next is the so repeated sequences. What are they? They are the stretches of DNA that are repeated many times. I gave you the example of microsatellite 1 to 6 base pair, mini satellite 10 to 100 base pair. Let's suppose 15 base pairs are repeated 30 times, like this. So it is unique to every individual, right? So they are not having any role in giving us the information for the protein synthesis. So sometimes they are repeated 100 times, 1000 times. So, but they shed light on the chromosome structure, about its dynamics, what kind of changes occurred, and the evolution. So chromosome 1 is having most genes, 2968. Why? The Hollandric chromosome having the minimum 231. Then scientists have identified about 1.3, imagine in our genome, 1.3 million locations where there is a change of the single nucleotide. So it is pronounced as SNPs, right? SNPs, SNPs. This stands for single nucleotide polymorphism. Remember, it stands for single nucleotide polymorphism. And students, when we discuss SNRNPs, then that was single nucleotide ribonucleoproteins. Remember, in spliceosome we discussed, and it is what? It is pronounced as SNRPs. So that is SNPs, right? Do not get confused, right? So this information revolutionized the process of finding chromosomal location and to trace the human history as well. So when we talk about SNPs, just see, all the DNA is same at this position only. The bases, they vary. So that is it. This is all about this topic. Finally, yes, we are able to, right, finish it. So next lecture, next session, it will be on, yes, next class is on Friday, on Wednesday. That is, one second, Mall Bio, Game of NEET, GON 3.0. Let me make the notes as well. So it is all about this particular chapter. It is done and dusted, guys. So next chapter we'll have on, just say now, so it's first now. We'll have the next chapter on 3rd of December, sexual reproduction in flowering plants. Then I have to check the schedule of others. Hopefully the same week. Yeah, hopefully on same week, on Saturday, hopefully I'll take the second chapter, okay? That is Saturday I'll go for biotech, okay? Biotech in one go. Biotech in one go, the evolution, the applications and the processes both. Then next week, we'll go for which chapter? Evolution. Then that is a lengthy chapter that will again take 10 to 12 hours. Then this month, hopefully, then we will go for microbes. Now, ecology. We'll start directly with the ecology. Okay. So, eco maybe we'll take 2 chapters together, 1 chapter singly. And microbes also, you know that is also a very simple chapter. So, we'll be left with what? So basically in December itself we will finish the 12th syllabus. That's what we can see. We can even finish the 12th syllabus in the December month. है ना? No, Harita, they can be in the night. See, if I would have started this at 5. So 5, 6, 7, 8, 9, 10, 11, 12, 13. Then you know na, matlab till 4 or 5, right? Till 4 or 5, we would have studied that. So why? Matlab, even if I'm starting at 2, how does it? How does it matter? Okay, how does it matter? Okay. Chalo, so this is all. So I'll share the notes in the Telegram group. So all the best. So see you all on 3rd of December. So the next chapter is sexual reproduction in flowering plants. So December May, let's finish 12 syllabus. So January May we'll even increase the duration because number of chapters are more in 11th. So let's try to finish it by February. So we will finish the syllabus till then. Then March and April will be for the revision only. Okay. So take care till then. Bye-bye.